perfectly audible and visible to everybody? Hello, am I audible? Am I audible? No, I'm not. Yes, I am. So, hi everyone, am I audible to all of you? Am I audible? Hello. Do let me know in the chat section, am I perfectly audible and visible to all of you? Yes, please. Do let me know in the chat section, am I perfectly audible and visible to all of you? I believe I am perfectly audible and visible. Okay, so how are you everybody? Excited for the session? How are you everybody? Excited for the session? Voice is low. Is it so? Is it so? All good? Okay, great. So I am going to start the session and I hope you all are excited for these marathons because in 15 marathons, we are going to complete your syllabus. So in today's marathon, right, we are going to target the human physiology, structural organization in animal cell cycle and the biomolecule, right? So I'll be picking up the chapter that you want me to teach first, okay? So I am going to start with the chapter that you people want me to start first. So tell me, which chapter you people want me to start? Yes? Which chapter you want me to start? Whatever chapter you people will say I am going to start with that particular chapter. So do let me know in the chat section quickly. Everybody, quickly. Yes, so today's marathon is for 100 marks. Literally, it is for 100 marks. You know that human physiology, structural organization, animal kingdom, cell cycle is there. And minimum 25 questions. Minimum 25 questions are expected. Minimum 25 questions. Okay, and of course, the basically from today's session, 25 to 30 questions can come. Okay, so you have to be very attentive and trust me, I will pick up the most important topics from each and every chapter because you know that it is biology, right? It is biology and here we have so many things that we need to understand, that we need to learn. So we are going to pick up the most important things, most important chapter. But before starting the session, I want everybody to hit the like button. Quickly, do it. Everybody hit the like button, quick. Whatever chapter you people will tell me, I'll start it first. Trust me, right? Today, I don't have any plan that I'll start with this, I'll start with that. No. Whatever you people are going to tell me, I will start with that. But first of all, hit the like button, everybody. Quickly. Hit the like button, right? Mark your attendance. If you are new to our channel, just subscribe our channel and then let's start the session. And tell me which chapter. Okay, animal kingdom, human physiology, animal kingdom. Acha, biomolecule, it's for zoology. It is the zoology only today. So tell me, biomolecules, structural organization. So quickly, everybody tell me. Everybody tell me. Everybody tell me. I'll go as per majority. I'll go as per majority. Quick. Structural organization, animal kingdom. Acha, you want me to take the poll? I wish I could. Let me see. Yes. Okay. So I have asked my team, but let me see here. Animal kingdom, structural, biomolecule. Animal kingdom, structural, biomolecule. Okay, so how many of you want me to start the animal kingdom first? With fire emojis, please tell me. How many of you want me to start the animal kingdom first? How many of you want me to start the animal kingdom first? With fire emojis. Quick, everybody. Quick, everybody. With fire emojis. Animal kingdom with fire emojis. So I'll start it. Animal kingdom, biomolecule is also there. Arey, human physiology, so we have to complete. And you know the last series, top 50 concept, I have picked up the most important concepts there. Don't worry, we'll discuss everything here. We are not going to leave anything. Don't worry about that. Don't worry about that. Quick, everybody, quick. Yes? Quick. So So I think when it comes to biology, students are having many things, many demands. Okay, so they are like, some are like biomolecules, some are like this, everything we are going to cover. And let me tell you, students, I have completed the entire syllabus on Unacademy NEET English channel. Okay, entire syllabus on Unacademy NEET English channel. So don't worry about that, right? Even if you want me to start the human physiology, it will also be done, and that is the major part. That is the major part, right? So if you want to copy the notes, you guys can, right? The important things I have already added. Don't worry, we will manage. Okay, animal kingdom. Chalo. So let's start with the animal kingdom first, because majority of students are saying that they want the animal kingdom first, and animal kingdom is going to give you 4, right? Animal kingdom is going to give you 4 questions. So let's start with the animal kingdom, then let's go for the structural organization. Okay, and if you are concerned about the biomolecule, don't worry, everything will be discussed here on this channel. Okay, okay. And do not forget that I am also posting the short videos. I don't know whether you have seen it or not, and they are going to be very important, very useful for all of you. You people can see this, right? You people can see this. Plant growth and development in 20 minutes, right? Cell cycle and cell division in 20 minutes. Okay, so I'll be covering the most important chapters here in 20 minutes. Okay, but all I need is your support. Okay, so if you want me to take such sessions right after the class, do let me know in the comment section. So as majority of students are saying animal kingdom and structural organization, so chalo, let's get started. Okay, let's start. So I'm ready with everything, right? I don't have any problem. Whatever chapters you people will demand, will go as per that. Okay, we will go as per that. Okay, chali. And today, in a very simplified way, we will proceed. So the first chapter that we are starting is, and I think from so long we didn't revise the animal kingdom. So let's start the animal kingdom. So do you know the kind of questions that can come in your paper? Do you know the kind of questions that can come in your paper? But as I said, right, minimum, you guys can expect minimum 100 marks. 100 marks is not maximum, it is minimum. You can expect at least 30 questions from this particular session only. So basically 120 marks. And next session will be for the complete botany. Okay, so in 4 marathons, I'll be completing, I am going to complete your syllabus. And of course, right, from these marathons only, if you will watch them sincerely, if you revise the questions later on, definitely you people can score 300 plus marks. Right, 100% you people can score, right, 100% you people can score 300 plus marks, but you just need to stay tuned. Okay, so I have increased the volume as well. I hope now I'm perfectly audible to everybody. So now voice is fine. I believe it's fine now. Chali. So let's start the animal kingdom. So when we talk about this particular chapter, right, let's revise the few basic things first. We know that there is a scientist whose name is R.H. Whittaker. If you know it, do let me know in the chat section. I'll start with the most important points. R.H. Whittaker, what he gave us the 5 kingdom classification system. What he gave, my dear students, he gave us the 5 kingdom classification system. So when we talk about these 5 kingdom classifications, right, what do we discuss? Yes, we talk about kingdoms we are talking. So we talk about kingdom Monera, right? We talk about kingdom Protista, and then it is the fungi. Then here we have kingdom Plantae, and then we have kingdom Animalia. This is what we have, yes or no? Everybody, that is also a PYQ, right? I am not discussing anything which is not important. So right, so I will pick up all the PYQs here, you don't worry. So what I just said, I am talking about the R.H. Whittaker, and I said 5 kingdom classification was given by him. So that is again an important question. So we have kingdom Monera, we have kingdom Protista, fungi, Plantae, and Animalia is there. Monera, Protista, fungi, Plantae, Animalia. Everybody, repeat. But is there any issue with the voice? Is there any issue with the voice? Yes. Is there any issue with the voice? No? I think it's fine, right? Okay, so we have the 5 kingdoms, right? So when you talk about the kingdom Monera, you know that here you will see prokaryotes, prokaryotic organisms. Basically, here you will see bacteria. Now, these 4 kingdoms, they are for the eukaryotic organisms. And what we have to pick? We have to pick kingdom Animalia. We have to pick kingdom Animalia, right? What we have to pick? We have to pick kingdom Animalia here. So when you talk about this particular kingdom, of course, here all the organisms, they are going to be multicellular, right? They are going to be multicellular, and here all the organisms, they are eukaryotic for sure. So in animal kingdom, right? In animal kingdom, without any doubt, this is what you have to keep in your mind. In animal kingdom, my dear students, all the organisms, they are multicellular. What are they? They are multicellular. Now, I'll be writing the name of phylum here, right? So this is the kingdom. Okay, you know that we have the 7 broad categories. Okay, what do we have? We have the 7 broad categories, 7 essential categories. Let's have a quick revision of that as well. They say we have keep, this is the trick, keep pot clean or family gets sick. Keep pot clean or family gets sick, right? Keep pot clean or family gets sick. So K stands for kingdom, P stands for phylum, right? C stands for class, O stands for order, F stands for family, G stands for genus, and then S is here for the species. This is what we have. So kingdom, phylum, class, order, family, genus, species, that is what we have. So here in this chapter, we are talking about the kingdom Animalia. Then we have to discuss all the phylums. And when you talk about phylum Chordata, in phylum Chordata, we have to discuss up to class level. What we have to do? We have to discuss up to class level. This is what you have to remember. Okay, this is what you have to remember. Now, when it comes to the phylums, yes, when it comes to the phylums, so in the case of phylums, you have Phylum Porifera. Everybody please repeat the names with me in the chat section. Phylum Cnidaria is there, which is also known as Phylum Ctenophora, right? Phylum Cnidaria, which is also known as Phylum Ctenophora. Phylum Coelenterata. Wow, Ambika, wow. So Phylum Porifera, Cnidaria, then next is your Ctenophora, then next is your Platyhelminthes, right? In one sequence, please everybody just revise these names again, right? Complete your chapter in this way that today it is the last day to revise it, right? Today it is the last day to revise it. That is how you have to complete the chapter. So we have the kingdom Porifera, we have the kingdom Cnidaria, Ctenophora is there, Platyhelminthes Aschelminthes, then we have Annelida, Arthropoda is there, right? Mollusca is there, Echinodermata is there, Hemichordata is there, and then Chordata. These are the 11 phylums that we have. Porifera, Cnidaria, also known as Coelenterata, Ctenophora, Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, Chordata. So how many of you know these names in this one sequence? How many of you know these names in this one sequence, right? If it is a yes, do let me know in the chat section with some fire emojis that yes ma'am, we remember that. If you don't, just revise it with me, because today we have to complete everything from the class 11th zoology, so you have to revise the things with me. And if there is something which is new for you, mark that topic, be attentive, and later on after the class, right, do revise it. So everybody, Porifera, Cnidaria, Ctenophora, Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. That's what we have. Okay, that is what we have. Now, when we talk about these phylums are having different kind of organisms. Like if you will see, if you see around, you know that different type of organisms are there. Animals are there. We are humans, right? Cat, dog, spider, lizard, ant, right? There are so many different type of organisms. If you talk about the aquatic organisms, right? Fish is there, aquatic snakes are there, right? Other than that, you know that hydra, jellyfish, these are the different kind of organisms. So there are different kind of organisms in the nature, right? There are different kind of organisms in the nature, but we all are. And I should be very specific here, different kind of animals are there in this nature, but they all are placed in kingdom Animalia, right? They all are placed in kingdom Animalia. So basically we all are so different from each other. Like compare a rat with human, right? Still few similarities are there, but still we are different. Compare a mosquito with a human, we are so different from each other, but still we are the part of same kingdom. So there must be a reason, There must be a reason, right? So when you talk about that, so here what we have to see that there are certain bases of classification. What do we have, people? We have certain bases of classification. What do we have, people? Just tell me. What do we have? We have certain bases of classification. So when we are saying bases of classification, these are the characters that has to be considered, right? These are the things that we have to consider first, right? And on the basis of that, we place the organism, we place the animals in different phylums. The first part clear? The first part clear? So there are, what do we have here? The basis of classification. So when we talk about these bases of classification, so on the basis of that, we put the different animals into different phylums. So the very first thing is level of organization. What we need to discuss first, my dear students? Level of organization. Yes, what we have to discuss first, Biche? We have to discuss the level of organization first. Okay, so when we talk about the level of organization, so under this basis of classification, we have so many things. The first thing that we have to start is the level of organization. So when you talk about the level of organization, देखो, if it is a unicellular organism that we do not have in animal kingdom, so you know that here, what will be there? You will talk about, when you talk about a unicellular organism, you have to consider the protoplasmic level. You have to consider the protoplasm there only, protoplasmic level only. Okay, when you are talking about what? Unicellular organism. But we are talking about the animal kingdom, and here all the organisms are going to be multicellular. Basically, in detail, so we discuss so many things, the different type of classification, right? Right, but now because you know that, right, our exam is there and less than 1 month is there, so of course we have to stick to the NCERT. So here we will go as per NCERT. Okay, we will go as per NCERT. So when you consider the unicellular organism, you are talking about the protista. You're talking about the protista, but here what do we pick? We'll pick up from this part. We'll pick up from cellular level of organization, tissue level of organization, organ level of organization, organ system level of organization. That is what we have to consider. Okay, so cellular level, tissue level, organ system level. So cellular level, here you will just have a multicellular body. What you people will see? You will just see a multicellular body. So basically that organism is not just having the tissue, that organism is just having a multicellular body. And which phylum are we discussing here? We are talking about the phylum Porifera. That's why these names are in front of you, right? These names are in front of you. So from these names only, there will be many examples that you people are going to see. Okay, that you people are going to see. So first thing is the Porifera. Then there is the tissue level. So we know that, we know this basic hierarchy. We know that we have the cell. Cell together will form tissue. Tissue together will form organ. Organ together will form organ system. And then finally, what do you have? You have organism. What do you have? You have a complete organism. Okay, this is what we have. So after cellular level, tissue level is there, right? The organisms, they have the tissue level of organization here. So here you talk about the phylum Cnidaria, also known as Coelenterata, and phylum Ctenophora, right? Now let's move to the next phylum where you will see organ level of organization. And here, as per our NCERT and overall also, it is the Platyhelminthes. Okay, so in the NCERT, right, tables and theory part is there, no doubt. Mainly they consider the information which is given in tables. Okay, but if you ask me the accurate thing here, because in tables at some places confusion is there. So actually in organ, like in Platyhelminthes, it is just up to the organ level only. But when you talk about the organ system level, then you start from Aschelminthes to, yes bache, Aschelminthes to Chordata. This point is clear. Aschelminthes to Chordata. This point is clear. Everybody here in the class. So Aschelminthes to Chordata. So if you have any doubt here, do let me know. If you have any doubt here, do let me know. So that is why the name of the phylums is very important. So when you have the name of the phylums in front of you, right, and when you remember them, then of course you just need to understand the basic things about these bases of classification, and we will be able to answer everything. And trust me, 1 or 2 questions will directly come from this part, right? Then rest will be from the examples only, right? The rest will be from the examples only. And today, in a very quick way, we are going to revise everything. Trust me on that, right? Today we have to revise it very quickly. We'll just focus on the important things, right? When it comes to the examples also, we'll try to provide you the tricks as well, and I will just tell you the peculiar feature of a particular phylum so that you can revise it quickly. Okay, so that you can revise it quickly. Is that clear? Is that clear? Okay, so the next thing that we have to discuss here is Yes, after basis of classification, the next thing that we have to discuss is symmetry. Right, the next thing that we have to discuss is symmetry. What is the next thing that we have to discuss? Symmetry. So in the, like, here you are talking about the body organization, body symmetry. Like, there are certain objects which we can divide into 2 equal parts or 4 equal parts or 10 equal parts, and there are certain things which we cannot divide. Right, just say, do you like chocolates? Yes, people, do you like chocolates? Chocolates, right? This is my favorite is Kit Kat, right? I love Kit Kat. Literally, I love Kit Kat. I like Kit Kat. So when we talk about that particular chocolate, if you have seen, right, we can very easily cut it. Like, let's say it will have the 2 pieces. We can cut it, we can break it, we can distribute it. Right. There are, like, that is for, I think, 10 rupees or 20 rupees. I don't remember. There is one where 4 lines are there. So 1 line, 2nd line, 3rd line, 4 lines. So very easily we can distribute. Right. We can distribute. And now these, even Cadbury Dairy Milk. Okay. So here also you can easily break them into the pieces. Right? Take break. Kit Kat is there. So we have this. So basically you can divide it. Right? You can divide it. Very easily you people can divide it. This is what we know. Now when you are talking about the symmetry thing, they say asymmetry. Asymmetry. When we are saying asymmetry, means that organism is without any symmetry. Means any plane that is passing through the center of the body of that organism cannot divide the organism into 2 equal halves. Are you getting my point? Are you getting my point? Right. So when I'm saying asymmetry, when any plane passing through the center, when any plane passing through the center of the body, right, through the center of the body of that organism cannot divide the organism, right, into 2 equal halves. So doesn't divide organism into equal halves. Are you getting my point? So when you talk about this particular thing, so which phylum will show asymmetry? Porifera, right? The primitive, right? The primitive animals. And porifera means pore bearing organisms. What type of organisms do we have here, students? We have pore bearing organisms. Are you getting my point? And here also we have the exception. Do not forget about the exception as well. So exception is your cycon. Exception is leucosolania. Exception is cycon and leucosolania. They show radial symmetry. So when it comes to the symmetry, these are the kind of things that you have to remember when it is the NEET examination, that it is not in all the porifera. Not all the porifera, they show us the asymmetry, right? Not all the porifera show us the asymmetry, but mostly poriferans, they show us asymmetry, right? We cannot divide the organism into equal halves. This is the first point. Now, let's move to the second part. That is your, okay, radial symmetry. That is what? That is your radial symmetry. Next symmetry is what? Radial symmetry. So to understand this part, because now it's not the time to remember the definitions. It's the time to remember the actual point from where the question can come. So take the example of starfish. Just take the example of starfish. But am I too fast? I don't think so. Our chapter is lengthy. We do not have, like, it's not your subject physics where you know one concept, you have to solve the MCQs and it will be done and the question will come. It's theory. Each and every line is important. We have to discuss even the minor details. Okay, so I have to be fast. We don't have any other option. What say, guys? We don't have any option. I don't think it's too fast. It's fine. You can decrease the speed a bit. Right? Please try to understand, students. We have to finish these chapters in at least 10 to 12 hours. Okay, so I need your time. I need your time. So just take the example of starfish here. So this plane can divide the starfish into 2 equal halves. Plus here in starfish, even if I pass the plane like this, diagram is not that appropriate. Again, you will get 2 equal halves. If I'm cutting it like this, I'll again get 2 equal halves. So what am I trying to say? When it is the radial symmetry, any plane that is passing through the center can divide the body into equal halves. Are you getting my point? When it is, right, Biche Devanshu, when it is what? When it is the radial symmetry, any plane which is passing through the, right, any plane that is passing through the central axis can divide the body, into equal halves. Into what? Into equal halves. That is what we have here, radial symmetry. So again, the question will come from examples. So after Porifera, next is your Cnidaria and Coelenterata. Actually, there is a story behind. Do you know, initially the phylum used to be Coelenterata. And even Ctenophores, they used to be the member of Coelenterata only. Later on with further studies, we got to know that Ctenophora, right, they are the different organism, then different phylum was made, and then they divided the organism into two different phylums, right? They divided the organism into two different phylums, Cnidaria and Ctenophora, right? Earlier all the organisms, they used to be in the same phylum. They used to be in the same phylum. Are you getting my point? They used to be in the same phylum earlier. So this is what we have, Cnidaria and Ctenophora. What do we have here? Cnidaria and Ctenophora. So what you have to remember here, one thing that you all need to remember, and yes, that is the point from where the question can come, right? So that is in Phylum Echinodermata. When I'm talking about the Phylum Echinodermata, basically starfish. Your starfish belongs to this phylum, right? I'm writing the example here. In Phylum Echinodermata, example is the starfish, right? Adults, remember this point. Adults, they have radial symmetry. Which symmetry do adults have? Yes, people, please tell me. Adults, they have radial symmetry. And when you talk about the larvae, they have bilateral symmetry. Which symmetry do they have, students? They have bilateral symmetry. Bilateral symmetry. So because adults are having radial symmetry, that's why we say that this phylum is also having which symmetry? Radial, right? Because adults, they possess this symmetry. So overall we say that this phylum is having radial symmetry, but the larva will have bilateral. So is it an important question? Do let me know in the chat section. Is it an important question according to you? Is it an important question according to you? It's a very, very, very important question. It's a very, very, very important question. Right, bache? Right, bache? Are you sure? Are you sure? Are you sure? Sure. So is it an important question? Is it an important question? This question is going to be there. This question is going to be there in your NEET 2025 examination. Okay. This question is going to be there in your NEET 2025 examination. Okay. So make sure you remember this thing. Next thing that we have to remember is bilateral symmetry. Next thing that we have to remember is bilateral symmetry. So as the word is suggesting, bi means two, right? Lateral means you're talking about the sides. So here, right, you can consider your example as well. Our body can be divided into two equal halves when the plane, of course, it has to pass through center, but how? From top to bottom. But how? From top to bottom. But how? From top to bottom. So this is what bilateral symmetry is. So now, you know asymmetry, porifera, you know radial symmetry, then the remaining will be there in the bilateral symmetry, right? Remaining will be there in the bilateral symmetry. Few exceptions are there, but overall that is how we have to place it. Is that clear? Is that clear? So when the body can be divided into identical into two identical halves, that is into two identical halves, and that too how? When plane will pass from top to bottom. So basically only one plane can divide the organism like this, right? Only one plane can divide the organisms like this. So bilateral symmetry, when body can be divided into two identical halves. And which halves they are? Left and right halves, right? Left and right halves, right? By any one plane. By any one plane. We cannot say that any plane can divide it. No, no. So which example do we have here? Yes, people. Which example do we have here? Yes. Which example do we have here? So I know all of you know the answer. It will be Platyhelminthes to chordates. Everybody here in the class. It will be Platyhelminthes to chordates, right? Chordata. Understood? Platyhelminthes to chordata. Next, right? Next is about the germ layers. Now, I believe that all students that are watching my lecture right now, they know their class 12 syllabus. Do you know your class 12 syllabus? Yes. Do you have any idea of your class 12 syllabus? Tell me, do you have any idea of your class 12 syllabus? Hana? Hana? I think it's a yes. So when we study this part, we know that we all develop from the single cell zygote. We know that the cell will divide. The cell will divide. It will undergo cleavage, and cleavage, right? It will become multicellular. And there comes a time when it acquires a stage that we call as, that stage is known as gastrula. That stage is known as gastrula. So in the gastrula, we talk about what? We talk about the germ layers. Germ layers, to germinate, to form something. Right? Germ layers. So normally, initially, it is what? It is a multicellular body. After some time, what's going to happen? Some movements will be there. Different type of movements, right? We don't discuss about them in your class 11 syllabus, but yeah, different type of movements will be there. Just epiboly, ingression, delamination, many names are there, etc., etc., etc. Some movements occur. Because of that movements, what is going to happen? Some layers will form. And that layers, right, beta, that layers are going to form the body organs. When we talk about that layers are, right, that layers, they form the body organs. Are you getting my point? That layers, they form the body organs. Are you getting my point? Are you getting my point? Right? 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 That layers are what? That layers are going to form the different body organs. And that layers are the germ layers. Right? And when that germ layers will form, we will say it is the gastrula in which that germ layers will form. Right? It is the gastrula in which that germ layers will form. So now when you talk about these germ layers, my dear students, right, here we have three words. One is ectoderm. Of course, it is the outermost one. Then comes mesoderm, the middle one. Then comes endoderm, the innermost one. Are you getting my point? Ectoderm, mesoderm, and endoderm, these are the germ layers. Now, there are some organisms which are known as diploblastic on the basis of these germ layers. Why do we call them as diploblastic? Di means two. Why? Because they have two germ layers. Why? Because they have two germ layers, and that two germ layers are what? That is ectoderm. That is ectoderm and endoderm. That is ectoderm and endoderm, right? These organisms, they are going to have these two germ layers. And there are triploblastic organisms as well. There are triploblastic organisms as well. They have three germ layers. They have three germ layers. And here you talk about the ectoderm, outermost one, middle one is mesoderm, my dear people, and then comes the endoderm. Then comes what? Then comes the endoderm. So ectoderm, mesoderm, endoderm. Ectoderm, mesoderm, endoderm. That is what we have. Now, so diploblastic, two germ layers, triploblastic, three germ layers. Now students, when we talk about porifera, no doubt some teachers and even in some books it is written that poriferans, they are diploblastic, but actually they don't have germ layer, right? And in NCERT there is no clear-cut answer for this particular thing. But overall in poriferans, they don't have germ layer, right? So when it comes to the diploblastic organisms, which phylum do we have? Now you will realize the importance of remembering the phylums in one sequence. So in diploblastic, you have Cnidaria and you have Ctenophora. What do you have there? Cnidaria and Ctenophora. So of course, remaining will be triploblastic. Remaining will be triploblastic. So when I'm saying triploblastic, right? So what do you have? From phylum Platyhelminthes, right? From phylum Platyhelminthes to Chordata, they all are triploblastic. So let's suppose if in the paper there is a question that in animal kingdom, in which phylum for the very first time three germ layers were present, that phylum is yes. Yes. Answer this question quick. Quick. You tell me, in which phylum, for the very first time in animal kingdom, we'll see the 3 germ layers? We'll see the 3 germ layers, platyhelminths. In which phylum, for the very first time, we will see bilateral symmetry? In which phylum, for the very first time, we will see the bilateral symmetry? Again, platyhelminths. So these are the kind of questions that will come in your paper. Okay, these are the kind of questions that will come in your paper. Okay, so just keep it in your mind. So this is what you have to remember, right? This is what you have to remember. Is that clear? Is that clear? Right, now you know about the germ layers. Yes, you know about the germ layers. Now from this part, we need to understand something that is known as body cavity and coelom. That is known as body cavity and coelom, right? See, I'll make one thing clear here because I have seen students getting confused. I believe you all know about the poriferans, pore-bearing organism. When we draw their structure, right, it's a kind of flower-based type, na? Something like that, right? When we draw its structure, right, when we see the layers there, right, layers there, something like that. So we say that in between there is a body cavity, right? This is the cavity. We call it as spongiocele, if you people remember. We call it as spongiocele, if you people remember. So some students, they say that, ma'am, cavity is also there in the porifera, right? Cavity is also there in the porifera. Why is it so? Why is it so that cavity is there in the porifera, but we don't say that they are coelomates? It is not the cavity that I'm talking about. When I'm saying this body cavity, when I'm talking about this coelom, right? When I am talking about the coelom, please understand it, right? Please understand it. Please be attentive here. It is something important, right? It is the cavity which is present in between the body wall and the gut wall, and that is also lined with the mesoderm. I'm repeating this part again. When I'm talking about this coelom here, when I'm talking about this body cavity here, right, students? So here, what am I talking? I am talking about the body cavity. I'll show you as well. Present in between outer body wall and the gut wall. In between outer body wall and gut wall, right? And that too lined with mesoderm. And of course, in this cavity, what will be there? Viscera, body organs. Right? This is something that you have to understand when I'm talking about the coelom. Just say, for an example, let's consider this human body. Yeah, whatever body it is, right? Chalo, let's consider the Chintu's example. Okay, let's consider the Chintu's example. So we have our Chintu, right? Chintu is a human being. You know that in the case of Chintu, alimentary canal is there. I believe you all understand this thing, alimentary canal. I'm not talking about the digestive system. Achha, what is the difference in alimentary canal and digestive system, people? What is the difference in alimentary canal and digestive system? What is the difference in alimentary canal and digestive system? Anybody here in the class? Anybody? Alimentary canal, when you are talking about the tube-like, pipe-like structure from mouth to anus, right? And when we also discuss the digestive glands along with this alimentary canal, then we call it as digestive system. Are you getting my point? So alimentary canal plus digestive glands will make the digestive system. Okay. Now this is our Chintu. Our Chintu is having mouth, our Chintu is having anus. So now this is the outer covering of the Chintu, outer body wall of the Chintu. This is the outer covering of the Chintu, outer body wall of Chintu. And then this is the inner gut wall of Chintu, right? Inner gut wall of Chintu. I'll discuss two things here. I'll discuss the mesoglia as well. Don't worry. Don't be like, hey, madam, you have missed the mesoglia. No. Right? So this is the outer body wall, this is the inner gut wall. So now in between these two, right, you can notice it here also, outer body wall, inner gut wall. Outer body wall, inner gut wall. So there is a space in between. And this space, students, this space is also lined with what? It is also lined with the mesoderm. So this is actually your coelom. Right? The organism having this type of cavity is actually known as coelomates. They are the one which are known as coelomates. And here, what will you see? You will see body organs. You will see viscera here. So now let's say if I take the transverse section of that. If I do what? If I take the transverse section here. Okay? Transverse section here. So you people can see This is the, and this is the lumen, of course. This is the lumen, of course. So this is outer body wall. This is inner gut wall. And here there is a space. And as I said, it has to be lined with what? It has to be lined with the mesoderm, right? I'm just showing you it in this way, right? It is just lined with the mesoderm. So here you have the body organ. These type of organisms are coelomate. These type of organisms are coelomate, okay? Okay, so in the NCERT, if I talk about the diagram of NCERT, right, I believe you all remember the diagram also. It is showing something like that. It is showing something like that. I hope you all have seen that. It is showing something like that, a space like that. Yes, yes, the space like that. Yes or no? Yes or no? Right? Right? So here, when you people talk about this particular portion, right, that is inner gut wall, this particular portion, that is outer body wall. So of course, this outer body wall, it is made up of ectoderm, right, students? This inner body wall, it is basically the endoderm, and in between, right, this one which is lining the body cavity is mesoderm. That's what you people need to understand. So the coelom part clear, coelomate part clear. If it is a yes, I want to see some fire emojis in the chat section, guys. Be quick. Be quick. Yes, I want to see some fire emojis in the chat section, people, if this part is clear. Okay, so this is being a coelomate, right? Now you have to understand the coelom part and this germ layer part altogether. I'll show you the diagrams from the NCERT as well. This is, see, one thing, okay, this, array, this. Now, see, if this is your triploblastic organism having 3 germ layers, this one is what? This one is your inner endoderm, innermost endoderm. This one is your outermost ectoderm. This one is what? This one is outermost ectoderm, people. Now, let's suppose if it is a diploblastic organism, that point is same, that outer one will be ectoderm, inner one will be endoderm, because we don't have the mesoderm here. So in between these two, because these are the germ layers, cell layers, you can even make it like this. Let's suppose this part is showing the cells to us, right? Let's suppose this part is showing that it is a cellular layer, right? It is a cellular layer. It is a cellular layer. Now, in between these two, okay, in between these two, this part here, right, in between these two, this part here, can you see that, people? Can you see that? What is this, people? What is this? This is your mesoglia. What do we call it? What do we call it? Yes, what do we call it? We call it as mesoglia. What do we call it, people? We call it as mesoglia. Mesoglia. So it is a diploblastic organism having these 2 germ layers, and here in between you have mesoglia. This mesoglia is an undifferentiated layer. It is not cellular at all. It is not cellular at all. It is an undifferentiated layer. So if you ever get confused, just keep one thing in your mind, people. Just keep one thing in your mind. Take the example of jellyfish. In the jellyfish, that jelly part is this actually. Right? That is how you can keep the things in your mind. Right? That is how you people can revise it. Is that clear? Is that clear? Is that clear? Is that clear? Pakka? Sure? So this part is clear, mesoglia. But why are we talking about it? Now see, we are talking about the coelom. This coelom and this germ layer thing is related, right? This coelom and this germ layer thing is related. Just say here, I'm showing you that in the case of coelom, the cavity should be there, the space should be there in between body wall and gut wall, and that space should be lined with mesoderm. Now, from here, one question is that do all the triploblastic organism, do they have, yes, do all the triploblastic organisms, do they contain the coelom? Of course not. The answer is a big no here. Let me show you why. Let me show you why. ये देखो, something like this, right? Something like this. Like suppose this outer one is your ectoderm. This outer one is your ectoderm. This inner one is your endoderm. Right? This middle one, of course, is your mesoderm. Just look at it. Just look at it. So when you talk about the triploblastic organism, there are some triploblastic organisms in which, my dear students, the germ, the cavity is not there. So here, just remember one thing. This outermost, as I said, it is ectoderm. This innermost, as I said, endoderm. This middle one, as I said, mesoderm is there. But here in the mesoderm, there is no space. So yes, we do have examples like this. We do have examples like this. So when you talk about the coelom part, write down these examples, and again, then I will pick up the diagrams from the NCERT directly. So when it comes to the organisms which are acoelomates, that is without any body cavity, that is they are without body cavity, that is they are without body cavity, right? Without body cavity. So here the answer will be from Porifera to Platyhelminthes. Yes, you heard me right. In Platyhelminthes also the coelom is not there. Then there is another word which is known as pseudocoelomate, right? Pseudocoelomate. Pseudocoelomate. So pseudo means what? Pseudo means false. Pseudo means what? Pseudo means false. Coelom cavity. So where do you see this? You will see it in the phylum Aschelminthes. Where do you see this? You will see this in the phylum Aschelminthes. Actually, in Aschelminthes, students, what will be the point here? What's going to happen in Aschelminthes? Just imagine, this is your outer ectoderm. Okay, this is your then inner endoderm. Okay, so in between this ectoderm and endoderm, they have the cavity and space. This cavity is not completely lined with the mesoderm, but instead of that, mesodermal pouches are there. Instead of that, mesodermal pouches are there. Yes? Okay? Done? So this is the Aschelminthes. Mesodermal pouches are there. Mesodermal pouches are there. Is that clear? Now, after this, right? After the pseudocoelomates, you have the coelomates, as I discussed. Coelomates. So in the case of coelomates, of course, Aschelminthes, after Aschelminthes, we have Annelida. So from Annelida to Chordata. From Annelida to Chordata, right? From Annelida to Chordata, right? They are coelomates. But here there are two words that you need to remember. One is schizocoelomates. One is schizocoelomates, and the another word is enterocoelomates. And the another word is enterocoelomates. So schizocoelomates and enterocoelomates are there. So Annelida, your Arthropoda, and your mollusca. So the trick here is arm. The trick here is arm. A-A-M. Arm. Annelida, Arthropoda, mollusca. They will show schizocoelom, right? And here, E for enterocoelomate, E for Echinodermata. So your Echinodermata, your Hemichordata, and your Chordata, they are enterocoelomates. Is that clear? So remember this thing, my dear students. So Annelida to Arthropoda, Chordata, they all are coelomates, but that true coelom is also of two types, schizo and entero. So of course it is considered as the advanced one. And my dear students, when it comes to the schizocoelomate, do you need to remember that how does it form? Basically it's simple. S for schizo, S for split. S for schizo, S for split. Split. So here there will be a split in the mesoderm and the cavity will form. In the case of enterocoelomate, basically a pouch-like, right? From that gut part, a pouch-like structure will form that will further grow and then the cavity will form. That is not for you. That is not something that at this point of time you need to remember. But yeah, these examples you can keep in your mind. Okay, you can keep it in your mind. Okay. Okay, so is it clear? Sure, 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 sure. Pakka, pakka, 110%. ये देखो, चलो. Let's ठीक है, right? So they're looking like that spectacles, you know. So this is something that you have to remember regarding the coelom. Now, diagrams. See this. Diagrams are very important. We know it very well. So that's why I'm showing you this part here. So this is showing diploblastic organism, triploblastic organism. In the case of diploblastic organism, as I said, in between ectoderm and endoderm, mesoglia is there. And this mesoglia is undifferentiated, right? It is not a proper cell layer. It is something which is undifferentiated. Now here ectoderm, mesoderm, endoderm is there. Ectoderm, mesoderm, endoderm is there. Ectoderm, mesoderm, endoderm is there. Am I clear here? Am I clear here? Now, this diagram is for the triploblastic organism, right? But in the NCERT, when you see acoelomates, what type of acoelomates? Which are triploblastic as well, you'll see the same type of diagram. See, look at this. Look at this. So here, they are the coelomates. They are the coelomates. They are the pseudocoelomates. And this one is acoelomate. So the diagram for triploblastic organism and the pseudocoelomate is acoelomate is same. It is showing that here in mesoderm there is no pouch, there is no cavity, there is no space. Right? Right? So do not get confused here. Is that clear? Do not get confused here. Is that clear? Any doubt? Yes, people. Any doubt? Any doubt? Chali. So the next thing that we have to discuss is symmetry. But before that symmetry part, there are again few terms that we will discuss, and that is segmentation. Sorry. So let's discuss the segmentation, then come to the body plan rather. Segmentation, which is also known as metamerism. It is also known as metamerism. Is that clear? Segmentation and metamerism. Actually, what is happening that, like, see, when we move from Porifera to Chordata, we are moving from simplicity to complexity. Poriferans are the simple type of organisms. Chordatas, they are complex. So from Porifera to Chordata, like Porifera, very simple. Cnidaria will show some complexity in comparison to the Porifera. Then further the next phylums are like this. Okay, then further the next phylums are like this. Now, what am I trying to tell you here? What am I trying to tell you here? Segmentation or metamerism. Like in some organisms, their body is divided into some segments, externally, internally, right? Both. Okay, plus there is the repetition of certain organ. So what is happening here when the body is divided into segments or parts or metamers? Right, externally or internally or both, and there is repetition of certain body organs, right? Of certain body organs. That is what we see in the segmentation. And the best thing, like, where do you see this? Firstly, for the very first time, the true segmentation is there in Annelida, right? Phylum Annelida. In their case, my dear students, we see true segmentation in animal kingdom for the very first time. So you know that even in earthworm, right? The body is like this. We can see it very clearly, right? You can talk about the leech. You can talk about the nearest example here. So externally also segments are there, right? Externally also segments are there. These grooves, right? These groove-like structure here, we call them as annulus, and the plural is annuli. And here in between, right, internal segmentation is also there. We have septum. So septum is singular, septa is plural. And you know that there is the repetition of their excretory organ, that is nephridia. So for the very first time, where will you see the segmentation? For the very first time, where will you see the segmentation? You will see the segmentation in Annelida, right? Right. So basically this segmentation, as per NCERT, it is written like that, na? Annelida Arthropoda. Your chordates, they will show this. Okay, because in mollusca you'll see segmented mollusk, unsegmented, there the story is different. In mollusca, we also talk about the torsion. So we'll tell you slowly. But as per NCERT, these three things are given. So true segmentation for the very first time, where will you see? In Annelida. So we have one more thing that is known as pseudo-metamerism. Basically, it is the false segmentation, and that is in the case of tapeworm. Where do you see this? In the case of tapeworm. So in the case of tapeworm, right, in the case of tapeworm, you'll see the segments like this. You will see the segments like this, right? Actually, these segments are not embryonic. These segments are not embryonic. They are not developed during embryonic development. So this is the basically the neck region. So from this part, this new segments will form. Okay, right? The new segments will come from this part. So it is the pseudo-metamerism, right? It is the pseudo-metamerism. Understood? Understood? Right? So there is serial repetition of at least certain body organs, yeah, at least some organs. So this is the segmentation, this is the metamerism. So I believe up to this part, everything is crystal clear to all of you, right? Any doubts? Any doubts? Yes, any doubts? Any doubts here? Are you sure? Are you sure? So now there is one more thing, that is notochord. We'll discuss it once we'll start the phylum Chordata. So if notochord present, organisms are chordates. If notochord absent, they are non-chordates. So of course, from Porifera to Hemichordata, they all are non-chordates. Hemichordatas are considered as connecting link in between chordata and non-chordata. So what is this notochord? It is a mesodermally derived. What's that? It is the mesodermally derived, right? Means it is made up of mesoderm. Mesodermally derived, right? What is it? It is mesodermally derived. Yes. A rod-like structure, and you know that in our case it is the one that will form the vertebral column. So this part is clear, this part is clear. So other than that, what else you need to know? You need to know about the body plane as well, right? You need to know about what? Yes, you need to know about what? Yes, you need to know about the body plane as well, right? Body plane as well. Just a cell aggregate, multicellular body. Only cells are aggregated. You know that we discuss it in Porifera. Yes. Then blind sac body plan first. Blind sac body plan. Now students, what's the point here? See, from this part you need to understand one thing. There are two things. One is your incomplete digestive system. Incomplete digestive system. And the another thing or another important point is complete digestive system. One is incomplete digestive system, another is complete digestive system. One is incomplete digestive system and the another is complete digestive system. So in incomplete, there is only one opening. There is only one opening. Only one opening is there. Only one opening. Take that opening will act like a mouth, will act like an anus. Means through this opening only, ingestion and egestion occurs. Ingestion and egestion occurs. So only one opening is there. So it looks like a, this now means it is closed from one side. It is closed from one side. Right? Right? So it is the blind sac. One opening is there only. Now, when you are saying, so the organ, and now, how is it related to incomplete digestive system? Because incomplete digestive system means mouth is behaving like anus as well. Mouth and anus is same, right? Mouth and anus is same. So this type of digestive system will be the incomplete one, and the organisms having this type of digestive system, they'll have blind sac body plan because they will have just one opening. Now when there is the complete digestive system, so there will be two different openings here, right? There will be a mouth and an anus will be there. Okay, so it will be like this, it will be like this. Okay, it will be like this. Okay, so here two different openings are there. So this is the outer body wall. This is the gut body wall. So it looks like what? Tube. So organisms having this, they'll have tube within tube body plan. Tube within tube body plan. Is that clear? It is tube within tube body plan. Is that clear? Yes. Done? Noted? Sure? Chalo. So, this part is clear? Noted? Noted? Pakka? Okay. Chalo, I'll do one thing. Not tomorrow because I didn't schedule it yet. Tuesday at 5:30 p.m. I'll keep a special class on Unacademy platform. I'll keep a special class on Unacademy platform, right? And here, and this is free of cost, right? You have to pay nothing for this. And here, all PYQs of class 11th zoology will be discussed. What say? What say? Okay, happy? Happy? Tuesday, 5:30 p.m. Special class on Unacademy platform. That is too free of cost. All PYQs of class 11 zoology will be discussed. Actually, I forgot to pin the link today in the description box. So after ending the marathon, I'll pin the link. Pin this link here in the description box. Plus, I'll make a separate video to remind you this because otherwise, though, you're not going to join it. Okay? So I want to see you all there. So we'll practice the question. What say? We'll practice the question. What say? Happy? Happy, happy, happy? Are you sure? Happy? Are you sure? Happy? So that is why I'm keeping it at 5:30 now. 5:30 or 6:30 practice the question. Right, that too on the Unacademy platform. At 7 p.m. attend the marathon. Who's stopping you? Who's stopping you? So if you are happy, quickly hit the like button as well. Hit the like button as well. And today nobody is going to drop the session. Okay? Let's revise the complete class 11th. Right? Let's revise the complete class 11th. Class 11th what? Zoology. Right? And you know that you know that tomorrow you have to give the equal love and support to our dear HSP sir as well, because his session is also going to be amazing. So click on this thing, notify me, and hit the like button here, right? Hit the like button here. So tomorrow you have to give your love for this session as well. Okay, chaliye. So let's get started. So do not forget about it. And yeah, if students are not going to join it, I'll not keep the special classes. I'm very clear about it, right? If students will join there, if students will revise it, right? If students will participate here in the question practice session, I'll keep such kind of sessions. And as I said, free of cost. We will charge nothing, right? We will charge nothing, because here on YouTube now I have to answer this on the platform. I'll just start the poll. I'll ask the question. I'll give you the poll. In 15 seconds, you have to answer the question. We'll move to the next one. We'll move to the next one. We'll move to the next one. That's how we have to proceed. Okay, ma'am, can we wish happy birthday to Wasim sir? Arey, we will wish him. Don't worry. After some time, I'll call him. Then you can wish him. Don't worry about that. Now, this part is clear. This part is clear, everybody here. This part is clear, everybody here, right? Chali. So the next thing that we have to revise here, and then we'll jump to the porifera directly, right? It is the circulatory system. Circulatory system. Where do you see this? Circulatory system. So in the case of circulatory system, you have open circulatory system. You have closed circulatory system. Closed circulatory system, right? Closed circulatory system. Open. What is going to happen here? There will be a heart. Of course, not all the non-chordates are having this, right? The one which are having heart. Two large blood vessels, let's say, right? These are the two large blood vessels which are attached to this heart. Heart will pump the blood. The blood will pass to the body cavity, to the sinus, to the body cavity here. So it is the open type of circulatory system where you don't have the closed, right? Where you don't have the closed, where you don't have what? You don't have the closed. You don't have what? You don't have the closed blood vessels that are innervating each and every body organ. You don't have that, right? You don't have that. Okay, the heart is there, the large blood vessels are there, heart will pump the blood will be there in the body cavity then. Okay, so in the arthropoda, you'll see such type of circulatory system. You'll see such type of circulatory system. Even in mollusca, some exceptions are there, I'll come to that. Closed circulatory system mein, there will be a well-developed blood vascular system, heart, and there will be proper blood vessels that are going to innervate each and every body organ. Like in our case, coronary artery will give the blood to the oxygenated blood to the heart wall. Carotid artery will give it to the brain, right? So renal artery will give it to the kidney, right? So there will be the blood vessels that are going to innervate. Blood will basically flow in the closed blood vessels, right? So obviously it is more efficient. Obviously it is more efficient. Now, one more thing that you have to remember. In non-chordates, okay, your annelida will have closed circulatory system, and even it is said that only Annelida is the only phylum of non-chordata where closed circulatory system is there, but that is actually not true, right? In mollusca we have the open circulatory system in general, right? जैसे arthropoda में you will have without any exception open circulatory system. In mollusca also open circulatory system is there, but there is an exception, and exceptions are always important when it comes to biology. So when it comes to the exception, you have the exception of class Cephalopoda, right, in which there will be closed circulatory system, Cephalopoda. जैसे octopus is there. What is there? Octopus is there in Cephalopoda. I'm talking about what? I'm talking about the Cephalopoda. So what do we have in Cephalopoda? Octopus is there. Done? Done? Sure. So exceptions are always important. So, and even in your Echinodermata, Hemichordata, right? And especially in Echinodermata mein to you don't have the closed circulatory system. That's what you have to remember. Okay, that's what you have to remember. Is that clear? Circulatory system clear? Now, some people, they don't discuss it, they ignore this part, but I believe you should know it, right? We are not going to leave anything important or anything which was there in the previous year question. But we have two words. One is protostomous type of organisms. Another is deuterostomous. Right? Right? Protostomous, deuterostomous. Here, you have to remember one thing. And what is that? That is bad. bad is good. This is the trick that you have to remember. What is the trick that you have to remember? It is bad. Bad is good. So here B stands for blastopore. Now you know during embryonic development this blastopore forms. A for anus, D for deuterostomes. Right? So here it means, what is the meaning here? You just remember this trick, bad is good. So blastopore will form anus first. So in deuterostome, it is the anus that will form from the blastopore. That's what you have to remember. So if this is happening in deuterostomous organism, so in protostomes, of course, it is the blastopore that will form the mouth. So mouth will form first and blastopore is going to form that. Is that clear? Is that clear? So in Echinodermata, Hemichordata and in Chordata deuterostomous development is there. And here from Platyhelminthes to Mollusca, you'll see this type of embryonic development. Doubt? Because basis of classification is done and dusted. We'll move to the Porifera now. Understood? Sure? Are you sure? Are you sure? Yeah, I am feeling very low. Right, I am feeling very low. Our students are not consistent here. Students are just like, alright, we are not going to revise, we'll do it later. Right, matlab nahi, your energy is not up to mark, yaar. I'm not liking it. Your energy is not up to mark. I'm not liking it. Nah. Maza nahi aa raha. Nah. I'll share the PDF in the Telegram group. I'll share the PDF in the Telegram group. Some more energies. Little bit more. Little bit more. Thoda aur. Little bit more. So, okay. Keep it in your mind. Tuesday, 5:30, special class, free of cost. After today's lecture, I'll ask my team, right, to pin this special class link in that description box of this particular video so that you can directly reach there. Tuesday, 5:30. Tuesday, 5:30, we'll have this class. Okay, chali. So, look at this table because question will come from this table, and now tell me, look at it. If you are doing this chapter, Animal Kingdom, for the very first time, still you will be able to nail this table and the question will come from this part. If you guys are having any doubt, let me know. Right? If you guys are having any doubt, do let me know. And you people can be the part of my Telegram group as well. That is Love Biology by Ambika. Right? Simply you have to search Ambika Sharma Biology. That's it. Okay? This is what you have to search. Ambika Sharma Biology. That's it. So you quickly tell me this part. Yes? You quickly tell me this part. Is it clear to you? Is it clear to you? Right? Yes, you can be the part of that link as well. That is Ambika Sharma Biology. Okay, that is Ambika Sharma Biology. That's it. So, this part is clear. So, ye dekho, cellular level porifera, tissue level, right? The tissue level, radial symmetry, these two. Then, organ level, bilateral symmetry. But this is without a body cavity, platyhelminths. And that's a PYQ. In NEET 2020 or 2021, I don't remember the year, I think it's NEET 2021, right? This either in 2020 or 2021, this question was there, that out of the following, which organisms here, which phylum is having the organism, is having the individuals with organ level organization, bilateral symmetry and acoelomate. This is how they ask you the question. Right, this is a question in 2020 or 2021 where they have asked out of the following, which organism, which phylum is having the individuals with organ level of organization, bilateral symmetry and acoelomates. The answer will be platyhelminths. Answer will be platyhelminths. That's what you have to keep in your mind. Then false Phylum Aschelminthes. Then here, this is what you have. Do not get confused with the Echinodermata part. I have already told you that adults are having the radial symmetry, so it will be here. But here, even in the NCERT, in the bottom, they have mentioned it, ki this is the exception. So keep it in your mind, people. Keep it in your mind. Take diagrams, done and dusted. All this point is done and dusted. Come to the porifera, where the trick is, pour all your sight. Pour all your sights. Right here, honestly, I'm telling you this thing, right? I'm not lying. In the animal kingdom, I don't believe in the tricks, because we have so many examples, and if I will make the tricks, right, you need, okay, you need too much. You need too much to remember, too much time to remember the tricks. Then you have to remember the examples. So it's better to focus on the examples only, right? Right? It's better to focus on the examples only. So in the case of Porifera, trust me, I will not touch anything extra. I'm touching that particular points which are there in your previous year papers, right? Just in Porifera, if I have to discuss, I will discuss so many things. I'll talk about spicules. I'll tell you on the basis of spicule, there are different classes. I'll tell you about the class Calcarea, right? And everything. But now I'm not touching it because this is not the right time for that, right? And we will discuss all the things in detail, keeping NCERT in mind in our regular batches now, right? Because here, you know that our exam is there after 20 or 25 days, okay, right? So we cannot take that risk. Don't worry about that. Don't worry about that. Whatever I'm telling you, it is as per NCERT. And trust me, you're going to love it. So in the case of Porifera, one thing that I know, they are the pore-bearing organism. So do I need to recall these things again and again, that they are acoelomates, right? That they have cellular level of organization? Do I need to, right? Do I need to discuss it again and again? Please tell me. If it is a yes, I'll do it. If it is a no, then please, that no should be there in the chat section. Ki, nahi, madam, there is no need for that. There is no need for that. Right? Because we have just revised the basis of classification. You know that they have cellular level. They are acoelomates. They don't have any symmetry. Right? Right? They are having no germ layer. Of course. So why to pick that up? So in the case of Porifera, as per NCERT, if I have to mention, you have to remember these kind of things. Choanocytes, collar cells, right? Or you can call them as choanocytes, collar like cells they are. So that's how you can mark it. This is the cell. This is a cell that we have. This is the cell that we have, right? This is the cell that we have. So it is the flagellated cell, right? It is the flagellated cell. And here in the case of Porifera, because they have pore bearing body, like let's suppose if this is a poriferan, it will have these small pores. It is going to have these small pores. And what do we call it? Osteitis. What is it? It is ostia, right? Different type of shapes they have. This open structure through which the water will come out is osculum. So here when we take the section of it, right? When we take the section of it, and we have to be fast, people. Let's suppose if this is the section, and in different poriferans, na different level of like different complexity of this part is there. Just a minute. Right? Something like that. I'm taking the section here. Now, when I talk about this, these are the layers, of course, in between, right? These are the layers, of course, mesenchyme and all is there. So this inner part here, you'll see these collar-like cells. You will see these collar-like cells, right? These are the flagellated cells. We are going to see these collar-like flagellated cells here. And what are we calling them? As I said, choano. What do we call them? We call them as choanocytes. What do we call them? Choanocytes. These are the collar cells. And they are lining a cavity here. This cavity is spongiocele. Are you getting my point? And this layer, because it is made up of choanocytes, so overall this layer is known as choanoderm. So here we talk about these special type of cells. This is something you need to keep in your mind, right? Are you getting my point? Right? So here in this case, right, this cavity is spongiocele. So these flagellated cells, beta, they will maintain the water current. Through this ostia, water will enter here. Then the water will be there in spongiocele, and through osculum, it will come out. This is the root, right? Ostia. Ostium is singular. Then spongiocele it will be. Let me write it properly. Ostia. Then there is the spongiocele. Then there is osculum. So Ostia, Spongocele, Osculum is there. Ostia, Spongocele, Osculum is there. This is the root of the water. So here in these type of simple organism, in poriferans, we talk about water canal system. It is having different grades, different complexity. That is not something that we have to remember, but yeah, this is an important question. This is an important question. So when it comes to porifera, other than basis of classification, it should be clear in your mind, right? It should be clear in your mind that we have the choanocytes and we have, thank you, water canal system. We have the choanocytes and water canal system is there. We have the choanocyte and water canal system is there. Please keep it in your mind. Please keep it in your mind. Okay. Okay. So they are saying that they are generally marine, right? See, that is how whenever I'll pick up any phylum, I'll pick up with the most peculiar feature. Is that okay for everybody? Is that okay for everybody? I'll pick up with the most important or the peculiar feature. This is the feature that you need to remember from this particular phylum. This is the feature you need to remember from this particular phylum. Take, and then we will just read the NCERT, because when it comes to teaching you extra here, there are so many things, because of course the chapter is interesting, the topic is interesting, and I just love it, right? And I love teaching. And I genuinely love teaching. If you will ask me now, I can tell you so many things, new things about that. And I want you people to ask me that new things again and again, so that even the questions again and again, so that even I can learn new things. But the point is, we have an exam, we have to prepare for that. So I have to, restrict myself. Nahi Ambika, don't tell this point. This is extra, but this is not relevant for NEET. So I have to do it. So is that fine? Is that fine? Is that okay? Is that fine? But today I really need your support. Trust me, I was not keeping well, right? But yeah, now we don't have the options to cancel our classes. So I was like, chalo theek hai, let's take the class, then we will see. Okay, so I need your support. I want you people to be active. If you people will just drop down, I will also say bye-bye to you. I will just end the class. I'm very clear about it. Okay, so just focus here. So the word is generally marine, matlab not all are marine. So freshwater example is of freshwater sponge is your spongilla, right? Example is important. This example is important, right? This example is important. So generally marine, matlab not all are marine, because spongilla, it is there in fresh water. It's a fresh water sponge. It's a fresh water sponge, right? Right. Next point. No. They have water transport or water canal system. Highlight your NCERT with me in this way. Water canal system, right? And the water transport system. Do not confuse it with water vascular system. That is different. Take. Okay. Water will enter through ostia, then spongocele, then osculum. Through osculum, it will be out. Again, a PYQ. This will help in, so this water canal system will help in food gathering, will help in respiratory exchange and removal of waste. Then, collar cells, they are flagellated, not ciliated. They are the choanocytes. Take, and body is supported by and digestion is intracellular here. Okay, digestion is intracellular here, right? What kind of digestion is there? Intracellular. Intra means within the cell, right? Within the cell. It is important. Yes, madam, it is important. So spicules are there which are supporting the body, and on the basis of that, poriferans are further divided into various classes, right? Calcareous hai, spicule, yeah, siliceous hai, and spongian fibers are also there. So sexes are not separate. Means in porifera also, right? This is the trick that everybody has to remember, right? That is BMH. What's the trick that we need to remember? It's BMH. So B stands for bisexual, M stands for monoecious, H stands for hermaphrodite. So bisexual, monoecious, hermaphrodite organisms are there, right? So basically these are the keywords that you have to pick, right? Okay, so sexes, basically these cells will form against sperm, same individual will form against sperm, right? And then beta, they can reproduce asexually as well. In the case of sponges, we will talk about certain endogenous spores. How can I forget gemmules? Do you remember gemmules? Some structure like this. something like this. Anybody? Have you seen this diagram? It used to be there in the chapter reproduction in organism. Something like that, if you remember. Anybody, if you will answer this to me, and I'll give you a chocolate. I'll give you a chocolate if you will answer this question. I'll give you a chocolate. Yes, budding is there, but this is something that we see. Anybody who can give me the answer? Arey nahi yaar. Dekho, poriferans, they can divide asexually. So when it comes to the asexual division, indeed, budding is there, that we discuss. Fragmentation is also there, indeed, that we discuss. But this is not something that I'm talking about here. I'm talking about something else as well. This. Anybody? Gemmules. Okay, so you were not able to answer my question. Okay, right? Gemmules, they are some internal buds, right? They can survive in the harsh conditions. Okay, they can survive in the harsh conditions, right? So gemmules, this is something you have to remember, right? This is something that you have to remember. Okay, so the diagram used to be like this in reproduction and organism chapter. So I don't want to enter the depth, I don't want to discuss it in detail. So that's it. So asexually, fragmentation is there, gamete formation is there, fertilization is internal, and development is indirect, having larval stage, which is morphologically different from adult. Larva doesn't look like the adult. Take. So example, Scypha, Spongilla, and Euspongia. So Spongilla is the freshwater sponge, right? Spongilla is the freshwater sponge. Euspongia is the bath sponge. Cycon. So Cycon is having radial symmetry. Do not forget about it. Cycon is having what type of symmetry, people? It is having radial symmetry. Done? Let's move to the next phylum. So the speed is okay. The speed is okay. Diagrams, do not forget about them. Do not forget about them. So, bhai, see, it's not my cup of tea to remember such big tricks, right? So, if you can go for it. But I am not in the favor of that. All I know is ki if you will keep on revising them, you'll be able to master it. Acha, how many of you struggle in this particular part? How many of you struggle in this particular part ki madam, we are not able to remember the examples here. We have tried a lot, but now we don't have time to remember. How many of you? Right? If you think the same, then you have to send me some yellow heart. Yes, I am the one who's not able to do that. Send me some yellow heart that I am the one who's not able to do that. Hannah, this is the issue. I'll tell you. Now do one thing. Remember the phylum name in one sequence. Right, phylum name in one sequence. Remember this basis of classification wala part. Then solve all the PYQs of last 15 years. Right, if not 15, at least of 10 years. All the animal kingdom MCQs for the last 10 years. Dekho, I have one very simple logic. Right, I have one very simple logic. When I used to study, right, I still study, but when I used to write these examination, when I was in class 11, 12th or graduation, right, when you know that we open our books few days before the exam, and whenever I used to get that anxiety, that nahi Ambika, you are not able to make it, you will not be able to make it. Then I used to think one thing, dekho, okay, right, it is my fault that I started late. It is my fault that I started late. Now what am I supposed to do? I need to make sure that I need to make sure that I should score minimum passing marks first. And in my case, like let's suppose if my exam is for 100 years, minimum passing marks are not 33. I always used to consider them as 70. Okay, I need to get 70 marks now. So if I need 70 marks, okay, I cannot think of getting 100 out of 100 or 95, 92. I started late. So now I just need to be smart so that I can just get minimum marks, right? So that I can qualify the paper. Not 70, you can keep it 60. So in that case, you have to take the risk as well, right? You have to take the risk as well because you know you are the one who started late. So the best thing is right now, because we cannot remember all the examples, okay, at least solve 10 years, right? Just eat that questions. Whatever examples are there in paper, just take it. And it should not be like that if you're reading a question, you're like, okay, this question, the answer is B, okay, answer should be B. No, they can just shift they can just shuffle the options, they can just change the options. So you pick up that particular question, right, which were there in last 10 years wala paper, then remember that, understand them, because after this lecture you will be able to understand it. Trust me on that, you will be able to understand it. So just pick up that questions, remember them, but with proper understanding. Remember them. That's it. No need to touch any other example. Let's suppose from animal kingdom, if you will get 4 questions, by this method you'll be able to attempt 3 at least. So what is better? Not to attempt anything or to attempt 3 questions? What will be better? Of course, this method. So go for it. So don't worry. Today we'll discuss the smart strategies as well. Okay, we'll discuss the smart strategies as well. Don't worry about that. Don't worry about that. Okay. Coelenterata. Now here, important points. Nidoblast cells, also known as nematoblast cells, also known as stinging cells. That's something you have to remember. Second important point is alternation of generation here. And when it comes to this alternation of generation, the another word that we use here is metagenesis. Nidoblast, nematoblast, stinging cell, right? Alternation of generation, that is metagenesis. So here, sexual stages and asexual stages. Which stages, beta? Sexual stages and asexual stages. They alternate. So what is the trick that we have to keep in our mind? It is PAM. It should not be MAP. It is PAM. It should not be MAP. It is PAM. So P stands for polyp. A stands for asexually. M stands for medusa. So here, your P for polyp. Will asexually form medusa. So if polyp is asexually forming medusa, right? Actually, this room is short now. This room is small now, so I don't have much space. So P for polyp, A for asexual, M for medusa. So polyps will asexually form medusa, and of course vice versa will be there. Medusa will sexually form, right? Polyp, if asexually forming, it is medusa. Medusa will sexually form, right? The zygote that will grow like this. So this is something you have to keep in your mind. And on the basis of polyp and medusa, my dear students, we have the different classes here. Number 1, right? This is the alternation of generation. Number 1. Another important point from this particular phylum is corals that you have to remember, right? Right? I'll move to the another part as well. Don't worry. Alternation of generation, PAM, yes. Now, when you talk about this, right, they are mostly marine, matlab not all are marine. They are sessile. When the word is sessile, means they are attached to substratum. They are fixed. They are attached to substratum, that they are fixed. Now, P for polyp. This is my way to remember it. P for polyp, P for plants. We know that plants, they are fixed. That's how I remember. M for medulla, M for mobile. Mobile means movable. Are you getting my point? It is mobile. Mobile means movable. Are you getting my point? P for polyp, A for asexually, M for medusa. So this medusa is mobile. So this is something you have to keep in your mind. So now we have the polyp, just a hydra. Imagine hydra. Hydra is what? Hydra is a polyp, right? Hydra is polyp. Hydra will have this mouth here. Below the mouth, there will be that hypostome structure. The tentacles will be there. The tentacles are having these nidoblast, nematoblast, the stinging cell. It is something like that. Now when it will become this. It will go down. You will have medusa, right? This umbrella-like medusa will be there, just like your jellyfish, right? All the arms, the tentacles will be like this, and that's how the mouth is going to come. That's how the mouth is going to come, okay? And they are diploblastic. They have that mesoglia. So that jelly structure is that mesoglia only. Aurelia, your jellyfish is the example. Your jellyfish is the example. So that is how we have to remember, okay? That is how we have to remember. Are you getting my point? Are you getting my point? Are you getting my point? Am I clear here? Sure? Am I clear here? Okay. So name Cnidaria is derived from nidoblast or nidocytes. They are having the stinging capsules, nematocyst. Now, this is the another mistake that students make. This is the cell that we have here. This cell is having, this is the stinging cell. And this cell is having stinging capsule. This cell is having stinging capsule here. Okay, so basically this stinging cell is known as nematocyte, right? Nematoblast or nematocyte. This capsule inside is nematocyst or stinging capsule, right? And this particular cell is undischarged. It is having the neurotoxins in it, okay? And the diagram that we have in the NCERT here, this is the discharged cell. Matlab, this cell has been used by that particular organism for its defense, for the food capturing. That's what you have to remember. So how many of you are knowing this for the first time? So this is a cell. This is nidoblast. This is right? Nidoblast, nidocyst. So for this capsule, that is the terminology that we have to use, right? For this capsule, that is the terminology that we have to use. Is that clear? Is that clear? Are you sure? Are you sure? Pakka? 110%. So this is discharged one, used one, and we can use these cells just once only. Then that cells will be replaced by the new cells. Just keep it in your mind. So let's revise it quickly, right? We have to move very, very, very fast now. Now I have to increase the speed. Is that okay? I have to increase the speed. Is that okay? I'll finish Animal Kingdom, then beta I will give you the break. Let's not break the flow. And I'll be fast. Trust me. Trust me. 1 hour more, chapter will be over. 1 hour more, chapter will be over. Dekho, I started at 7:30. 7:30 to 7:15. Let's keep it 7:30, 8:30, 9:30. 10. Up to 10 we have to study, na? Okay, so stinging cells, they are present on the tentacles and the body. So nidoblasts are used for anchorage, defense, and to capture the prey. So cnidarians, tissue level of organization, done and dusted. Now, they have a central gastrovascular cavity. Like if this is the hydra, this is something that I have to show you. It's a polyp. This is the mouth. So mouth, below mouth will be what? Hypostome? Yes or no? And these are the tentacles. So if I take the section here, there is the cavity here. So I also call it as gastrodermis, right? And here within this cavity also the food will get digested. So here intracellular and intercellular, both type of digestion will be there. So you people can see this. So they exhibit tissue level of organization. They are diploblastic. They have central gastrovascular cavity with single opening, mouth on hypostome. So mouth is on hypostome, or you can remember this is the mouth, below mouth. Below means hypo. So below mouth, so you can use the word hypostome here. You can use the word hypostome here. That's it. So as I said, digestion is both intracellular or extra. So these are the kind of bullet points that you can write down right now. Right? These are the kind of bullet points that you can write now. You can write right now. Right? Right? Okay? Okay? Right? Pakka? Theek. So, right? So, next is the corals are also there where the skeleton is having calcium carbonate and they have polyp which is fixed and medusa form. So polyp is sessile, cylindrical, just like your hydra and adamsia. Adamsia is what? It is sea anemone. We all know about it. It is sea anemone. Then umbrella shaped free swimming is Aurelia, jellyfish, your medusa, right? So alternation of generation, also the word is metagenesis there. That's what you have to remember. So in obelia, which is also known as sea fur, right? In this obelia, you will see both. Okay, in the obelia beta, you will see both. You will see polyp as well as medusa. You will see polyp as well as medusa. Any doubt? Any doubt? Any doubt? So here also you will see incomplete digestive system. Here also you will see incomplete digestive system, and in between gastrovascular cavity is there, right beta? So for this gastrovascular cavity, coelenterine, right? This is C. What is the word that we can use? So coelenterine is the word that can be used. Coelenterine is the word that can be used. Is that clear? Is that clear? So Physalia, Portuguese man of war. Adamsia, sea anemone. Penatula, sea pen. From pen, sea pen. Gorgonia, sea fen. And Meandrina, brain coral. Do we need to remember it? Of course. So why do we call Physalia as Portuguese man of war, beta? It is believed that Portuguese, na, when they used to attack their enemy, they used to hide themselves first and then all of a sudden they attack. They never, attack directly. Ki we are here, right? Okay, we are waiting for the enemies, we'll kill them. No, they used to hide themselves and then when they get the chance, they used to attack. Same is for the Physalia. Physalia, right? It is not directly going to, give that face off to the enemy. Physalia is like, okay, my enemy is there, suddenly will attack, right? So that is how it works. Fine. So next point. Hanji, Ctenophora. In the case of Ctenophora, so examples, make sure you remember them. So you will see bioluminescence. So what is the meaning of bioluminescence? You know that, and it is because of which protein? Luciferin protein. Luciferin protein is responsible for bioluminescence. So what is this bioluminescence? The organisms, they'll emit light. They are the one that will emit light. And here you will see, in the case of this particular phylum, right, there are some cells, coloblast cells. Lasso cells are present in these organisms. Lasso cells. Not given in NCERT, but yeah, it is important, right? So they help in that adhesion. So basically, this is the table that you people can make because, of course, that is important. And I'm not talking about the subphylums here. So phylums that are exclusively marine. Exclusively marine means all the organisms are present in the marine water only. So first is your this Ctenophora, then you have your Echinodermata, right? And then you have your Hemichordata. Is that clear? And I am not talking about the subphylums here because in Phylum Chordata we discuss the subphylums. So in the paper, please read the question carefully. What are they asking? The phylums which are exclusively marines or subphylum, right? So these are three phylums. So is it important? Yes, madam. It is very, very, very important. Okay. It is very, very, very important. So Phylum Ctenophora, right? They are also known as sea walnuts or comb jellies, right? Exclusively marine. Very important point. About symmetry and everything we know. So their body will have 8 external rows. Just ये देखो 1, 2, 3, right? Total 8 are there. And that will have ciliated cilia over it. Usually cilia we have seen in the unicellular organism for locomotion, but they also use the cilia for locomotion. So after that unicellular protista or in the plant, unicellular protista, other than that in the Ctenophora also you'll see that they will use the cilia for the locomotion. So here in some Ctenophora tentacles are there, but that tentacles will not help in the movement. So on the basis of tentacles, their presence of absence, they have divided this phylum into different classes. But yeah, here you have to remember that they have 8 external rows of ciliated comb plates. So it looks like a comb. ठीक, so they help in locomotion. So here in animal kingdom, you will see in Ctenophora cilia is helping in the locomotion, digestion, again extracellular, intracellular. I told you about the bioluminescence as well. Now another important point, that reproduction take place only by sexual means, right? Just say in Hydra also budding is there, but here there will be only the sexual reproduction will be there. Okay, so pleurobrachia and Ctenoplana example. So write down these two examples in the chat section. Very easy they are, very easy they are. Just quickly write it. Right, so they don't have nematoblast, right? They don't have nematoblast, but coloblast or lasso cells are there, which help in catching the prey. Okay. Next phylum is your Platyhelminthes, flatworm, right? Next phylum is your Platyhelminthes, flatworm. And yes, sea walnut, comb jellies, and even some are also known as sea gooseberries. So do not get confused. Take, chaliye, move to the next part. Then Platyhelminthes, flatworms they are, bache. Right, body is dorsoventrally, flattened. They have F for flame cells, which is also known as solenocyte, which is also known as protonephridia. ठीक, and it helps in osmoregulation, maintaining salt and water balance, and of course excretion bhi. ठीक, keep it in your mind, keep it in your mind. Okay, right? So they are mostly endoparasite. Not all are endoparasite, but mostly they are endoparasite. ठीक, जैसे here bilateral symmetry, triploblastic, done and dusted. So now because they are endoparasite, they are living within the body of an organism, so they should have some parasitic adaptation. That is also PYQ. So when it comes to their parasitic adaptation, hooks and suckers are there, which will help them to get attached to the host, right, to the host cells, okay, in their parasitic form. So they will absorb nutrient from the host directly, but right. So they have no digestive system, because they will get the digested food only, right? The parasitic forms will not have the digestive system. So they have flame cells, help in osmoregulation and excretion. Very important point. Another names are also important. Here also sexes are not separate. Fertilization is internal, and development is through many larval stages. So this example is very important, beta. Planaria. This planaria is also known as Dagasia, right? And it is free living. It is not parasitic, it is free living. And here you will see that it is having the great power of regeneration as well. So when you look at its diagram, it's something like that. It feels like that it is having an eye, but no, it's not like that. So regeneration is there. Why am I telling you that this part is important? Because this example is also given in living word chapter. And this is what I have noticed, that in NCERT, wherever you will see that two things are repeated again and again, they'll definitely ask question from that part. Okay, right? So tapeworm, where we have already discussed about the pseudo metamerism. Then this is the fasciola. Take, this is the fasciola. This part is clear. I'm just sticking to the NCERT, not picking up anything extra. Take, right? So one more important point here is ki nervous system here is ladder like, just like ladder. Transverse commercial, longitudinal commercials like this. Understood? Understood? So one more exception I would like to mention here, but any doubt? Any doubt? Taenia solium clear? Taenia solium clear? This tapeworm, Taenia solium. So beta, it is also the pork tapeworm, right? It can cause the disease to the humans, right, bache? So just keep it in your mind. So it can cause teniasis to the human. And this is the liver fluke fasciola. Here the host is your sheep and the goat. They are the primary host. Secondary host is a snail here. Take, snail here. Again, different larvae are there. Dekho. Now one more exception. In animal kingdom, the next phylum to it is Aschelminthe. In animal kingdom, in the Aschelminthe, the sexual dimorphism is seen. What is the meaning of sexual dimorphism? That is, there will be two different sex, right? The male and female are in different body. So what am I talking about here? I am talking about the organism being dioecious. Take. So in Aschelminthe, right, which are also known as roundworms, because when we take their cross section, the body is circular, right? This is what we see. And you know that, if you remember, they are pseudocoelomate. But why am I telling you this point here? Because there is an exception. In flatworms, in platyhelminthes, my dear people, there is one example, that is schistosoma. It is known as blood fluke. What is it? It is known as blood fluke, right, bache? So it is in the human, it is found in human bladder and intestine. Where can we see this? We can see it in human bladder and intestine. Where can we see this? In the human bladder and intestine. So here male will carry the female, right? They are having a kind of groove. The male is going to carry the female. So in flatworms, this is where you will see that male and female are present. Okay? Okay, so here this schistosoma in flatworm is unisexual. No doubt, mainly we see this in for the first time we say that we see it in Aschelminthes, right? Majority of organisms will show the or the organisms here will show the sexual dimorphism, but schistosoma from the flatworm is also an example. So wherever I'm getting the exceptions, I'm just mentioning it. Okay, I'm just mentioning it. Okay, next. Right, so platyhelminthes done. And see, I was telling you about the ladder-like. So see, this is the eye spot. This is the cerebral ganglia. So ganglia, they are composed of, right? They are the aggregates of cell body of that neuron. And see the ladder-like nervous system. So this is your planaria, also known as digasia, having great power of regeneration. So it's a common question, right? It is common to 2 chapters, so the question can come. And this is the flame cell, which is meant for excretion and osmoregulation. Okay, chaliye ji. Next part is, okay, Aschelminthe. So the trick here is, if you can remember, you can go for it. Agent Ron will find all the hackers. Agent Ron will find all the hackers. So Ascaris, which is also monogenetic. What is the meaning of being monogenetic? Means it is having single host, right? I hope you have studied the human health and disease. Single host, and that is human. That is human. Okay, Baki Vicheria. If you remember Vicheria, Vencrofti is there, right, bache? Vicheria malai is there. So it is the one that causes filariasis. Is it important to remember? Yes, because it is also in the human health and diseases, also known as elephantiasis, filariasis or elephantiasis. It will block the lymph nodes, right? Lymph nodes. Okay, then Cyclostoma, which is also known as the hookworm. And yeah, about this Vicheria, remember, it is also known as filarial worm. It is also known as filarial worm, and it is considered as viviparous. Right, viviparous. So let's get started. Let's talk about the Aschelminthe. And yes, they are also known as nemathelminths, because there are certain nematodes which also cause disease to the plants as well. Okay, disease to the plants as well. So let's start it. And here, when you talk about the special cells here, you'll talk about H-shaped rennet cells, and these cells are for excretion here. Okay, these cells are for excretion here. Theek? Done? So they have circular in cross section. That's why roundworms, they may be free living, they may be aquatic, terrestrial, or parasitic in plants and animals. Right, plants and animals. Okay, do you remember in the biotechnology? We talk about that melodigen incognita. Remember? It was decreasing the yield of tobacco. Remember? So that's how you can mention. Okay, compare, and that's how you can just relate the things and you can revise. Right? Okay, Chaliye. So rest of the points are simple. Bilateral symmetry and all we have covered. So alimentary canal is complete here. So here you people are going to see tube within a tube body plan. Tube within tube body plan. So you know the meaning of tube within tube body plan as well. Okay, and here we also talk about the muscular pharynx, right? Here we also talk about the muscular pharynx. So excretory tube is there which will remove the body waste from the body cavity through the excretory pore. Okay, so male and female, they are different, and yes, often females are longer than males. Take. So fertilization is internal, it can be direct or indirect. Okay, Ascaris, Vicharia, and Cyclostoma, which is also known as hookworm, is the example. So hookworm, it is found in small intestine. It is found in small intestine. There are many other examples also, but yeah, the one that are given in NCERT, at least remember that. So Vicharia, which causes filariasis, right? The Vicharia, which causes filariasis, beta, it is considered as viviparous, the one which will give birth to the young ones. Actually, in its body itself, from that eggs, the filarial worm will form. So placental connection is not there. Placental connection is not there, but that is how it is going to give birth. So ovoviviparous, yeah, viviparous we call it. Okay. Okay, and yes, in the so WC, take Culex mosquito, female Culex mosquito is the host, sorry, is the vector there that will spread this disease. So it will cause elephantiasis or filariasis. Important it is. Take, important it is. Next part, Annelida, the phylum where for the first time we will see metamerism, the phylum where you will see closed circulatory system, right? So the trick here is no person should exit exam hall before time. But I don't need this trick because I know here I just have very simple examples. I just have very simple examples, that is earthworm, Nereis, which is also known as clamworm. Okay, so earthworm, but zoological name is your Ferretima. There are other genus and species also. Then you have your Nereis, right? That is also having heteronereis form. And then you talk about the leech. That is your Hirudinaria. That is your Hirudinaria leech, having anti-blood coagulant hirudin. Take. So this is what you have to keep in your mind directly. And this Nereis is unisexual. Okay, this one is unisexual, that is dioecious. Again here, you'll see that earthworm and leech, these two, they are bisexual. They are bisexual. Am I clear here? Again, it's a PYQ. That's why, beta, I'm directly highlighting this portion. Is that clear? Is that clear? Are you sure? Are you sure? So they can be aquatic, okay, terrestrial, free living, and parasitic also. Okay, so I'm not discussing bilateral symmetry and all. I have already discussed it. So they have the segments which are also known as metamers. That's why the phylum Annelida. It is from the Latin word annulus, means little ring. Okay, so they have longitudinal and circular muscles that will help in locomotion. Jesse in our case, it is ICOL na, inner are circular, outer are longitudinal. But here it is opposite. Here inner are longitudinal muscles and outer will be then circular. In our case, the story is like this: inner circular, outer longitudinal, but here inner muscles will be longitudinal, outer will be circular, beta. And then in the case of Nereis, right, in the case of Nereis, what will you see? It is known as the sandworm and the ringworm. Clamworm is also the name. Sandworm or ringworm. So of course the body is segmented. So let's suppose if this is a segment, substructures like that. So parapodia is there for the locomotion. Is it important? Very important. Okay, so closed circulatory system is there, and here you have nephridia. Nephridia, just in earthworm, different type of nephridia is there: septal, pharyngeal, integumentary, right, like this, like this. Okay, so help in excretion and osmoregulation. Neural system is having paired ganglia, right, connected by lateral nerves. So they have double ventral nerve cord. जैसे देखो, in chordates, wherever in non-chordates, you'll see heart. जैसे our heart is what? Our heart is on ventral side. In their case, if heart will be there, that will be on dorsal side, right? And whenever you will talk about nerve cord, that will be double, right? That will be double or single? Double, solid, ventral. Just in our case, the nerve cord is on dorsal side. Here it will be ventral nerve cord, right? It will be ventral nerve cord. Is it important? Yes, ma'am. It is very important. It is very important. It will be double, it will be solid, but it will be on the ventral side. Take. So Nereis is aquatic form, but this example here is dioecious. Earthworm and leeches, they are monoecious. You know that in the case of leech also, suckers are there. So this leech, it feeds on human blood. So we use the word sanguivorous for it. Sanguivorous, that feeds on blood, right? It's a parasite. It feeds on blood. It feeds on what, beta? It feeds on blood. It feeds on blood. So the example clear? Example clear? Sure. Chaliye, so let's move towards the largest phylum, right? That is our Arthropoda, right? Largest phylum of animal kingdom. Let's talk about what? Let's talk about the largest phylum of animal kingdom, right? That is your Arthropoda, number one. And yes, just here we talk about the parapodia, na? Parapodia for locomotion. We also talk about the chitinous setae there, right? We also talk about the chitinous setae there. What is this? Chitinous setae, right, beta? Right, bache? So that is, right, that is also helping even the parapodia. Parapodium is singular, podia is plural. They also help in locomotion. It also help in locomotion. Is that clear? Is that clear? And bache, in this case, right, matlab here in the phylum Cnidaria, that will also start in, in the phylum, what do we say? In your phylum Annelida, you will see that syphilization. What is the meaning of syphilization? What is the meaning of syphilization? Head formation. So you will see that head formation will be there. Even in the Phylum Aschelminthes, you will see at some level it is there, but here you will see that syphilization is present. Where the syphilization is present? In the case of Nereis. In the case of Nereis. And again, one more PYQ. I thought I should tell you right now only, right? Though we will discuss that in our special class as well, but I believe I should discuss that with you right now. So it is the phylum Cnidaria, right? Where for the first time you will see that a diffused network of nerve cell is there. That is also PYQ. Diffused network of nerve cells. It is the diffused network of nerve cells. For the very first time, you will see the nervous system is Cnidaria. That is also PYQ, right? Right, in animal kingdom for the first time, here we will see. So it is a primitive type of nervous system. It is a primitive type of nervous system, but yeah, it is present, right? It is present. It's a primitive type of nervous system, but yes, people, it is present, okay? Done. So largest phylum of animal kingdom. And Annelida will have what type of circulatory system? Closed, right? It is the first phylum of this animal kingdom for the very first time. The closed circulatory system you will see here. And in non-chordates, this phylum will have closed circulatory system, and the exception is Mollusca. The class Cephalopoda of Mollusca is the exception there. Take. So largest phylum, without any exception, open circulatory system. Here, right, when we are saying largest phylum, so beta, out of 10 animals on this planet Earth, you know 7 are insect. 7 are insect. Again, it's the line of NCERT only. It's the line of NCERT only. Out of every 10 animals, 7 are insect. So it is the largest phylum of animal kingdom. It is having the open circulatory system and the chitin is there that is making the exoskeleton, and due to this exoskeleton, right, the level of protection is more, right, the level of protection is more there. So this chitin, remember, it is a homopolysaccharide, right, it is a homopolysaccharide. Homo means same, poly means many sugars. It's a homopolysaccharide. It is made up of NAG, N-acetylglucosamine. It is the NAG, N-acetylglucosamine, right? It is the NAG, N-acetylglucosamine. So these are the direct point. And here it is the phylum where for the very first time you'll see the respiratory system is there. Take the respiratory system is there. Just here you'll talk about the tracheal system. So we discussed the cockroach is a part of our syllabus now. There you will discuss it, right? So the tracheal system, right? Tracheal system is there, and this is not the only thing. There are other type of the respiratory systems are also there, right? So tracheal system, of course, in insects, right? Of course, in insects. Then, other than that, we also talk about book gills. Book gills or book lungs? What's that? Is it book gills or book lungs? I don't remember. That's why I'm asking you. Is it book gills or book lungs? Is it book gills or book lungs? Beta, both. Book gills are also there. Book lungs are also there. You also discuss the simple type of gills as well. Book gills, book lungs, both are there, right? So they are different types of respiratory organ, right? Tracheal system will be there in insects. Just cockroach is the part of our syllabus. We discuss is there. So when it is the book gill, where are you going to see this? Just book lungs are the part of your scorpion, right? Means phylum, sorry, means class Arachnida. Book gills will be there in your limulus, also known as king crab. Beta, this limulus or king crab is the living fossil. So when we say that something is living fossil, like in plants you have the cycas, ginkgo biloba, they are living fossil, means they have the characters just like, they have very primitive character, just like the fossilized organism. Or you can say that they just don't evolve, they have the characters of the organisms which are now fossilized, right? Which are now fossilized. Okay, which are now fossilized. So this is important. Simple gill you can see in the case of prawn as well, right? Prawn as well. But remember, no doubt, different type of respiratory systems are there, but again here the direct type of respiration is there. Means what is going to happen here? Blood, even in the insects also, blood is not the one that will transport the oxygen. Directly through diffusion, the body cells or the tissues will get the oxygen. That is the point that you need to remember. So let me mark it here. So they are simply saying that largest phylum of Animalia, which include Insecta, is your Arthropoda. It means jointed appendages. It means jointed appendages. What is the meaning here? Jointed appendages. Take over 2/3 of all the named species on earth are arthropods, right beta? Rest everything we have discussed. So body is, of course, here also chitinous exoskeleton is something very important that has provided them more degree of protection. So where else do we study the chitin? In the fungal cell wall. In the fungal cell wall also we talk about the chitin. So head, thorax and abdomen is there. In some cases beta, head and thorax, it is fused. Head and thorax, it is fused. So we also use the word cephalothorax there, because head and thorax is fused. So head formation is there. So definitely cephalization is there. Cephalization is there. Do not forget about it. So jointed appendages, respiratory organs are gill, book gills, book lungs and tracheal system. Circulatory system of open type without any exception. Antenna, eyes, which are compound and simple. Stetocyst is for the balancing there, right? So when I'm talking about the eye here, right, simple eye. In the case of cockroach also we discuss it. We call it as oscillus, singular. Ocelli is plural. And we talk about the compound eye here. Actually, this eye will just, it is basically sensible. It will tell us whether there is dark or light. Okay? Right? This simple eye. Oscillus. Okay? Right, it will not be it doesn't help the organism to see the object. It will just simply tell whether light is there or not. When it comes to the compound eye, it provides the mosaic vision to that particular organism. Mosaic vision to that particular organism. And here the single subunit is ommatidium, right? Basically, so many ommatidia. You can say around 200 or 2000, right? These are hexagonal structures, na? These are hexagonal structures, na? Yes or no? Right? Just a compound eye will provide the mosaic vision. Compound eye will provide the mosaic vision here. That is something you have to remember. So, of course, it is a part of the sense organs. It is the part of the sense organ. So, it is the ommatidium. It is the ommatidium is singular. Ommatidia is plural. Right? Ommatidium is singular. Ommatidia is plural. That will make this compound eye. That will make this compound eye. Okay? Around 2000. Hai na? Hai na, Priya? Okay, so this is something that you have to remember. Mosaic vision it is going to provide here, right? So stetocyst is also there. And now when it comes to the excretory system, but I am picking up the PYQs here. Okay, I am picking up the PYQs here. Please pay attention. Kindly pay attention. Right, beta? So when you talk about the excretory system, you have Malpighian tubules. And that too in insects. And that is again a PYQ of the cockroach. That is again a PYQ of the cockroach. That Malpighian tubules, they are present at the junction of, they are present at the junction of midgut and hindgut. Midgut and hindgut. They are present at the junction of midgut and hindgut, excretory system. So it is not just the only excretory system that we have. Antennary gland or the, right, antennary gland or the green glands are also there. Green it is. So it will be there in prawn, that is crustaceans. You have coxal glands also. In spider, belongs to class Arachnida. All are PYQs. Write down. All are PYQs. Write down. So, in Arthropoda, high degree of syphilization is there. Mostly dioecious beta. Fertilization internal and they are oviparous. So, economically important, honeybee, bombyx, that is silk worm, lessifer, lac insect. Anopheles is the vector, they spread the disease. Culex, just I gave you the example of Wicheria. Then Aedes is also there. So gregarious pest. So gregarious in Hindi, gregarious in the meaning of Hindi, the meaning of gregarious in Hindi is milansar, right? In Hindi we call it as milansar. So when you are talking about the gregarious pest bache, they will not live alone. They are basically in the groups, right? Just locust swarms, right? They form. They are basically in the groups. Then living fossil is your Limulus or king crab that we have already discussed. Now let's move to the second largest phylum of animal kingdom, kingdom Mollusca. It is the second largest phylum of animal kingdom. It is kingdom Mollusca here. So people of Paris save light current on all days. So again, as I made it clear initially that I do not support the tricks here, but if you want it, I have given you. One more thing is there, beta, silverfish. Silverfish. It is an arthropod. Arthropoda. Right? It is lepisma. It's this zoological name is lepisma. Just in when you don't wear clothes for long period and they are in your almirahs, you'll see a kind of shiny, very small insect, especially in the books when you go to the library. If there is a section where people don't visit that frequently, you're going to see, okay, you are going to see the kind of insect, right? You are going to see the kind of insects which are very small, very shiny. That is lepisma. That is a silverfish. So remember, they are not the true fish, right? True fishes belongs to your Pisces only. They are not the true fish. This kind of question rarely, but yeah, it can come. Just say here we have the example Phyla, apple snail, pink tada, pearl oyster, again PYQ. This is the one that will give you the pearl, right? Pearl. This is the one that will give you the pearl. Why? Because there is a layer nacre. So that pearl formation is a kind of defense mechanism here, just to protect its body from the impurities, from any types of, if some thing enter in their body that is just like the irritate. In response to that, the necker will form the pearl. So this is what you have to remember. Baki sepia, loligo is there. Sepia is also cuttlefish. Again, it's a false fish. Loligo, squid. Then Chaetopleura, which is chitin. So when you look at this chitin, when you look at this, it is having 8 plates. So sometimes, right, they can ask the questions like this. Baki octopus, which is the devilfish, right, which is the devilfish, and you know that, right, they belong to the class Phylopoda. They have closed circulatory system. I'll come to that. Aplysia, see here, dentalium, tusk shell. That is also a PYQ about the dentalium, tusk shell. Take. So in mollusca, what do we discuss? Muscular foot. First of all, they have shells, right? This shell can be internal, it can be external, it can be absent. So when you talk about this shell absent, you have to remember octopus. Number 1, you talk about the muscular foot here, you talk about the radula here, which is the rasping organ in the buccal cavity. It is there for the grating. You talk about the ctenidia here, ctenidia. These are very thin, filament-like gills, right? These are feather-like gills or filament-like gills they are. So they are not just playing, helping in respiration, but along with respiration, they are also helping in excretion. How do they help? They are also helping in excretion. How do they help? They are also helping in excretion. So how do they help in respiration as well as in excretion? So I have picked up the important point. Muscular foot, shell, radula, ctenidia, right, ctenidia, and one point, here you see that there will be organ of Bogenus, which is also known as Cabers organ. That is basically for excretion here. Okay, that is also for the excretion here. And here in some classes you will see torsion, right? That's a kind of, right, that inversion here. We'll discuss about it. So these are the key points that I have mentioned. And yes, how can I forget mantle and the visceral hump? Allow me 1 minute. We'll discuss that too. Mantle and the visceral hump. Mantle and the visceral hump also we have to discuss. Take. So pick up the most important point. Any doubt? Any doubt? Yes? Any doubt? Sure. So second largest phylum, terrestrial or aquatic, they are organ system level of organization, bilaterally symmetrical, triploblastic and coelomate. Simple it is. They have the calcareous shell. But now what you have to remember, please pay attention. That is again a PYQ. They are unsegmented. They don't have segmentation. This is important. This is important. What I just said, that they are unsegmented. Unsegmented. Mollusca are unsegmented. Here again, we have exception. That exception is also a connecting link. That is Neopilina. Neopilina. That is an exception, my dear students. Right? What is an exception? You have the exception Neopilina, which is the connecting link between Annelida and Mollusca. So it is a segmented mollusk. There are many connecting links. The examples are not given in NCERT. Just say peripatus is there. Many examples are there. I'll just touch the NCERT part. Ambika, just stick to the NCERT. So this is important. Yes, madam, this is important. Okay, so they are basically they are unsegmented, but this is the example of segmented mollusca. Right? Do make the notes. So they have the distinct head, indeed. They have the muscular foot for locomotion. They have the visceral hump as well. Visceral hump as well. So beta, when I am saying visceral hump, so this visceral hump, when I am saying visceral hump, this visceral hump contains all body organs. It contains all body organs. What do they have? They contain all body organs. Is that clear? And soft and spongy layer that they have. Ye dekho. Soft and spongy layer that they have, right? Layer of skin, it forms a mantle over the visceral hump. Just say for an example, Oi, oi. Right, so do not forget these points. They are important, that's why I'm mentioning it. So now we are saying, right, that there is a layer that is known as mantle. So the space between hump and mantle, right, first of all you have to remember that, just say, let's suppose this is the visceral hump. Visceral hump matlab it is having all the body organs. It is having all the body organs. This is the visceral hump. This is the visceral hump. So there is a layer of skin that is covering this visceral hump. We are calling it as mantle. We are calling it as mantle. And we know that they are having the shell as well. They are having the shell as well. So now the space between, where? Between this hump and mantle, not between the shell and mantle, because that is also a PYQ. That's why I'm mentioning it. Please pay attention. It is a PYQ. It is a PYQ. That is why I'm highlighting it. So here, in between this mantle and the visceral hump, there, the space present in between is mantle cavity. The space present in between is mantle cavity. So it is not the space in between shell and mantle. It is a space in between visceral hump and the mantle. That is the mantle cavity. Is that clear? That is the mantle cavity. Is that clear? Is that clear? Okay, so this mantle, it usually secretes this calcareous shell. Take. Okay, look at this. Look at this. So this is a snail, right? You can see the different organs there. Osteichthyes is there, which will check, you know, which is basically for the it is also the sense organ in simple words. Mantle cavity, visceral hump is there, right? And I told you radula you have to remember, which is the rasping organ. So you can see this part here. You can see the diagram here. Any doubt? Any doubt? So, feather-like, filament-like I mentioned, initially feather-like gills are there. So, the name for it is Ctenidia. Not mentioned here, but that is important. That is important. Feather-like gills are there. What do we call them? We call them as Ctenidia. Write down. We call it as Ctenidia. Right? Done? Done? Sure? Right? Baki, anterior head region has sensory tentacle, file-like rasping organ, radula is there. So it is just like a grater. It is just like a grater. So usually dioecious, not all are dioecious usually, and they are egg-laying with indirect development. So when we say the development is indirect, matlab larval stages are there. So Phyla, apple, snail, pink tada, I told you, sepia, cuttlefish, loligo applies here. So examples are important, and please mention this point, everybody here in the class, please mention this point, please mention this point, that when you talk about the cephalopod, so cephalopod mein you have the example of octopus, you have the example of sepia, you have the example of loligo. They belong to the class Cephalopoda. Cephalopoda. So they have, right, they have closed circulatory system. So yes, this is important to remember. What do they have? They have closed circulatory system. What do they have? They have closed circulatory system. Is that clear? And in your loligo, which is also known as squid, sometimes this kind of questions can also come. The radula is absent in squid. It is important. Radula is absent there. Okay? And here the shell is internal. Shell is absent. So on the basis of number of their tentacles, we differentiate them. So sepia is your cuttlefish. Take. Okay. Right. Take. Done. So examples are given that are important. So dentalium, tusk shell, and here in this tusk shell, right, the respiration is through mantle. And in Phyla, lungs like, matlab a pulmonary sac like structure is also there. Next is Echinodermata, right? starfish. Okay, next is Echinodermata. So bache, you can compare them with the chordates as well. Why? Because here you will see that mesodermally derived endoskeleton will be there. Mesodermally derived endoskeleton will be there, right? This is the point that you have to remember. You know that they are enterocoelomates and you know that they are deuterostomes. Okay, okay, and here we talk about the water vascular system. So do not confuse it with the water canal system of Porifera. So this is the water vascular system, which is also known as ambulacral system, which is also known as ambulacral system. Ambulacral system. Take. Okay, so Echinodermata, spiny bodied, all are marine. So told you already, it is also the phylum which are organisms which have organisms which are exclusively marine. Any doubt? Any doubt? Yes. Any doubt? Sure. So adults are having radial symmetry, larvae is having bilateral, already discussed. Digestive system is complete here, but here beta, mouth is on the ventral side and anus is on the upper side, that is on the dorsal side. It is important. Take the most distinctive feature, as I said, is water vascular system, and this system actually helps in locomotion, capturing and transferring of food and the respiration. So here you can say they don't have no excretory system, right? Trust me, no brain, no heart here. In the Echinodermata, no brain, no heart. So sometimes, right, I mentioned this to my brother that you are starfish. You don't have brain, you don't have heart, you don't have even the excretory system. Take. So sexes are separate, reproduction is sexual, and external fertilization is there. So indirect development means larval stages will be there. So example mein Asterias, starfish, Echina, sea urchin, Antedon, sea lily, Cucumeria and Ophiura is there. So if you can remember it, very good. If you cannot, the best way is to revise the PYQs. Okay, it is to revise the PYQs. Okay, so here open type of circulatory system is there, right? But when it comes to the circulatory system, it is beta reduced and open type, and no heart is there. Take, it is reduced and open type, and as I said, no heart is there. No heart is there. Take. Right, and here you talk about the tube, feet and all, we'll discuss it. So respiration ke liye beta, body surface is there, gills are there, dermal branchia are there, and here you can see the ambulacral system. Ye dekho, madreporide, sieve like structure from where the water will enter, ring canal is there, these are the radial canals here. So these are the tube feet in the surrounding as well. Okay, right beta, tube feet are also there, right? Okay, yes or no? Is that okay? Baki to you know that there are so many important things that we can discuss here, but chalo anyhow. Baki beta dekho, starfish mein male and female is different, right? But you will not externally, by looking at them, you cannot say that which one is male, which one is female. So sexes are separate, but that dimorphism is not there, right? That we can say that this one is male, this one is female, this is like this. And there are many other extra points, but you know that we are running out of time. We have the exam soon, so we have to stick to the most important topics only. Now beta, hemichordata. Initially they were placed in the chordata because of the presence of what? Because of the presence of stomachode. But that is somewhere, you know, related to the digestive system part, right? It is associated with that only, right? So here they are earlier considered as a subphylum under phylum chordata, but now it is under a different phylum under non-chordata. So they have rudimentary. Rudimentary means, rudiment in Hindi means avshesh, remaining part, right? Remaining part there. So in the collar region that is present, that is stomochord. So they confuse it with the notochord. They confuse it with the notochord, right? They confuse it with what? They confused it with the notochord. Is that clear? Is that clear? Sure. So stomochord it is again tube like beta, right? And it is somewhere there in the proboscis. Okay. Okay. So it was considered the same as notochord, but it is not true, right? So it is during the embryonic stages. Baki they have worm like they are. Okay. They are marine animals. So you know that three phylums that are where the organisms are exclusively marine. Again, I'm revising it, right? Where organisms are exclusively marine, right? So number 1, Ctenophora, Echinodermata, Hemichordata. Take, do not forget about it. Okay, so here beta, the body is cylindrical, it is having anterior proboscis. So if I have to confuse in paper, I can put posterior here. But remember, proboscis, which is also helpful in the excretion, it is present anteriorly, then a collar and a long trunk is there. As I said, circulatory system is of open type. So no doubt, Phylum Annelida is the only phylum of the non-chordates having closed circulatory system, but along with that, class Cephalopoda is also there of mollusca. Right? Right? Class Cephalopoda of mollusca is there. Okay? Okay? So gills are there for the respiration, proboscis gland is there for the excretion, sexes are separate, fertilization is external, development is indirect, balanoglosses and saccoglosses, worm-like body they have, right? Worm-like body they have, right, bache, right? Okay, so let's start the phylum Chordata. Let's start the phylum Chordata. Okay? So in the case of phylum Chordata, dekho, I know, you want break. Okay? But if I'll give you break now, right? You tell me. See, even I am hungry, right? Even I am hungry. And as I said, I was not keeping well. Okay, so sometimes you'll see me sitting like this. So I am saying it's simple. If I'll give you break now, let's suppose I'll join after 30 minutes. Then now we have that momentum now. So if, right, in continuity, if we'll finish this part now, it will hardly take 40 minutes to finish this chapter. Right? And it is one of the length, right? It is also a lengthy chapter. So basically, you know that 4 questions are expected, 16 marks. Yes? Hannah? So if I'll give you a break right now, after the break, when I'll join, that momentum, it will not be there. We will not gain the same momentum. It will take some time to gain that. So right now, if in one go, if I'll proceed now, we can finish it in next 40 minutes. Because I have to just pick up the main things now that will come in paper. Isn't it? So let's continue. So if you all are aligned, write this thing in the chat section. Let's continue. After 40 minutes, let's have a break. At 10:30. I'll give you the break for 30 minutes and then let's continue for the next 2 chapters, tissue and breathing and exchange of gases. Or whatever chapter you will say, I'll pick it up. Okay, it will be as per your choice. Whatever my students are going to say, I'll pick that chapter up. Continue. Pakka? That's like a good boy or good girl. Chalo, let's continue. Now, the next thing that we have to discuss is phylum Chordata, where we have to read the things up to class level. Here, on what basis we say chorda? Chorda is the word. Chorda is the word here that you need to remember. And yes. You know na, hemichordates, they are the connecting link in between the balanoglosses. It's a connecting link between non-chordates and chordates. Connecting link between non-chordates and chordates. And it is also known as your tongue worm. Take tongue worm. Okay, which worm? Tongue worm or acron worm. Now phylum Chordata. Take, what is the word? The word is phylum Chordata. So chorda mane thick string. Chorda. What is the meaning of chorda? Thick string. Ata, to have. So they have a thick string here, right bache. So here we are talking about what? We are talking about having notochord, right? And on the basis of that, we are putting the organism like this, na? Right? Having notochord. What do they have? Notochord. So majority of animals are non-chordates, only few animals are chordates, right? Basically 3 to 5% animals are chordates. So when it comes to the chordates, beta, most important characters are this, like 4 basic characters of chordates. Presence of notochord, of course. Presence of dorsal hollow nerve cord. Presence of pharyngeal gill slits, presence of post-anal tail. Four main characters. These are that four main characters. Some students, when they ask four main characters, they consider heart is ventral. No. These are the four main characters that we have to study. Now, look at this diagram. Look at this diagram. Here in this diagram, notochord is there somewhere in the middle. This part is the notochord. So it is a mesodermally derived. What's that? It's a mesodermally derived structure, rod-like, flexible structure it is. It's a flexible structure, my dear students. Just above this notochord, this part, it is your dorsal hollow nerve cord, right? So it is not solid just like non-chordates. So if non-chordates will have nerve cord, that will be solid, that will be double. Here it is single dorsal hollow. These are the pharyngeal gill slits. But gill slits means you are talking about the openings. They are not the gill. They are the, like in the pharyngeal region, you have certain openings by which the exchange is there. And this one is the post-anal tail. So these are the four basic characters. These are the four basic characters that we have to study. Is that clear? Then other than that, no doubt, because we are also the chordates. Our heart is going to be on the ventral side. These are the points which we need to focus. There is no point in that, right? There is no point in that. But up to this part all clear? So aquatic chordates and lower chordates, they will have gill slits for throughout the life. But in other organisms, the gill slits will be there in the embryonic forms, because later on, what will be there? Lungs will be there. So now remember this flow chart. We have phylum Chordata. Here, bache, we have three subphylums. Subphylum is not the broad category or the essential category, but here we need to remember. So here you have trick, you can win. This win is not W, this V. U mane urochordates. C means cephalochordata. V means vertebrata. You can win. This urochordata and cephalochordata, they are protochordates. They are the primitive one, and they are acraniata. They don't have cranium. Invertebrata, they are also the craniata, means they have cranium. What is this cranium? This cranium is the brain box. What is this cranium? This cranium is the brain box. This cranium is the brain box. Am I right? So you can win. That is the first thing. Is that clear? So they are acraniata or lower chordates, protochordates, right? And these are the advanced ones. So firstly, tell me this part is clear. There are two flow charts that we have to discuss here, but you tell me this part is clear. Are you sure? So these two subphylums, they are also exclusively marine. Now, in the paper, you have to make sure what are they asking. Are they asking the phylum or the subphylum? What are they asking? Are they asking the phylum or the subphylum? You have to keep it in your mind. Take. Done. Take next part. Now when this subphylum vertebrata is there, so beta in subphylum vertebrata you have two division. One is Agnatha, that is without jaw. Gnethostomata, jaw is present. Now here you will talk about the class Cyclostomata, having circular mouth. There is one more class there, Ostracodermy, but here all members are extinct. So in NCERT, we just discussed this part. Now in Gnethostomata, you have two superclass. This one is Tetrapoda, and you'll have superclass, firstly you will write Pisces. Firstly we will write what? We will write Pisces. And here we will write what? We will write tetrapoda. We will write tetrapoda. Okay? So under this superclass, you have, there used to be one class placodermy, but again, all the organisms are extinct here. So you will talk about the class Chondrichthyes, right, where we discuss about the cartilaginous fishes, and Osteichthyes, where we discuss about the bony fishes. And when it is tetrapoda, here the trick is aram or aram, whatever you want to call it. Aram or aram, whatever you want to call it. So A stands for Amphibia here, Reptilia here, Aves here, Mammalia here. These are the 4 classes here. Take, so if in the paper they'll ask superclass, tetra means 4, foda means limbs. Here superclass is spices, right? So class placodermy, Chondrichthyes, Osteichthyes, this one is extinct, so we have to focus on these 2. Okay, we have to focus on these 2. So that's something you need to keep in your mind. Okay, so this flow chart is important. It's very important. So Urochordata is beta very sad. You know that? Urochordata is very sad. Okay, I'll tell you why. So S means salpa, A means acidia, D means doliolum. Right? So this is a trick. So our urochordates, they are very sad. They are very sad. So whenever you talk about, right, I'll write it here so that we can just quickly complete it and write it. So Urochordata, but Euro means tail. Right? Here in this case, what's going to happen? These organisms, they don't look like advanced one. Trust me on that when you see their pictures. Urochordates, they have the tail. They have the, in their tail region, in their larval stage, in their tail region, they had the notochord. So on the basis of that, they took the entry in the chordata club. Okay, but later on, when they, in their adult life, they don't have any chordate-like character except the pharyngeal gill slits. Okay, okay, okay. Are you getting my point? Are you getting my point? So all the members, so I told you already, exclusively marine, right? So all protochordates are exclusively marine. We are not supposed to forget it because this can be asked as a question, right? And in the case of these protochordates, here I told you, na, they are protochordates. These 2 are protochordates, primitive one. They all are exclusively marine, and in their case in adult life, pharyngeal gill slits. This chordate-like character is for throughout the life. This chordate-like character is for sure for throughout the life. This is what you have to remember. Okay, this is what you have to remember. So let's focus on this part, guys. Let's quickly focus on this part. So Urochordata, tail, right? So here remember, your adults, they are fixed to the substratum, and larva is motile, right? So larva is advanced, adult is not. Okay, so first of all, Euro means a tail. So what you have to remember, that the notochord is there in the tail region in larval stages only. In larval stages only. Is that clear? Is that clear? And here in Urochordata, you'll see their body is covered by test, which is also known as tunic. So we call them as tunicates. So this is extra, but this test point is important. Take, they are tunicates. Take, okay, okay. So Euro means tail. So just because they have the larva in their, notochord in their tail region only, they just got the entry here in the chordata club. Rest, they don't have any chordate-like character, right, for the rest of the life, right? Right? Even the dorsal hollow nerve cord, it is also there in the larval stage. It is also there in the larval stage. Is that clear? Is that clear? Is that clear? So now, bache, can I say that larva is comparatively showing advanced character and adult is showing primitive character. Matlab it's like you in class 10th you scored 98%, in class 12th you scored 60%. So you are not progressing, you are regressing. So this type of metamorphosis is retrogressive metamorphosis. Retrogressive metamorphosis, where adult is showing the primitive character but larva is having the advanced character. Is that clear? Is that clear? Is that clear? So dorsal hollow nerve cord is in the larval stage only, then it is replaced by, in adult they have just nerve ganglions. Okay? So imagine, so larva is advanced but the adults are not. ठीक? Okay? Right? Nerve ganglion or neural ganglion. ठीक? Is that clear? Is that clear? So blood vascular system is also open type. Okay, it is also open type. Any doubt? So it is a pharyngeal gill slit character that will be there throughout the life. But all chordate-like character are there in the larval stages, but in adult only pharyngeal gill slits will be there. Right? So blood vascular system is open type, but because it is a chordate, so heart will be on the ventral side. Heart will be on the ventral side only. Take? Heart will be on the ventral side only. So they have the retrogressive metamorphosis, right? So that is the trick here. Selpa acidia doliolum. Selpa acidia doliolum. Selpa acidia doliolum. Next. So only cephalo. Remember beta, it is the pharyngeal gill slit only that will remain throughout the life. Then comes cephalochordata. Cephal word is associated with head. They are of course exclusively marine. They are also exclusively marine. Right? Here also, take. So now let's suppose in this case, what you people will see, you will see that notochord will persist throughout the life and it is present from head to tail. Right? So notochord persists throughout the life and is present from head to tail. Am I clear? It is present from head to tail. Take. So larva and adult, they both are free swimming. Here you see progressive metamorphosis. Matlab adult is advanced in comparison to larva. Okay, right? Right? Okay, so notochord will persist throughout the life. Take, right? Okay, so blood vascular system here, it is of closed type. It is of closed type. Do you have any doubt here? Do you have any doubt here? And here, even in the excretory product and its elimination chapter we discussed now. So beta, here also for excretion, you will talk about protonephridia, also known as flame cells or solenocytes. This is something that we discuss in the excretory products. That is why I'm mentioning it here. Okay, that is why I'm mentioning it here. So do you have any doubt here? Yes, do you have any doubt here? So this is the first complete chordata. It is having all chordate-like character throughout the life. Take throughout the life. Okay, chaliye. So this table is clear. Now we have to move very quickly. We have to move very quickly. We'll jump to Cyclostomata. We are about to finish it, right? And trust me, before 10:30, I'll give you the break. I can understand that you people are hungry, but if I'll give you the break, you know, now this momentum, we have to spend some time to regain it. Okay, and let's start another chapter after the break. After the break, we have to complete. What do you want to complete? Structural organization in animal first, or you want me to start breathing and exchange of gases? Yeah, neural control and coordination first. Tell me, which chapter you want me to start first? SOIA, structural. ठीक, chalo. Without wasting any time, let's go. So this table clear? I believe it is. This part is done and dusted. Cyclostomata, circular mouth. Let's pick up the most important points here. Circular mouth. Why? Because they don't have no jaw. So they are ectoparasites, not ecto. Like if there is a large fish, they'll join with that. If there is a large fish, they'll attach with that large fish, and whatever food that large fish will leave, they can even eat that. So another point is they have 6 to 15 pair of gill slits. If in the paper they are writing 6 to 15 gill slits, this is incorrect. It should be 6 to 15 pairs of gill slits. 6 to 15 pairs of gill slits. Okay. So sucking and circular mouth is there without jaw. Body is devoid of scales. Means scales are absent. Not just scales and even paired fins. Just like true fish, they don't have paired fins. They are absent. Is it important? Very important. Right? But yes, because they are craniate, cranium is there. Vertebral column is there, because you know that when it is the phylum vertebrata, what is going to happen in phylum vertebrata? The notochord is replaced by vertebral column. The notochord is replaced by vertebral column. The notochord is replaced by vertebral column. Take, so circulation, closed type, of course. They are marine but migrate for spawning to fresh water. So after spawning, within a few days, they die. The larvae after metamorphosis return to the ocean. So petromyxin and myxin is there, which is hagfish. So here in myxin, right, no migration is there. Myxin will not show any migration. There is no migration here. These are the important points that you can write. Take care. And it is this petromyxin, beta, it is an ectoparasite on true fishes. True fishes means I'm talking about the Pisces here. I'm talking about the Pisces here. Take, okay, right? And which larva do they have? Ammocoete larva. So it is a connecting link in between this and cephalochordates. Okay. So myxin or hagfish, it is attached with the gill of the host. It will not show migration. Why are you telling this to us, ma'am? Because it is a previous year question. It is a previous year question. Okay. So usually in the Cyclostomata, larval stages are absent, but your petromyxin will show the larval stages, and that will not be there in this myxin part. Okay, so ammocoete larva is definitely there, right? But here in this, you will not see this. Take, done. Cyclostomata. Take, next. Chondrichthyes, cartilaginous fishes. They are marine with a streamlined body. Cartilaginous endoskeleton is there. Here mouth is located ventrally. Now, when you will compare them with Osteichthyes, in their case, you will see the mouth is a terminal. We see the mouth is a terminal. That's how we have to remember. So, notochord is persistent throughout the life. Gill slits are separate. They are without operculum. So, in Osteichthyes, with operculum they are. Right? They have this operculum. Right, the skin is tough, containing minute placoid scales, right, and these placoid scales are dermal in origin, right, they are dermal in origin. Now bache, in the case of Osteichthyes, there are the scales like cycloid, ganoid, ctenoid scales are there. Take, ctenoid scales are there, but here the scales are placoid. That is also a PYQ. Right, teeth are modified placoid scale, backwardly directed, jaws are very powerful, and they are predaceous. They are predators like shark. Shark is the example here. Okay? So Chondrichthyes, also known as elasmobranchi. Right? Osteichthyes, teleosti. Right? So here 5 to 7 pairs of gills, and they are mostly viviparous. Right? Is that clear? Sure. Now, due to the absence of air bladder. So, here air bladder is absent, right? So, no air bladder means no buoyancy. Now in Osteichthyes, ear bladder present, ear bladder present, right? These are the points. That's how you can compare and revise. That's how you can compare and revise. So they have to swim constantly so that they can avoid the sinking. Heart is two chambered beta, one auricle, one ventricle, but two accessory chambers are there, right? And that chambers are sinus venosus, truncus arteriosus. Take accessory chambers are important. Take so torpedo, edo, E, electric organ. Trigon, G is there. Sting, poison sting. So torpedo having electric organ, right? Electric ray it is the having the poisonous sting. It is the stingray. That's how you have to remember. Okay, that's how you have to remember. So trigon, G, gun. You can remember sting ray it is, electric ray it is. Right? Baki they are beta cold-blooded. Poikilotherms means they cannot maintain their internal conditions constant. Only birds and mammals, they are the ectotherms. That's what we have to remember. So sexes are separate means unisexual they are. Males are having the pelvic fins that bear claspers, that is basically to open the female genitalia. So internal fertilization, and many of them are viviparous, important. So pristis, it is the sawfish bache, right? So if in paper it is written out of silverfish, devilfish, out of silverfish, devilfish, cuttlefish and sawfish, which one is the true fish? Out of silverfish, cuttlefish, devilfish and sawfish, which one is the true fish? Which one is the true fish? People quickly. Which one is the true fish? Which one is the true fish? Sawfish, pristis, sawfish. Take baki this Carcharodon, great white shark. There is one chimera. Chimera. It is a connecting link in between cartilaginous and bony fishes. Any doubt? Any doubt? And the study of fishes, ichthyology. Take ichthyology, again a PYQ, that's why I'm discussing it. Okay. Chali. Next is Osteichthyes. So this is the pristis sawfish. This is the Scolodon dogfish, right? Osteichthyes, the word that we can use here is teleosity. Take, right? Teleostomy rather. Teleosity, nahi, teleostomy, Ambika. Teleostomy it is. Okay, so they can both marine and freshwater, bony endoskeleton, body streamlined, mouth is mostly terminal. Not always, but mostly it will be terminal. So here, how many pairs beta? 4 pairs of gill slits are there, right? 4 pairs of gill slits are there. That's how we have to compare. You know that they have air bladder. I told you about the scales as well. So there they have air bladder. So buoyancy is there. Heart is again 2 chambered. And you know that in fishes, the heart is venous heart, right? The heart is venous heart. Why? Because it pumps deoxygenated blood only. Deoxygenated blood only. You know that here there is the single circulation. There will be gills, right? There will be gills for oxygenation. There will be gills for oxygenation. They are also cold-blooded. The fertilization is external, mostly oviparous. Development is direct. So Exocetus, flying fish, because it's, and the fins are paired here, because you know na, that fins, that pectoral fin is quite elongated there. Hippocampus, it is the seahorse. And in the animal kingdom, here, it is the only male which basically, right, here, the male is having the brood pouch beta. It is the only male which carries the baby. Yeah, it is also known as the male is having the brood pouch. So it is also known as pregnant male. So in the entire animal kingdom, it is the only male member that is pregnant. So female will release the gametes in the brood pouch, right? And there, you know, further the babies will develop and the male is going to take care of that. Baki labio, catla, clarius is megar, aquarium fishes are beta and pterophyllum. So yes, we have to remember it. Do it quickly. Okay. So pregnant male is your seahorse, hippocampus only, right? So there is a pouch, right? And that is a brood pouch. So male will collect the egg there. So it is secondary vivipary. So parental care is also there. Is that clear? Parental care is also there. Is that clear? Sure. So no cloaca is there here. Well, cloaca, you know, it's a common chamber in amphibians and reptiles. We'll discuss that. Having digestive system, excretory system, reproductive system, all will open in cloaca. It will be absent there. It will be absent there. Now let's move to the next part. Pregnant male done. Amphibia. Amphi means dual. They have dual habitat. So here they need water for the right sexual reproduction. So you know that most of them are having two pair of limbs, right? So body is having head and trunk. So there is no neck. Why I'm specifying this? Because frog is the part of our syllabus. So we need to know that. Tail may be present in some. So jaise anura, apoda, urodela is there na? Apoda without tail. Anura, tail is there. One is urodela. No. Apoda without limbs. Anura, basically we have two words, three orders here under this class, amphibia. We have the order Anura, we have the order Urodela, we have the order Apoda. Apoda mane without limbs. Ichthyophis is the example, limbless amphibian, right? Anura, your frog, right? So then there is Urodela, tailed amphibians will be there, like your salamander. Okay, so skin is moist without scales, right? Eyes are having eyelids. Tympanum will represent the ear. So tympanum is the middle ear. External ear we will only see in the case of mammals. So alimentary canal, urinary, and the reproductive one in open into common chamber called cloaca, which will open exteriorly. So you know that gills, lungs, and skin, everything is used for the respiration. Heart is 3 chamber, 2 auricle, 1 ventricle. So in this case we'll see incomplete double circulation. What are we going to see? Incomplete double circulation. Take sexes are separate, fertilization is external, they are oviparous, development is indirect. Examples are important. Okay? So as I said, we have a lot that we can remember, right? We have a lot that we can remember, we can discuss here, but yeah, we have to stick to the topic, right? So they are also poikilotherms, so they have to go for the hibernation, estivation. That's it. So I believe this part is clear. I believe this part is clear. Are you sure? Are you sure? So in this case also, because frog is the part of our syllabus. So remember, renal portal system and hepatic portal system both are present. Both are present. Next, so tailed amphibian, take axolotl larva which show neoteny. That is what we discussed, but chalo anyhow. So class Reptilia, right? Name referring creeping or crawling mode of locomotion is there. Mostly terrestrial, their body is covered by dry and cornified skin. They are the true terrestrial animals because lungs we are going to see here. Okay? So their scales are epidermal. This is what you have to remember. So they don't have external ear opening. External ear we will see in the mammals only. So tympanum will represent the ear. Limbs when present are 2 pairs. And here also this example of Ichthyophis, limbless amphibian. Ichthyophis example is very important. Ichthyophis example is very important. Okay? Okay, so heart again beta here usually 3 chambered, or you can say that, let's suppose auricle ventricle, the ventricle is partially divided, take, but we have 4 chambered heart in the crocodile as well. They are also poikilotherms. Snake and lizards, they shed their skin or skin cast, which is known as ecdysis or molting, right? Of course hormones are going to play the role here. Take, so sexes are separate, that is internal. Examples are important. So beta chilon or testudo, how will you differentiate? This one is aquatic. Here you will see the digits, they are not present. This part is fused here. Here you will see some digits like this. Chameleon, tree lizard, calotus, garden lizard, crocodile, alligator, hemidactylus is the wall lizard. So do not confuse calotus, the garden lizard, and hemidactylus wall lizard. Baki beta poisonous snake mein naja, cobra is there, naja, cobra is there, right? So Bungarus and vipera. So write it down. So again, as I said, we don't have any choice. We have to stick to this. So this Bungarus is basically, It is the poisonous snake. It is neurotoxic. Indian cobra, naja naja is also the poisonous one. Vipera is basically, right? Vipera is also the viper scale. Okay. Take any doubt? So even remember python. Python is non-poisonous. Do not confuse it ever. It is not at all poisonous. Python. It is very dangerous, but it is not poisonous. Okay. Okay. Sure. Right? And do you know one Heloderma? Only poisonous lizard. It is the only poisonous lizard. It is the only poisonous lizard. Okay. Okay. Heloderma. Only poisonous lizard it is. Heloderma. Okay, Gila monster. It's Gila monster. Only poisonous lizard it is. Take doubt? Okay, any doubt? Sure? Right? Any doubt? Take. So, baki beta, it should be bun, B-U-N, bungarus, like this. So, do not make the mistake here. Okay? Do not make the mistake here. Take. Common crate it is. It is not the crate, even it is the common crate. Okay. So, poisonous one examples are very important. Anything else? 9. That's it. So, Indian cobra, bungarus, the bandit crate, done. Okay. Then, ये देखो, this is testudo. नहीं, testudo tortoise, chelon. Chelon it is. Chelon it is. Turtle it is. Testudo is tortoise. Then aves, birds. They are, yes beta, they are warm-blooded. They are warm-blooded. Here you have to remember that they have ear sacs that will increase the efficiency of respiration. Here you have to remember the bones are pneumatic, mane hollow bones, right? Hollow bones. They have ear spaces. Okay, these are the points. So birds, they have the feathers and they can fly except your flightless bird, right? So they have the feathers. This is important. This is what we see here. Okay, feathers are there. Feathers are there. Okay, what do they have? They have the feathers. And yes, one more thing. Your fishes, your amphibians, they are amniotes. जैसे amnion is an extraembryonic membrane ना. बाकी your reptile, birds, your reptile, aves and mammals, they are amniotes. And they even have 12 pairs of cranial nerves. Sorry, these two, aves and mammals, are having 12 pairs of cranial nerves, right? Right, just we also have 12 pairs of cranial nerves. So even your reptiles, they are also having 12 pairs. So fishes and amphibians, they have 10 pairs of cranial nerves. Rest all are having 12 pairs, right? Rest all are having 12 pairs. Take, chale. So they possess beak, right? Jaw is modified into beak. The forelimbs are modified into wings. Hind limbs, they are having the scales, which are modified by walking and clasping. So skin is dry without gland. There is only one gland, which is the oil gland. But this oil gland is also known as preen gland. It is also known as uropygial gland because it is present there in the tail region. Now, so just birds, they'll, with the help of their beak, they'll take the oil and they'll do the oiling of their feathers as well. You might have noticed this. So this is what you have to remember. Endoskeleton is fully bony and long bones are hollow with ear cavities. Why? Because they have to make their body light so that they can fly. Baki digestive tract is having additional chambers. The crop and gizzard. That's a PYQ. Bache. That is again a PYQ of 2022, I believe. Okay, so this crop and gizzard, we also discuss it in cockroach also. These are the additional chambers. We also discuss it in cockroach also. That's how you have to compare the things and you have to revise. So G for gizzard, G for grinder, right? And it is also known as proventriculus. Proventriculus. So in the crop, the grains will be stored and here it will be grinded. Heart is completely four-chambered, warm-blooded, that is homeotherms, right beta. And endotherms, they are able to maintain a constant respiration is by lungs. Air sacs are attached to the lungs. So air sacs will not help in gaseous exchange, right? They will have the air, they'll pass it to the lungs. So ultimately lungs are going to, right? Lungs are going to do that gaseous exchange. So air sacs will just supplement the respiration. Right? And here in this case, in the case of Aves, it is the syrinx that is basically sound producing. That is basically sound producing. Syrinx, that is basically what? That is basically the sound producing organ. So they do have the larynx, but when it comes to producing the sound, it is the syrinx in the birds. It is the syrinx in the birds. Is that clear? Sure? Sure? So sexes are separate, fertilization is internal. So in their case, we talk about the keel sternum to which the flight muscles are attached. Right? Right. So do write it, beta. So that's it. Baki, nahi, that will be 2 extra for you. So cranial nerves are 12. And that's it. Take. Yes, any doubt? So this part is noted. Right? So overall in the birds, urinary bladder and copulatory organs are absent. They don't have urinary bladder. They have to make their body lighter, na? And even the copulatory organ, it is absent. So examples are important, right? But examples are important. We cannot leave them. Literally, we cannot leave them. Okay, they are important. Baki Archaeopteryx lithographia. Do you remember? That is a missing link, not connecting, missing link in between the reptiles and birds. And that is why the birds, they are also known as glorified reptiles. They, like the birds evolved from reptile-like ancestors. That's what you have to keep in your mind. Okay. That is what you have to keep in your mind. Any doubt? Any doubt? Chaliye, so let's move to the class Mammalia, right? So study of birds is ornithology and study of mammals is mammalogy. Baki ye dekho ear sacs, right? Ear sacs are there. Baki Mammalia, right, highly advanced. So they are in polar ice cap also, deserts, mountains, forests, grasslands everywhere. They can also fly, they can also live in water, of course, to the land as well. So unique character is the presence of milk producing gland, mammary glands, beta. And here are also there, you know that. Okay, so these here are also there, that is very unique here. And here in the mammals only, you will talk about the external ear. And even in the mammals only, we talk about the diaphragm, right? We talk about the diaphragm. And liver is also having the glissons capsule. That is something that we see in the case of mammals only. Okay, heart is 4-chambered, homeotherms. Baki, that's it. Examples are important. So now here, I'll just add few, right? Few points here, right? And that is not too much, right? That will be something basic only, beta. But I want you people to know about it. Just when we talk about the mammals, class is Mammalia. Right, class is Mammalia. So it is having further classification. So accordingly, na, we have to discuss it. Right, accordingly, we have to discuss it. So don't worry, I will not take your much time. Okay, so just few important points I'm going to discuss with you all. Okay. Any, right? जैसे mammals में overall ना, class is Mammalia. So here we also talk about Prototheria and Eutheria. Do you know that? Prototheria and Eutheria, right? What do we discuss? Prototheria and Eutheria. And in that Eutheria, right? Prototheria and Theria. In the Theria we will discuss. These are the Eutheria and Metatheria. Ma'am, do we need to remember this? No. Then why are we discussing it? There is just one or two points that I want you people to know regarding your examples only. Okay? It's not something that I'm picking up extra. I will just discuss the examples here, right? So these are the subclasses, these Prototheria and Theria, and then a Prototheria and Theria. Then they have the infraclasses, right? Then they have the infraclasses. So basically, जैसे we have the examples ना, examples Prototheria. We have the example of Ornithorhynchus. Ornithorhynchus. It is your duck-billed platypus. Right, it is your duck-billed platypus. In the case of mammals, we know that they have placenta, but this one is your egg-laying mammal. So it's a connecting link in between reptiles and mammals. Okay, it is the egg-laying mammal. It is a connecting link in between reptiles and mammals. Right, bache? It is a connecting link there. Now, Eutheria. Basically, they are the subclasses, they are the infraclasses. So, Metatheria and Eutheria is there. So, here in Metatheria, you are talking about the marsupials, right? Kangaroo, right? Here the placental one. So, marsupials or metatherians, we have the Macropus. Macropus. M for Macropus, M for marsupial, right? So, then obviously it is your kangaroo. It is your kangaroo. That's how you have to remember the example. Rest all will come under the Eutherians, okay? Rest all will come under the Eutherians, okay? Right? So, yes, you should know egg-laying mammals and their example. So, egg-laying mammals are Prototherians. Monotremes they are beta. Monotremes they are beta. So, examples are important. Okay, examples are important. So, duck-billed platypus is there, and other than that, one is your spiny ant eater, echidna. Take, it is your echidna. Okay, right? So they are egg laying. They are what, bache? They are egg laying, right? So this echidna is spiny ant eater. Okay, it is spiny ant eater. Done? Sure. Spiny ant eater. So this is all about the animal kingdom. Wow, this is for the very first time I finished it in 3 hours, right? Otherwise, I always take 4 hours here. Dekha, even I am evolving. Okay, I'm learning this, how to finish everything, right? How to finish everything in 2 hours or 3 hours and with proper content which is required. I'm not saying I have shared every particular information with you all, no, but we have completed the entire NCERT. Rest is the table for the revision. And now, of course, right, the most weighted thing, the break, right? So let's have a break for 30 minutes. Let's have a break. Hannah? Kit Kat break. Okay, let's meet at 11 p.m. Then be ready for next 3 hours. So which chapter you want me to take next? So Wasim sir, seriously, you want me to call him? Chalo, we'll call after break. Okay, we will call after break. Next is human physiology. Okay, we'll pick up human physiology. Even I am evolving literally. I hope I'm perfectly audible. Okay, what do you want me to start? Ma'am, call Wasim sir. Arey, kyun? Let him enjoy his day. Let him enjoy his day. Okay, so what do you want me to start? Biomolecule, structural organization, biomolecule, cell cycle, human physiology, ma'am. Acha, theek hai. Okay. I have one simple solution here. Just a minute. Just allow me 1 minute. Huh. I believe it's clear to all of you. Okay. So, how many of you want me to start structural organization in animal? I'll see. How many of you want this? Cell cycle. See, bache, cell cycle, देखो, रुको. I'll tell you one story. I'll tell you the issue with the students. See, we started top 50 concepts, right, of biology, in which I took 3 classes and I have completed the majority of topics there, right? Literally, I have completed the majority of topics there. But many students are like, Arey, wow, you are just changing the series. You're not teaching us this. You're not, Arey, I am regular. I'm teaching everything. You just check the content on Unacademy NEET English channel. You will get all the chapters of biology. Okay, you are literally going to get all the chapters of biology. One student mentioned, ma'am, human health and disease in detail is not available. It is also available. Do check the channel. Right, number one. Now, I am, right, regularly academic sessions and everything are there. Cell cycle, I have discussed multiple times. In the top 50 concept also, I started with that. And now, as I said, that when it comes to our channel, we are providing you all the essential things. Jaise here dekho, plant growth and development in 20 minutes, right? All the important questions are covered there. Cell cycle and cell division, right? This is again the short series where I have completed this topic, right? So I have already provided you the content which you need for the NEET examination. Okay, you are still asking for the same chapters, right? And you're not attending the classes properly. So you tell me, what do you want? Neural coordination, theek. Okay, anything else? Chalo. So just for your satisfaction, let me tell you, if you talk about structural organization in animal chapter, bit of frog and cockroach we are not going to discuss today, right? I have a very simple logic. These chapters, they need minimum, both these chapters, they need minimum 3 hours, right? Minimum 3 hours. Minimum, I'm saying, I'm not saying maximum, minimum 3 hours, these 2 chapters they need. And you will get either 1 or 2 questions, right? We have another important topics also from where we can get 3 to 4 questions, right? From where we can get 3 to 4 questions. So don't you think that we need to focus there, right? Don't you think we need to focus there? That is more important. Yes? Don't you think so? है ना? So cockroach and frog, before 15th of April, it will be there on our channel in the recorded format. That's a promise. I rarely use this word promise. That's a promise. Frog and cockroach chapters, right? These two topics rather, they will be on Unacademy NEET English before 15th of April, right? Before 15th of April. ठीक है? Okay? Okay, 1 minute. Done? Okay, chaliye. So let's start, right? Let's start the chapter. Let's start the chapter. But yes, I have done some more homework for you, right? So here one more PDF is there. So here I have simply mentioned the examples, because when it comes to the tissue part, right? When it comes to the tissue part, they ask the questions from the examples only, right? Which we have to remember. I'm not saying that I'm not going to discuss this chapter. I will, right? I will. For sure I will. But let's say, जैसे majority of students are, अरे भाई cockroach and frog is the part of syllabus. Those who are saying it is deleted, right? Do reconsider your decision of giving the NEET examination. At least here you should know about the syllabus. Okay. So see, this is the homework that I have done for you. This is the homework that I have done for you. Okay, so I'll share this PDF in the Telegram group, in our official Telegram group, and even in my Telegram group, that is Ambika Sharma Biology. So here you can see the examples are written directly. This is simple squamous epithelium. Another names are pavement epithelium, wavy in appearance it is, tessellated epithelium it is, right? So alveoli of lungs, endothelium, women's capsule, mesothelium, they are having the simple epithelial tissue. Right, they have the simple squamous. So squamous means irregular cells, wavy kind of cells are there. So basically the exchange will be occur through this. When it is simple cuboidal, that is a PYQ. That is a very important PYQ, right? Your germinal epithelium that lines your gonads, right? That is basically the simple cuboid epithelium. Beta, it is believed that this germinal epithelium, it makes germ cells, but it is not true, right? It is not true. Precursor primordial germ cells, they'll find their way from the yolk sac to the gonads. They are going to form that. They are going to form that. So happy with this also? Happy with this also? All the examples are written here, right? All the examples are written here. So germinal epithelium is the simple cuboidal epithelium, again the PYQ. So tubular part of nephron will have, sweat glands will have that. Now when I'm saying brush bordered cuboidal cell, they are cube-like and they have finger-like enfoldings, right? So that's why we call them as brush bordered. That's why we call them as brush bordered. Okay, so they have these microvilli, finger-like enfoldings. So of course they are here to increase the surface area, without any doubt, right? Without any doubt. Okay, so proximal convoluted part of proximal convoluted PCT, proximal convoluted part of nephron. So again, it is also a PYQ, right? When it is ciliated, so cube-like cell, they are having cilia over it. The collecting duct is the example. Now let's move to the next part, simple columnar epithelium. So columnar, what kind of cells are they, beta? Right, when we are saying simple columnar epithelium, what kind of cells are they? They are tall and slender and the nucleus is present at their base. They are tall and slender, the nucleus is present at their base. So bile duct and liver is there. Bile duct and liver is there. Then comes brush bordered. So when it is brush bordered, it's simple. These tall and slender cells, they will have finger-like enfoldings like this. Okay, so gallbladder is there. When it is glandular, means it is secretory, right? But those who are not able to understand, I cannot help it. Right? I cannot help it. This is a simple chapter, tissue. And you know that from this part, the examples can come in the paper. And I'm picking up all the PYQs. What else you want me to teach you here? Right? There is a detailed one shot of this chapter as well. You can simply check it. Okay? You can simply check it if you want to. Okay? Right? Okay? Done? And I don't know, why do you people think that you will, not study for the complete year and all of a sudden in one marathon you want each and every topic, right? And then you think, nahi, we'll complete each and every topic in just one session. It is not like that. It is a quick revision session. You have to understand this thing. You have to understand this thing. It's a quick revision session in which no doubt I'm picking up the things from the basics, but we have to remember so many things. So let's focus on the things which are very important for the exam. So that is why all the examples are given here. We will discuss it as well, right? If you want a particular chapter only, you know that the content is available on our channel as well. Okay, so frog and cockroach, this is my suggestion. If you have completed your entire syllabus, then pick frog and cockroach, because one question will come from that part, right? One question will come from that part. So if only one question is going to be there from that part, then why to waste 3 hours for that topic? I'll put the separate videos, the brief one from where you can revise it quickly, right? Don't you think it's the best option right now? Don't you think it is the best option right now? Tell me please, don't you think it is the best option that a teacher is going to put the recorded video, and that is very, that is not something easy for me. That is going to take a lot of time if I have to put the recorded video, and that too very short. Right? So why to waste 3 hours for one question when we can finish most important topics in that 3 hours? Hannah, isn't it good? Isn't it good? You tell me, isn't it good? So if you all are aligned, then I want to see some fire emojis in the chat section. I literally want to see the fire emojis in the chat section if you are aligned. Yes, ma'am, it's obvious. It's natural. Hannah? Quick. Yes. Quick. So ready to start it? So again, simple columnar epithelium, tall and slender. And when it is the simple is the word, matlab it is single layered, right? Compound may it will be multi-layered. So when it is brush bordered, matlab your tall and slender cell, they will have finger-like projection, right? They will have microvilli, right? Gallbladder is the example. So when it is glandular, glandular means it is secretory, it will release something. Okay, stomach is having glandular columnar epithelium. So when it is glandular brush bordered, whenever you will read the word brush bordered, it means the cells are having microvilli. It means the cells are having microvilli. And again, beta, this is also a previous year question, that ciliated columnar epithelium is there in fallopian tube. And this year also you can expect this question. So when you talk about the fallopian tube, you know that the ovum has to move. So here you have the ciliated epithelium, but it is ciliated columnar, not cuboidal. Important direct PYQ. Focus on this part. Focus on this part, okay? So ependymal cells of brain ventricles. In the brain, the cavities are there, the spaces are there. That cavities are lined with the cells. This is, suppose in the brain there is a cavity that is lined with the cell. You are calling this cell as ependymal. But what kind of shape that ependymal cell is having? It is having columnar. Right, columnar shape. That is how you have to read the points. So pseudo means false, stratified means multi-layered, right? Pseudo means false, stratified means multi-layered epithelium. So what is this? This is another important PYQ. So I have picked up all the PYQs here from the epithelial part. Right, I have picked up all the PYQs there from the epithelial part. So you have pseudo, stratified, ciliated, columnar, glandular epithelium. Pseudo, stratified, ciliated, columnar, glandular epithelium. Okay, so where will you get it? In your windpipe trachea, your bronchioles. They are going to have that. Okay, they are going to have that. Okay, next part, compound epithelium. So when it is compound, it is multi-serrated or multi-layered. It is. It is multi-serrated, it is multi-layered. Is that clear? It is multi-serrated, it is multi-layered. So it is the stratified. Stratified means layered it is. Stratified squamous is the word. The topmost layer will have flat, irregular shaped cells. Remaining layers can have any type of cell. So when, right, you talk about your skin, we say now our skin, right, keratinized it is. Keratin protein. You know, keratin protein, the topmost layer of the cells, here it will become, this layer will become dead. Why? Because the cytoplasm here will be replaced. The cytoplasm here will be replaced by the keratin protein, right? And it will not allow the water to get evaporated as well. So keratinized stratified squamous epithelium, your epidermis of skin is having that. Right, the topmost layer, it is the dead layer, right? It is the dead layer, it is the keratinized one. It is having the keratin, it is having the keratin. Are you getting my point? It is having the keratin. Are you getting my point? Okay, okay. Done. So epidermis of skin, horn, nails, they all have that. Then non-keratinized stratified epithelium, right? It again, it is multicellular, right? Sorry, it is multi-layered. It is multi-layered squamous epithelium, but keratin is not there, like your buccal cavity, pharynx, and cornea of eye. So this cornea of eye point is important. This cornea of eye point is important. Right, bache? This cornea of eye point is important. Next part, stratified cuboidal. Here, the topmost layer will have cube-like cells. right? So your pancreas, salivary gland, secretory ducts are having that. I'll tell you which one is a PYQ. Baki stratified columnar, again, the topmost layer will have tall and cylinder cell. So if they are having cilia, like in the case of pharynx, non-ciliated epiglottis, not that important. Take baki glands, bache, endocrine, exocrine or heterocrine, which chemical control and coordination chapter mein we will pick. Baki you know that on the basis of nature of secretion, the glands are of different types. Jaise eccrine, apocrine, holocrine. In the eccrine, this gland, when it will pass its secretion, it will simply do exocytosis. Means something will get synthesized in this gland, right? It will just simply release it. Exocytosis, after outside the cell. The cell will not rupture at all, like your sweat gland, goblet cell, that is your mucus cell, mucus gland, your salivary gland, mucus gland. Take apocrine, A for apo, A for apical, mane tip. So here in this gland, the secretory substance will be accumulated here at the top. So what will happen? This upper portion will get removed, right, bache, will get removed. This is the mammary gland is the example. This is important, PYQ. And then holocrine, where holo means complete. That's how you can remember. So whenever in the cells here, something will get synthesized, they will rupture, they'll pass their secretion. These are the examples here. So I have picked up the most important things, the most important examples for you, or from the epithelial tissue part, so that you do not miss it at all. I know we have to discuss the connective tissue as well, but muscular tissue, neural tissue, so we are going to complete, right? Neural tissue and muscular tissue we are going to complete in human physiology. Connective tissue also I will discuss. For epithelial tissue, I have mentioned all the important examples here, right? So why I mention everything on a paper, right? Why I uploaded it here? So that we can save the time, right? These are simply the examples that you have to keep in your mind. What do you want me to teach you? Cuboidal epithelium, where the cells are cube-like. Columnar epithelium, where cells are tall and cylinder. Of course not. Of course not. We just need to focus on the examples here, right? We just need to focus on the examples here, and other important questions also I will pick up, okay? No worries. No worries. Why are you people getting worried when I'm saying we are here and we'll complete everything? Okay, then, right? So what's the point of being sad? Take. Okay, chaliye. So let's start the human respiratory system, and in 1.5 hour, let's revise the most important topics. So are you ready? Are you ready? Lalita, again I'm telling you, I just picked up the most important topics. We will discuss this topic in detail, but firstly let's finish the other important part. From structural organization in animal, if I have to talk about the tissues, 1 or 2 questions will be there, right? 1 question will be there from the epithelial tissues example that we have discussed. We'll share the PDF with you all. Second question, right? From cockroach and frog part, yeah, or it can come, but other than that, any muscle example or something like that can come, and that also we are going to discuss, right? That also we are going to discuss. So why you people are getting worried about it, right? You know that we are here. If we have planned to take your marathons from 7 to 7, right, for 10 hours, for 12 hours, we have the plan, na? We know what to deliver. So don't you people trust that? Don't you people trust that? Don't you? Chalo. So next is the human respiratory system. Next chapter that we are going to pick is what? It is the human respiratory system. Dekho beta, again, my agenda right now is what? My agenda right now is to complete the most important things first, because I know, again, still you are charged enough. Right, still you people are charged enough to complete these topics. Later on, what's going to happen, right? You people will drop down. You will be like, okay, we'll watch these lectures again, and that is not going to happen. Trust me on that. Trust me on that, right? If you think, nah, I'll watch the lecture tomorrow morning, it's not going to be here. It's not going to be there, because again, you have the chemistry, right? After that, again, you have the chemistry, physics, and biology. Right? This is what you people are going to have. So if you think that today I will not study and tomorrow I will definitely manage it, I will definitely pick it up. No, it's not going to happen. Trust me on that. Trust me on that. Right? You have wasted your sufficient time. Right? And whenever you think that you are tired, just ask one question from yourself, that this is what I have done, right, in this 1 year. But now it's not the time. Now it's not the time. So this is something that you have to remind yourself. Okay, and you will feel motivated, you will feel charged. Trust me on that. So next chapter is the human respiratory system. Okay, next chapter is the human respiratory system. Dekho, let's pick up human respiratory system, then I'll tell you the sequence now so that even you'll feel good. The sequence is breathing and exchange of gases, neural control and coordination, chemical coordination and integration, excretory product and its elimination, locomotion and movement, body fluid and circulation. Is that clear? That is the sequence. Is that clear? Is that clear? Let's start this, then neural control and coordination as per the student demand I'm going, right? Then chemical coordination and integration, this, and this. And after that, let's go for the biomolecules. After biomolecules, let's pick up structural organization in animal, right? The epithelial tissue, cell junctions, and connective tissue. Because muscle and neural system, so we will complete here. So now ready? Happy? Happy? Chaliye. So let's start. So when it comes to the breathing and exchange of gases chapter, you know that the transport of gas is very important, be it of oxygen or it is of CO2. They both are very, very, very important topic. From the complete human physiology, from the disorders only, we have 6 chapters. All the chapters are having the disorders. And from that disorder part, 3 to 4 questions can come. That too only from the disorders. We are going to discuss them today. But we have a separate session on Unacademy NEET English channel as well for the disorders only. So don't worry about it. It will be repeated as well. Now it is the human respiratory system. So I am talking about the human respiratory system. How can I forget about the lungs? How can I forget about the lungs? Because this is what we have when we talk about what? When we talk about the respiratory system. Now, let's relate it with the animal kingdom a bit. So quickly, everybody here in the chat section, quickly mention the name of the phylums. Everybody here in the chat section, please mention the name of the phylums. Everybody here. If you really want to finish it, if you really want to nail this chapter, then do let me know, people. Do let me know, people, quick. Yes, do mention these names. Done? Are you sure? Are you sure? So from Porifera to Aschelminthes, you know that it is the general body surface through which the exchange will occur. Yes or no? Yes or no? So till then, there will be no respiratory organ. Even in Annelida, respiratory organ is not there. Here we talk about the cuticle. So no respiratory organ, the exchange will be there. How that exchange will occur? Of course, the diffusion is going to happen. Of course, what's going to happen here? The diffusion is going to happen, right? What is going to happen here? Diffusion. So no respiratory organ is there. No respiratory organ is there, right? Diffusion is there, right? Diffusion is there. And of course, gaseous exchange will be through diffusion only, and that is through general body surface. That is through general body surface. Then we have the Annelida. When we are talking about the Annelida, we will pick up the cuticle. Now in the Arthropoda, for the very first time, we will see the respiratory system. Just now I told you that tracheal system is there. Yes or no? Yes or no? Just now I told you tracheal system is there. Where do you see the tracheal system? We will see it in the case of insects. Other than that, book lungs, book gills. Yes, do you remember that? Book lungs, book gills, simple gills are there. Simple gills are there. So where do we see that gills? You remember prawn. What about book lungs and book gills? Let me check your revision as well. What about book lungs? Where are you going to see the book lungs? Spider, basically your arachnida. Your arachnida will have that. And this one you will see in the case of Limulus. This one you will see in the case of Limulus. That is king crab. That is king crab. King crab. Are you getting my point? That is king crab. Are you getting my point? Are you getting my point? Right? Right. Any doubt? Now, when the next phylum is molluscan. Just now we have discussed it as well. Here we talk about the Ctenidia. These are the feather-like gills. Am I right? Now in the case of Echinodermata, do we have the proper respiratory system here? Right. Here you know that ambulacral system is there. So here body surface, here the gaseous exchange will be through the body surface. So when I'm saying body surface, it is called dermal branchia as well. Right. What do we discuss here? Body surface is there. We will call it as what? We will call it as dermal branchia as well. Yes or no? Yes or no? Yes or no? What do we have here? We have the dermal branchiae, and other than that, you know that tube feet and all, right? Other than that, you know that tube feet and all. Now, what about hemichordata, people? What about hemichordata? Chordata, maybe we have discussed it almost, because that is how these chapters are related. So in the case of hemichordata, how that, right, how that gaseous exchange will be there? Tell me quickly how that gaseous exchange will be there, because in chordata you know that fishes are having gills, frog is having the skin, the buccopharyngeal cavity is also there, the lungs are also there, reptiles, birds, mammals, they are having the lungs. But what about this hemichordata? What about this hemichordata? What these hemichordates have? What these hemichordates have? Yes, what about these hemichordata? Tell me. Yes, how will they respire? Anybody? Anybody? How will they respire? Hemichordata. Don't you remember the gill slits in their case? Right? Do they have the gills or gill slits? Do they have the gills or gill slits? Yes. Yes. Do they have the gills or gill slits? So in the case of hemichordata, you'll talk about the gill slits. You'll talk about the gill slits. So we have the lungs. What do we have? Lungs. So when lungs are there, what type of respiration it will be? It will be pulmonary respiration. What type of respiration it is going to be? It is going to be pulmonary respiration. What type of respiration, people? Pulmonary respiration will be there, right? Pulmonary respiration will be there. So what if it is what if it's skin? What if it's skin? If I talk about the skin, then what do you have? What should be the word then in the case of skin? If skin is used for respiration, what should be the word then? Cutaneous. Excellent. And if gills are there, then yes, if gills are there, then so in the case of gills, the word will be, anybody here in the chat? Branchial. The word will be branchial. The word will be branchial. So you know that what do we have. So I will just write the path here, right? Then we will jump to the important topics, because this is something which is very basic, right? So external nostrils, yeah, external nares is there. This is the first thing, right? Through which the air will enter. Through which the air will enter. Yes or no? Yes or no? Then you have the nasal chamber. Then what do you have? You have the nasal chamber. After that, what do you have? You have the nasal chamber. Then after this nasal chamber, bache, we have internal nostril, yeah, internal nares. This is what we have. And then where that air will go? Where that air will go? Of course, that air will go to the pharynx. That air will go to the pharynx. So when you people talk about the pharynx, just remember one thing. Here we will discuss about 3 parts, and the trick here is known. What is the trick here, students? The trick here is known. So it will be nasopharynx. It will be oropharynx. It will be laryngopharynx. Are you getting my point? So nasopharynx is there, oropharynx is there, laryngopharynx is there. So basically, students, air from internal nares, it will pass to the nasopharynx. Now, you know that mouth part is also there, mouth, then buccal cavity, then further that food has to pass to the pharynx as well. So it is the oropharynx which is the common passage. It is the oropharynx overall. In the NCERT, it is written, it is the pharynx which is the common passage for both, right? But here, mainly, what's that? It is the oropharynx, right? What's that? It is the oropharynx. What's that? It is the oropharynx. Done, bache? Done, bache? Done, bache? So naso, oro and laryngo. Naso, oro and laryngo. So after pharynx, what do we have? Right? Just mouth, buccal cavity. Then we talk about the pharynx part. So these are the 3 things that we have. Now from this laryngopharynx, right. Of course, next part that you have to remember is larynx. Larynx, which is also known as the voice box. Right, which is also known as the voice box. It is basically cartilaginous. It is made up of cartilages. Right, it is basically cartilaginous. Am I right? Am I right? Am I right? Am I right? So you know that there has to be an opening here. Right, chaliye. So let's discuss this part. Let's discuss this part as well. So basically, students, your laryngopharynx. your laryngopharynx. It will carry that air, right, of course, to the larynx. So when you are talking about this larynx, this opening here, right, there is an opening here. What do we call it? That opening is known as glottis. That opening is known as glottis. What is the name of that opening, bache? This opening is known as, yes, this opening is known as glottis, and it is covered with a cartilaginous flap, and that cartilaginous flap is known as epiglottis. That is a very famous PYQ. I hope you remember, right? So epiglottis, what's that? It is the cartilaginous flap. What's that? It is the cartilaginous flap. Don't worry, we will discuss everything in the structural organization as well. Take. So you have the larynx. The opening of the larynx is glottis, which is covered with the cartilaginous flap, that is epiglottis. Then of course, it will carry that air, right? Where? It will carry that to trachea. Where? It will carry that to trachea. Yes or no? Yes or no? Yes or no? Right? So this trachea, this is the windpipe, this is the tube-like structure, right? This is how it is going to bifurcate. And when you talk about this trachea, right, you know that here we also talk about C-shaped cartilaginous rings. How many of you remember this? How many of you remember this? So there will be C-shaped cartilaginous rings. They are not completely covering the trachea. They are just C-shaped. They are providing it the protection. They'll make sure that when there is a pressure difference, the trachea should not burst, right? The trachea should not burst. Are you getting my point? So this trachea will be there. So you know that this trachea will, right? This trachea will divide into two. How many of you know that? It will divide it. It will bifurcate. It will bifurcate. And at what level? At what level? Anybody here in the class? This trachea, you know that we are talking about the trachea here. So this trachea, beta, it is what? It is a straight tube. What kind of tube it is? It's a straight tube. It is the windpipe covered with the C-shaped cartilaginous ring. I'm picking up all the important questions here, bache. So at the level of, right, when will it bifurcate, beta? At the level of fifth thoracic vertebrae. At the level of fifth thoracic vertebrae, it will bifurcate into two. Are you getting my point? Are you getting my point? It is a straight tube. It will bifurcate at the level of fifth thoracic vertebrae. It is a PYQ. I'm directly picking up the important question. At the level of what? At the level of fifth thoracic vertebrae, it will bifurcate. Is that clear? Is that clear? Yes, people. Everybody here in the class. Everybody here in the class. At the level of fifth thoracic vertebrae. Right? Right? At the level of fifth thoracic vertebrae. So if you don't remember this part, take, so I am, nahi chalo, fifth thoracic vertebrae, you have to remember it. Then you have primary bronchi. Then you have these primary bronchi will further divide, they will form secondary, then tertiary, and this is how it is, right? This is how it is. So after a trachea, right? After trachea, you will get primary bronchi, secondary and tertiary. Yes. After trachea, this is what you are going to get. Primary bronchus or primary bronchi. Then secondary bronchi, then tertiary bronchi will be there. Yes or no? Then tertiary bronchi will be there. Yes or no? Yes or no? Yes or no? Yes or no? And then finally, after that tertiary, you will have terminal. What do you have here? The terminal bronchi or terminal bronchioles. What will you have here? There's a primary, secondary, tertiary bronchi. Then you will have terminal bronchioles, right? You will have terminal bronchioles, yes or no? Because before these terminal bronchioles, you have total, right? You people can write it as well. What will you have? You will have total pulmonary bronchioles, and then there will be the terminal bronchioles, right? Then there will be what? Then there will be the terminal bronchioles. Just here you can see initial and then you can say terminal as well. So it is one and the same thing, no problem there, right? So firstly there will be bronchi, then there will be bronchiole. Bronchi, then there will be bronchiole. So after this terminal, you will have respiratory bronchiole. After this terminal, what do you have? You will have respiratory bronchiole. After this respiratory bronchiole, what do we have? We will have the alveolar duct. Alveolar duct. And finally, bache, finally we will have alveoli. So it is the alveoli which is the primary site for exchange of gases. Do you remember that? It is the primary site for what? For exchange of gases. It is the primary site for what? For exchange of gases. Am I right? Am I right? So alveolar duct and directly, acha theek, how can I alveolar duct is there, right? Atria is there, alveolar sac is there. Alveolar sac is there, and then finally there will be alveoli. So it is the primary site for exchange of gases, right? In the lungs, at the level of alveoli, the exchange of gases will occur, right? The exchange of gases will occur. Is that clear? Is that clear? Are you sure? Are you sure? So this is, so here what we need to remember, if you will focus bache, we started from this part. We started from this part, right? Just to make it clear, the difference in glottis and epiglottis, I mentioned it on the different slide. Now again you will revise it with me. So after the larynx, right, we have the larynx, then we have the trachea, that's what I told you, then we have the primary bronchi, right? We have the secondary bronchi, we have, after the secondary bronchi, we have the tertiary bronchi, we have the terminal bronchiole there, but before terminal we have what? Pulmonary bronchioles there, terminal bronchioles there. After that terminal bronchioles, you have respiratory bronchioles, yes or no? Yes or no? You have respiratory bronchioles, yes or no? Then you have what? You have alveolar duct. Yes, you have alveolar duct. Yes. Do you have, what do you have here? After respiratory bronchiole, please revise it with me, right? Alveolar duct is there, yes or no? Atria is there, yes or no? Alveolar duct is there, atria is there, alveolar sac is there, right? And then finally the alveoli is there, which is the primary site. So from this external nostril part to this terminal bronchiole part, right? This complete part, what is it, bachche? It is the conducting zone. Which zone it is? It is the conducting zone. And this part here is considered as the respiratory zone, right? Conducting zone, and this part is the respiratory zone, and this is the zone where the exchange of gases will occur. Where what is going to happen? Where the exchange of gases will occur. Up to this part all clear? This root clear? Right, this root clear. So up to this tertiary bronchi, right, जैसे pulmonary bronchiole, ठीक है, up to this tertiary bronchi, right, from beta, this is again a PYQ that I'm mentioning you, right, so I am mentioning it here that from trachea to pulmonary bronchiole, sorry, they all are having pseudo-stratified ciliated columnar glandular epithelium, PYQ, please mention it. Pseudo-stratified ciliated epithelium, right? Pseudo-stratified ciliated columnar glandular epithelium it is. Is that clear? Is that clear? Everybody here in the class, is that clear? Sure? Sure? Right? So up to this, right? This is the first PYQ. This is the first PYQ. The second PYQ here that we need to remember is that from trachea to initial bronchiole, from trachea to initial bronchiole, what do they have? They have C-shaped cartilaginous rings. They have C-shaped cartilaginous rings. Is that clear? What do they have? They have C-shaped cartilaginous rings. Is that clear? Is that clear? Is that clear? So from terminal bronchiole further, you have the respiratory bronchiole. So this is what you have to remember. So from trachea to initial bronchiole. Okay? Okay? Yes? Right? So what is the role of this conducting zone here? Just I have mentioned that the conducting zone is present here. So what is the role of conducting zone, bache? So it will carry the air to exchange zone. Of course, it will carry the air to exchange zone. Number one, it humidify it. It clears it from foreign molecules. Right? If there is any dust, anything is there, it will clear it. It will humidify it and bring it to the body temperature. Bring it to the body temperature. That's the role here. Any doubt? That's the role here. Any doubt? Yes, that's the role here. Any doubt? Any doubt? Any doubt? Ye dekho. So now these are the words which are given here. Pharynx, we have discussed the three parts. Sound box, cartilaginous box, we have discussed it, right? So trachea is a straight tube extending up to the mid-thoracic cavity, which divides at the level of fifth thoracic vertebrae into right and left. Primary bronchi discussed. Theek bacha. So tertiary bronchi, then there is the bronchiole that will end in a very thin terminal bronchiole. So trachea, primary, secondary, and tertiary, and initial bronchioles, they have incomplete cartilaginous rings. Is that clear? Is that clear? Is that clear? So then beta, you have the alveoli, right? So after that you will see, after that what will you see? After that you will see network of bronchi, bronchioles, alveoli, right? So we have two lungs. Now this is what we have to discuss. So dekha, in a very quick manner we have revised it, right? In a very quick manner we have revised it. Yes or no? Yes or no? Tell me, yes or no? So now look at these. Look at this part. You know that our lungs are present in thoracic cavity. Where our lungs are present in thoracic cavity? Firstly, let's talk about these lungs. Firstly, let's talk about these lungs. So you know that when we talk about the lungs, what exactly do we have? Right and left lung? Yes or no? What exactly do we have? Right and left lung? Yes or no? Yes or no? So you know na, at the time of birth, they are pinkish. They are pinkish, right? They are pinkish at the time of birth, and other than that they are, you know, that soft and spongy. Where are they present? In thoracic cavity. Now here you have the lungs. We have the right lung and the left lung. Now when you talk about the right lung, in the case of right lung, what you have to remember, my dear students, that basically this is shorter and broader, having three lobes. Having three lobes, 1, 2, and 3, right? 1, 2, and 3. Superior, middle, and inferior lobe. What do we have here? We have three lobes here. Superior, middle, and, right? Superior, middle, and inferior lobe. Is that clear? Is that clear? How many lobes do we have here, bache? How many lobes do we have here? 3 lobes. Now, in this case, in the left lung is narrower. Left lung is narrower and longer and narrower it is, right? Longer and narrower it is. Now you know that the space present in between the lungs, we call it as mediastinum. We call it as mediastinum, and the heart is present there. Heart is little bit pushed towards the left side. That is why left lung is having what? Cardiac notch. So if somebody will ask you that cardiac notch is there in the right lung, yes or no? Cardiac notch is there in the right lung, yes or no? Is cardiac notch present in right lung? Is cardiac notch present in right lung? Done? Done? Sure? Sure. So cardiac notch is there in the left lung. So overall in the case of lungs, you know that when we talk about it, it is covered with what? It is covered with the pleural membrane. It is covered with what? It is covered with the pleural membrane. So beta, we have outer, which is also known as parietal pleural membrane. Yeah, pleuron it is. Then we have inner, also known as visceral pleural membrane. Also known as visceral pleural membrane. And in between these two, there is a pleural cavity. There is a pleural cavity having pleural fluid. Any doubt here? Any doubt here? That is how we can revise with the help of example. So you know that it is going to reduce the friction on the lung surface. What is the role of this fluid? What is the role of this fluid? It reduces what? It reduces the friction. It reduces what? It reduces the friction on lung surface and act like a shock absorber. Okay? And act like a shock absorber. Is that clear? Is that clear? Are you sure? Chalo, let's quickly revise it here. Ye dekho. So the branching network done. So see, the lungs are covered with a double layered pleura, right? Having the pleural fluid, which reduces friction on the lung surface and will protect it, act like a shock absorber, right? So the outer pleural membrane is in the close contact with the thoracic lining, and the inner membrane is contact with the lung surface. Is that clear? Is that clear? And here, bache, in the NCERT itself, it is clearly mentioned that the conducting part, right, will carry the atmospheric air to the alveoli, will clear it from the foreign particle, humidify it, will bring it to the body temperature. We have discussed it. We have discussed it. So any doubt here? Any doubt here? So I told you we will complete the NCERT, but yeah, we have to increase our speed. Otherwise, we will not be able to finish the syllabus. Right, we will not be able to finish the syllabus, and we have to, right? You know that we have to. Okay, chaliye. So now, bache, when we talk about the lungs, where are they present? In our body cavities, then cranial cavity, thoracic cavity, abdominal cavity, pelvic cavity. Now, when you talk about the thoracic cavity, trust me, the way is going to be little different from the usual sessions, but we'll complete everything important. Right, we will complete everything important, trust me on that. Okay, so what I just said that we have the lungs are there in the thoracic cavity. Now, bache, when you talk about the thoracic cavity, actually lungs, they are present in an airtight chamber. It is anatomically an airtight chamber. What's that? It is anatomically an airtight chamber. Am I clear here? What is the meaning of that? Your lungs, they are protected with the help of rib cage, the thoracic basket, right? The rib cage, the thoracic basket is there. So what do we have, right? What do we have in that airtight chamber? We have the rib cage, also known as thoracic basket. We have the rib cage, also known as thoracic basket, So when we talk about this, right, what do we have? Beta, dorsal side. This is our dorsal side. Dorsal side is having the vertebral column, right? It is having the vertebral column, and to be more precise, here we will have thoracic vertebrae. Dorsal side will have thoracic vertebrae. Ventral side will have what? Ventral side will have sternum, right? And laterally, what you people will see? Laterally, you will see ribs are there. Laterally, you will see ribs are there. And beta, posteriorly, right? What do you have there? Posteriorly, diaphragm is there. And we know that diaphragm is very important for the respiration. And diaphragm is what? It's the unique feature of mammals. What is this diaphragm? This diaphragm is the unique feature of mammals. Am I clear here? It is the unique feature of mammals. Am I clear here? Yes. Am I clear here? Right? Right, so this is what you have posteriorly in this rib cage. And if you look at the anterior structure, here you will talk about this beauty bone, this collar bone, the clavicle, right? That anteriorly this clavicle is there. So this is an airtight chamber. What kind of chamber it is? It is an airtight chamber. This is something that you need to keep in your mind. Okay, now there is one very important point. So as I said, our lungs are there in the thoracic cavity. This is, let's suppose, right, these are the lungs, they are present in the thoracic cavity here. Right? Now, bache, if you talk about these lungs, they cannot control their volume, right? They cannot cause changes in their volume. They are not. They are not. In our body, my dear students, I'm sorry, in our body, right? Keep it in your mind, people. In our body, this setup is in such a way. Just I just said that lungs, they are present in thoracic cavity. I told you they are in anatomical airtight chamber. So here I am saying that these lungs are placed in such a way. These lungs are placed in such a way. These lungs are doing what? These lungs are placed in such a way that if there is, means this arrangement and setup is in such a way that if there is any change, right, if there is any change in volume of thoracic cavity, that will reflect in the pulmonary cavity. So our lungs are present in thoracic cage in such a way, if the volume of thoracic cage will increase, the pulmonary volume will also increase, right? If the volume of thoracic cage will decrease, the volume of pulmonary cage will also decrease. So this is something very important. Do mark it, my dear students. So here you guys can see this, that the lungs are situated in the thoracic chamber, anatomically airtight chamber, right, bache? So anatomical setup of lungs. This anatomical setup of lung and thorax is such a way that if there is any change in the volume of thoracic cavity, that will be reflected in the lung or the pulmonary cavity. Any doubt here? Any doubt here? This is very important for the respiration. So if there is any change in the overall thoracic volume, the volume of lungs will also change. The volume within the lungs will also change. So let's suppose, answer this question. If the volume of thoracic cavity increases, what will happen to the volume of the pulmonary cavity? What will happen to the volume of the pulmonary cavity? If the thoracic cavity's volume increases, what will happen? Yes? Of course, it will increase because this is how the setup is. Okay, so overall in this chapter, these are the 5 steps that we have to discuss, right? Basically, it is the respiration that we discuss here, right? Basically, it is what that we discuss here, people. It is the respiration that we discuss here. It is the respiration that we discuss here, right? So basically, respiration, you know that. What kind of change it is? Catabolic. Why? Because here we talk about the breaking of carbon-carbon bond, and that too we discuss in class 11. But for this respiration, for this breakdown of glucose, for taking out the energy, the very first step is the breathing or the pulmonary ventilation. What is the very first step that we have here? It is the breathing or the pulmonary ventilation, which means simply the exchange of gases, right? So atmospheric air will come in and the CO2 rich alveolar air will go out. Atmospheric air will come in and the carbon dioxide rich alveolar air will go out. This is the first step that we have here. This is the breathing, my dear people, and how this breathing is possible with the help of diffusion, right? So gases will move from their higher concentration to lower concentration. That is the main funda here. So once the gases, they reach the lungs, the alveoli, their diffusion will occur across the alveolar membrane. Then blood has to transport it, and then finally the gases will diffuse. The oxygen will pass from blood to tissue and CO2 from tissue to blood, and finally that oxygen will be utilized by the cells for the catabolic reaction, for the breakdown of glucose, and the energy will be produced. So this is the cellular respiration, that glycolysis part, that Krebs cycle part that we discuss in the respiration in plant chapter. So this part is clear. This part is clear. Are you sure? Now see, this is that complete anatomical setup. That is how our lungs are present here, right? That is how our lungs, they are present here within this thoracic cavity. Take care. Thank you. Within this thoracic cavity, that is how our lungs are present. Okay. Now when you look at this setup, see this, bache. See this. These are the ribs. These are the ribs. Yes or no? These are the ribs. Yes or no? Tell me quickly. Tell me quickly, these are the ribs that we have here? Yes or no? These are the ribs that we have here? Yes or no? Yes or no? Definitely. Definitely. So now when you look at these ribs now, right, firstly understand this arrangement. Now when you look at these ribs, in between these ribs, right, in between these ribs here, this is, suppose that's the sternum. Ribs are attached like this. Take in between these ribs, here we have the muscles. Firstly, understand these muscles. They are known as intercostal muscles. Which muscles are they? They are intercostal muscles. Which muscles are they? Intercostal muscles. Then you have the diaphragm here also, and this diaphragm is also having these diaphragm muscles, or peripheral muscles. So remember, this diaphragm is going to, it's a muscular structure that is going to separate thoracic cavity from abdominal cavity. So the muscles clear, which muscles are present, is that clear? So these are the ribs. In between these ribs, what do we have? We have, right, what do we have? Intercostal muscle. Now, bache, in between these ribs, let's suppose if these are two ribs, if the muscles, if they are present towards the outer side, towards the external side, they are the external intercostal muscles. They are the external intercostal muscles. Okay, what are they? They are the external intercostal muscles if they are present on the external side. Let's suppose. Now, if these muscles, if they are present on the internal side, you will call them as internal intercostal muscle. So intercostal means, inter means in between. They are costal, hyaline cartilage, costal cartilage, ribs. That's how you have to remember. So these are the ribs that we have. In between the ribs, we have the muscles. We are calling them as intercostal muscles. We are calling them as intercostal muscles. If they are present towards the exterior side, external intercostal muscle. If they are present on the internal side, they are the internal intercostal muscles. Any doubt here? Any doubt here? Any doubt here? Any doubt here? External, internal part clear? External, internal part clear? Right? So these muscles, why do we need to remember it? Because now we have to start the next topic, the mechanism of breathing. And for that, these things needs to be very clear. So here you people can see, very clearly you people can see, what do we have? External intercostal muscle, internal intercostal, innermost and leave innermost, internal intercostal muscle. Is that clear? So outermost, this one is external, that they are showing here, and this one, let's suppose this is the internal intercostal muscle. So the muscles clear. So the muscles clear. So the muscles clear, yes or no? Muscles clear. The position of muscles clear. Now, external, internal muscles clear. Are you sure? Are you sure? Are you sure? Okay. So, now, bache, when we are talking about this, so muscle position clear. Chal. Let's come to this concept now. Now, what is the word that I have used? It is the breathing. What is going to happen in this breathing? Right? There will be beta inspiration. There will be expiration. Inspiration is also known as inhalation, right? It is also known as inhalation. Second word is what? Second word is expiration. It is also known as exhalation. These are the two things that are going to happen. How these two things will occur? Of course, diffusion will be there. What is the meaning of diffusion now, students? What is the meaning of diffusion now? You know that diffusion, it is the movement. Right here, what is it? It is the movement of gases. Movement of gases from their higher concentration to their lower concentration. So basically passive movement it is, right? Along the concentration gradient movement it is. From high to low this movement is. From high to low this movement is, right? How it is? It is from high to low. It is from high to low. Is that clear? Is that clear? So movement of gases from their higher concentration to their lower concentration. From their higher concentration to their lower concentration. This is what the diffusion is. Take the now inspiration. We have to inhale. What we need? We need oxygen. What we have to exhale? CO2. Why? Because this is our metabolic waste. है ना? This carbon dioxide is actually our metabolic waste. है ना? This is what we have to do. Now, when you are talking about the air, beta air, it's not like specifically you are only taking the oxygen. Nahin. Right? We are taking the air in, and air is the mixture of gases. Air is the mixture of gases. It is not the oxygen only that we are inhaling, right? It is not the oxygen only that we are inhaling, right, bache? What are we actually inhaling? What are we actually taking in? Air, right? The complete air, which is a mixture of gases, which is having oxygen, carbon dioxide, nitrogen, everything is there, right? Now, here in the mixture of gases, each gas or each component is having its own pressure. Having its own pressure, and that will contribute, all the gases and their pressure, right? Everything will contribute, and then finally this air will have a pressure. So each gas is having its own pressure, and for that we use the word partial pressure, because it is not the complete pressure of the air, right? Air is having many components, and that components are also having the pressure. We use the word partial pressure for that, right, bache? Right, bache? Right, bache? Right, bache? So what do we need? What do we need? The partial pressure, right? Here, what are we talking about? We are talking about the partial pressure. So we discuss about the partial pressure of oxygen and partial pressure of CO2 here. Now, for this mechanism of breathing, what we need to focus? We need to focus on the Boyle's law. What do we need to focus? We need to focus on the Boyle's law. Boyle's law. What we need to know? We need to know about the Boyle's law. What this Boyle's law is saying, bache? It is saying if volume is increasing, pressure is decreasing. Means volume and pressure, they are inversely proportional to each other. What are they doing? They are inversely proportional to each other. What are they, bache? They are inversely proportional to each other. So if volume is increasing, pressure is decreasing. If pressure will increase, volume will decrease. That's what you have to keep in your mind. And this is what we need to know for the breathing. Right? For the breathing, this is what we need to know. Take. Now, you know that thoracic volume and the pulmonary volume, it is linked to each other. Now, if I have to inhale, right? If I have to inhale, if I want the air to enter, or particularly oxygen to enter within the lungs, if I want this, right? What I need to do, bache? I need to increase the volume and I need to decrease the pressure. Right? If I want, right, to inhale, if I want the air to come in, so obviously air will move from its higher concentration to lower, yeah, higher pressure to lower pressure. So within the lungs, right, within the lungs, what do we need? Within lungs, the pressure should be low and volume should be high. So basically we have to here, what we have to do, we have to increase the volume of thoracic cavity. That will increase the volume of pulmonary cavity. Yes or no? Yes or no? Yes or no? What we have to do here, we have to increase the volume of thoracic cavity. That will increase the volume of pulmonary cavity. This is what we need. Yes or no? Yes or no? Yes or no? So we need to know something here. Here some muscles are going to play the role and we call them as inspiratory muscles. Right? We call them as inspiratory muscles that are going to play the role here. Who's going to play the role here? Inspiratory muscles. Which muscles are inspiratory muscles? Beta, your external intercostal muscles and your diaphragm muscles. They are actually your inspiratory muscles. And why? Because they contract during inspiration. Why? Because these muscles, they contract during inspiration. So we call them as inspiratory muscle. So which muscles are inspiratory? Your external intercostal muscle and your diaphragm muscle. Now firstly, when you talk about your external intercostal muscles, when you talk about your external intercostal muscles, but when these muscles, they contract, right, they are present in between the ribs, so what is going to happen here? Your rib cage, it will go upward and right outward and upward rather. Just you remember this, there is a dance step that we have to remember here. This and then this, right? This and then this. But firstly, it's like that. Then it has to be this. So what is the meaning of that? So firstly, when you're talking about the external intercostal muscles, what is going to happen when these muscles, they contract, right? The rib cage, it will go outward and upward, right? So imagine your rib cage, imagine your rib cage was like this. The external intercostal muscles contracted, so it will go outward and upward. So the volume, right? So it will result in increase in the volume towards the dorsal ventral axis. There will be increase in the, right, increase in the volume towards dorso-ventral axis. Yeah, it will increase the volume of dorsal ventral side or dorsal ventral axis. Any doubt here? Yes. Any doubt here? Yes or no? Any doubt here? Yes or no? Basically, we have to increase the pulmonary volume here now. We have to increase the pulmonary volume here now. So what will happen? There will be an increase in dorso-ventral axis. There will be an increase in the volume towards the dorsal ventral axis. Hanji. So there will be the increase in the volume of thoracic chamber in the dorsal ventral axis. In dorsoventral axis. Now, your diaphragm muscles. What will happen to your diaphragm muscles? Bache, diaphragm muscles, when they contract, what is going to happen? See, firstly, right, diaphragm was what? Diaphragm was what? It was dome shaped. It was dome shaped, right? Then, after contraction, it will become flat. It will become flat. Just firstly, this is the condition. Now, when it will become flat, just earlier it was like this. Earlier it was like this. Now it will become flat. So if it will become flat, it will increase the volume, right? If it will become flat, it will increase the volume. There will be an increase in volume of thoracic chamber. There will be an increase in volume of thoracic chamber, right? Thoracic chamber in anterior-posterior axis. How? In anterior-posterior axis. So can I say that? Yes, everybody here. So can I say that due to the contraction of inspiratory muscle, the thoracic volume increases and the pressure decreases. So intrapulmonary pressure here, what is it? It is negative. It is less, right? Within the lungs, the pressure is less. It is negative. Yes or no? Yes or no? Yes or no? है ना? Of course, ma'am, this is going to happen. So there will be an increase. There will be an increase in the thoracic volume and the pressure will decrease. So air will definitely come in. That is the point here, right? That is the point here. So remember, when you talk about the inspiration, you need to focus on the volume and pressure. You need to focus that which muscles are going to contract, and you need to remember that it is an active process, right? It is an active process that will take place for 0.2 seconds. Is that clear? It is an active process that will take place for 0.2 seconds. Is that clear? Are you sure? Are you sure? Sorry, not 0.2 seconds, 2 seconds, my bad. 2 seconds, right? This inspiration is for 2 seconds. Is that clear? So it is an active process. So now, what's going to happen in expiration, beta? When there is expiration, it is passive process. It will occur for 3 seconds. So what's going to happen in this expiration, right? Your external intercostal muscles, they relax, right? So because of that, your rib cage, it will go downward and inward. Your diaphragm muscles, they also relax. So diaphragm will become dome shaped here. So that is important. That is important. So, and when there is a forceful expiration, when the expiration is what? When the expiration is forceful, my dear people, so it is an active process. Remember this. Then your internal intercostal muscles, they will also contract here, and even your, right, some additional muscles are also going to help of abdominal cavity. The additional muscles of abdominal cavity will also help. Okay. Okay. So with the help of additional muscle in abdomen, they will also help in that. Okay, they increase the strength of overall inspiration and expiration. Fine? Fine? Sure? Doubt? Doubt? Any doubt here? Haan beta, energy is used. That's why it is active. That's why it is active. ठीक. So ये देखो mechanism of breathing, right? These points that I have discussed, they are given here. So relaxation of the diaphragm and intercostal muscle return the diaphragm and sternum to their normal position. That's what we have discussed. Take, okay, right? So for expiration, you know that the conditions has to be reversed. So volume needs to be down, pressure needs to be high so that the CO2 will go out. Okay, okay. So here it is clearly written, bache, the ability, we have the ability to increase the strength of inspiration and expiration with the help of additional muscle in the abdomen. So on an average, bache, on an average, human breathe 12 to 16 times per minute. So volume of air involved in breathing movement can be estimated with the help of spirometer. Okay? It can be checked with the help of spirometer. But remember, spirometer cannot check residual volume. It cannot check RV, residual volume. ठीक? It cannot check RV, residual volume. Understood? Understood? Chaliye. So this is the diagram. Diagram is important. So diaphragm will contract. It is coming down means it will become flat. Rib cage. Dekho, ribs and sternum, they are raised. So volume of thorax will increase here. Right? Volume of thorax will increase here. Is that clear? So diaphragm will relax. It will upward. Ribs and sternum will come back to their original position. Volume decreases. Air will expel from the lungs. Any doubt? Any doubt? Better diaphragm is going to separate the thoracic cavity and the abdominal. It is going to separate them, right? So it will relax. The muscles will relax during expiration. They will contract during inspiration. That's it. Anything else? Anything else? Okay. Now pulmonary cavity. Dekho, we have the respiratory volumes and we have the respiratory capacities. Respiratory volumes are there, respiratory capacities are there. So spirometer will help to check these volumes, but as I said, it will not be able to check the residual volume. It will not be able to check the residual volume. Is that clear? What will it not be able to check? The residual volume. So here, firstly, the tidal volume is the thing which is very less, that is 500 ml per breath, right? So it is the volume of air that can be inspired or expired during a normal respiration, normally. So we, on an average, we breathe 12 to 16 times, right, per minute. So 12 into 500 or 16 into 500. So it will be 6,000 to 8,000 ml per minute, right? Per minute, it is like that. So you can say 6 liter to 8 liter per minute. Now, inspiratory reserve volume, right, related to inspiration. Additional volume of air a person can inspire, can inhale. Additional volume a person can inspire or inhale. Theek? So, it is due to the forcible inspiration. So, here what do we have? 2500. Here what do we have? 2500 to 3000 ml. What do we have here? 2500 to 3000 ml. Then expiratory reserve volume, that forcefully you have to exhale. That forcefully you have to exhale. Forcefully you have to exhale. So that will be 1000 to 1100. That will be 1000 to 1100 ml. That will be 1000 to 1100 ml. Is that clear? Then there will be the residual volume. Your lungs will not be empty. You cannot say that we are going to inhale forcibly and all the air will go out. No, some amount of air will still be there in your lungs. Just it's like, right? Just it is like somebody, your friend is asking you for the money, that I need money, it's too urgent, right? It's too urgent, I really need it, I need it badly. So you are like, okay, fine, I just have 1000 or 1100 rupees, you can take it. Now the point is, you are also smart enough. Now you know that what if you have to go out, what if you have to buy something, you trust your friend, they are not going to return your money soon. So you will keep something with yourself. You will keep something with yourself, residual, remaining, your secret money. That is the residual volume, 1100 ml to 1200 ml. Okay, and beta, when you are talking about this tidal volume, na, out of this 500 ml, just keep it in your mind. Just keep it in your mind. 150 ml is not at all available for the exchange. It is just 350 ml that is available. Why? Because this 150 ml is making the dead space, and it is anatomical dead space. I'm not saying physiological. You have to be specific here. It is anatomical dead space volume, not available for the exchange. So this 350 ml is available for the exchange, right? In my detailed lectures, I have explained you the reasons. Okay, I have explained you, I have given you the reasons here. So please do remember this. Now, these capacities, beta, देखो, when we add the respiratory volumes, we will get the capacities. So if we cannot measure residual volume, so of course the capacity where we need residual volume, we will not be able to measure it as well. So when it comes to the capacity, as I said, it is the combination of addition of the volumes. So you have to remember few things. जैसे the first capacity that we have to read is inspiratory capacity, IC. Then it will be बच्चे expiratory capacity, EC. Then it is vital capacity, VC. Then बच्चे it is total lung capacity, TLC. And then you have functional residual capacity. So tell me everybody, do you remember these names? Yes or no? Do you remember these names? Yes or no? How many capacities are we going to study? 1, 2, 3, 4, 5. How many volumes we have studied? Tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume. 4 volumes, 5 capacity. 4 volume, 5 capacity. Everybody please repeat what we have studied. 4 volumes, 5 capacities. 4 volumes, 5 capacities. This is what we have read, right? This is what we have read. 4 volumes, 5 capacities. 4 volume, 5 capacities. This is what we have read. Now when it is the inspiratory capacity, देखो, tidal volume is the normal, मतलब without any force, forcibly, right? Without any force, without any, externally you are not putting the efforts. Like I'm talking to you, still I'm inhaling and exhaling. That is making my tidal volume. Okay, that is making the tidal volume. So we have to keep the tidal volume in all the cases except the residual capacities. Except the functional residual capacity, you have to keep the tidal volume because it is normally without any external pressure. We are just inhaling, right, the air in. Now when it is the inspiratory capacity, it will be inspiratory reserve volume. Expiratory capacity, expiratory reserve volume. Vital capacity mein you have to add 2. You have to add 2. And when it is total, in the total you have to consider everything, my dear people. In the total you have to consider everything. You have to even consider the residual volume. So when it is the functional residual capacity, it is just residual volume and expiratory reserve volume. Firstly, remember these formulas. Give it a try, right? Yes, quickly revise it. Do it quickly. Yes, done? Sure? Is it done? Do it quickly, please. Yes, please. Yes, please. Done? So this formula is on your fingertips. Is it on your fingertips? Is it on your fingertips? Pakka? Sure? Are you sure? Okay. So this is what you have to remember. So now let's suppose if the inspiratory capacity you have to calculate 500 plus let's suppose 2500. That is how you can count it. That is how you can check it. Okay. So inspiratory capacity is what? total volume a person can inspire but after a normal expiration. Matlab you have to check the capacity of the lungs, you have to check the ability of the lungs, isn't it? Isn't it? You have to check the capacity of the lungs, you have to check the ability of the lungs here, my dear people. So after a normal expiration, right, what is the capacity of your lungs to inhale, to inspire? That's what you have to remember. Okay, that is what you have to remember. Okay. Okay, so this includes tidal volume and inspiratory reserve volume, the very easy formula here. Very easy formula here. When it is expiratory capacity, so again, you have to check the capacity of your expiration, capacity of your exhalation, right? The amount of air that you can exhale, but yes, after normal inspiration. Means normally you are inhaling the air. So what is the amount that you can exhale as well, right? Total volume a person can exhale after a normal inspiration now, right? So then what is functional residual capacity? Remaining thing is there, right? So volume of air that will remain in lungs after normal expiration. Normally you are exhaling, what is left in the lungs? So definitions are also very easy. I don't know why you consider this topic as the difficult one. So vital capacity, so it is simple, TV, IRV, ERV. Okay, and total lung capacity, it's also simple, TV, IRV, ERV plus RV. Is that clear? So the maximum volume of air a person can breathe in, the maximum volume that you can take in after forced expiration, vital capacity. Forcefully you are going to exhale, then what is the amount of air that you can take in? So just here, remember one thing, focus on understanding these definitions. That's it. That's it. Nothing else. Nothing else. Right? Just focus on that part. Then total lung capacity, total volume of air accommodated in lungs at the end of forced inspiration. That's it. Now, exchange of gases, simply diffusion is going to occur here. Right? Diffusion is going to occur here. So when there is an exchange of gases in between alveoli and the lungs, alveoli and the blood, right, blood, this is internal respiration. When there is an exchange of gases in between the blood and tissue, this is beta external respiration. It is external, this is external respiration. This one is internal respiration. And at the level of cell, when cell is going to take the oxygen and going to break the glucose, cellular respiration. Now, this table, very, very important. Right? It is very, very important here. Now, I would like to ask you one question. Dekho, this is the partial pressure of oxygen, right? It is in mmHg. Yeah, dekho, it is in mmHg. So, atmospheric air, the partial pressure is 159 mmHg, but alveoli will have this. So, what about the balance here? What about the balance here? This is what you have to answer in the comment section after the class. Right? Why is it so? Right? If you are a serious NEET aspirant, definitely you are going to answer this question. Right? If you are a serious NEET aspirant, then definitely you are going to give the answer of this question to me, that madam, this is the reason. Right? That is why it is happening. Right? So that's what you have to tell me. Okay? That's what you have to tell me. Take, so atmospheric air, alveoli, right? Look at the difference. Now in the deoxygenated blood, remember, it's not like that if the blood is oxygenated, only oxygen will be there, CO2 will not be there, CO2 will also be there. If the blood is deoxygenated, it's not like that only carbon dioxide will be there, oxygen will not be there, oxygen will also be there, but its partial pressure will be less. So the blood is deoxygenated, the more partial pressure of CO2, less will be of oxygen. When the blood is oxygenated, more partial pressure of oxygen, less will be of CO2, and this is what we have in the tissue. So yes, that's something you need to remember. Okay, that's something you need to remember. Is that clear? Is that clear? Sure. Are you sure? Now you know that actually where will be the exchange occur? Exchange will be there in the alveoli. Where that exchange of gases will occur? That exchange of gases will be there in the alveoli. Right? Alveoli. Just say this is what we have here. Now, bache, when you talk about the alveoli, right? Alveoli. What is the word? Alveoli. So you know that they are the structural and functional unit of lungs. Yes or no? I believe you all know that they are structural and functional unit of lungs. What they are, bache? They are structural and functional unit of lungs. Is that clear? Is that clear? So they have two type of cells which are known as pneumocytes. What are they? Pneumocytes. So pneumocyte 1 is there, pneumocyte 2 is there. These are the two types. Other type of cells are also there. I'm just telling you this. So this pneumotype 1, they are bit smaller. This one is larger. So this larger one actually, it releases lecithin. What is lecithin? I hope you remember in the biomolecule also this is given. So when we talk about the lecithin, my dear people, when we talk about the lecithin, what is it? When we talk about the lecithin, you know that it is the most abundant phospholipid. It is the most abundant phospholipid. What is this lecithin? It is the most abundant phospholipid. Are you getting my point? Are you getting my point? Most abundant phospholipid. It is acting as a surfactant, right? It is act as a surfactant, right? It is acting like a surfactant. Who? Your lecithin. So it is the phospholipid. It is acting most abundant phospholipid. It is acting like a surfactant, bache. So it will reduce the surface tension of alveoli. What will it do? Reduces surface tension of alveoli, will keep them open so that gaseous exchange can occur, right? So that gaseous exchange can occur. Is that clear? So that gaseous exchange can occur. Is that clear? Sure? Are you sure? Are you sure? And pneumocyte 1, they actually will help in gaseous exchange. So each lung will have, yes, do you know that each lung will have, your total number of alveoli in the lung will be 300 million there. 300 million there. Now here, the gaseous exchange will occur. The simple diffusion I'm not going to explain. So, bache, this is the alveoli, right? I told you the example also, na. So here you have simple squamous epithelium. Wherever there is diffusion, the exchange, this is the epithelium that you are going to get. And when we talk about the epithelial tissue, my dear students, when we talk about the epithelial tissue, it lies on what? It lies on the basement membrane. So this basement membrane, right? It is having basal lamina and fibrous lamina. So the connective tissue beneath the epithelial tissue, right, will make the part of basement membrane, and some part is made by this epithelial tissue as well. So this basement membrane is acellular, always remember that. So structural organization also we are covering. Now this is the blood capillary, right? So when you talk about this blood capillary, very thin walled it is, bache, it is having endothelium. What does it contain? It contains endothelium, okay? It contains endothelium, and this endothelium is also having the basement. Matlab, basically here the gaseous exchange will occur. So you will have simple squamous epithelium of alveoli, you will have endothelium of this blood capillary, you will have the basement membrane present in between, basement membrane of both present in between. So this is the layer through which the exchange will occur, and it should be very thin, bache. Its thickness should be less than 1 mm. This is the respiratory membrane. Okay, this is the respiratory membrane. So its thickness should be less than 1 mm, right? Here you people can see. Here you people can see. So squamous epithelium of alveolar cell. So when I'm saying simple squamous epithelium means it is uniserrated, means it is single layered. Take. Okay, so blood capillaries, RBCs, endothelium of blood capillary, basement membrane is there. This is the alveolar cavity. Okay, okay. So here you guys can see the layers as well. ये देखो, done. So their thickness, it should be less than a millimeter. Okay, okay. So this is very important here. This is very important here. Okay, so for the diffusion, you know that firstly the gradient has to be maintained, the concentration difference should be there, and as the solubility right? So this is basically the diffusion membrane through which the gaseous exchange will occur, my dear students. And remember, the solubility of CO2 is 20 to 25 times higher than the oxygen. So CO2 can diffuse very quickly right through the diffusion membrane per unit difference. Okay, done? Chalo. So transport of gases mein, we will start with the oxygen. Right, we will start with the oxygen. So I will take another 30 minutes. We'll finish this chapter. We'll move to the next one. So next one will be neural control or chemical control. Which chapter you want me to start? Next, neural or chemical? Madam, now we don't want anything. We just want to sleep, right? We are done and dusted. Arey nahi, nahi, no. Ma'am, is it possible to complete all the chapters? Dekho bachche, and when we have to complete all the chapters, we will, but then we have to focus on the most important things only, right? Here now also we were more concerned about the most important things, but still some basics here I'm considering right now. Neural chemical, yeah, equal votes are there. Neural chemical. Chalo, I'll start with the neural. Okay, more votes are there for the neural coordination. More votes are there for the neural coordination. Sleepless nights. Same. Now, transport of gases. So when you are talking about the transport of gases, right, oxygen and CO2. So 97% of the oxygen will be transported with the help of hemoglobin. So now let's discuss that. Take, you know that RBC, red blood cell. Remember, in body fluid and circulation also we will talk about it. Red blood capsule it is, right, biconcave, enucleated, biconcave and enucleated at maturity. Why this shape, biconcave shape, is there? To increase the surface area. So this RBC, it contains hemoglobin. What do we have in the RBC? Hemoglobin. What is this hemoglobin? It is the respiratory pigment. And beta, this is the respiratory pigment due to which the blood is red in color. What? Hemoglobin. So hemoglobin is the respiratory pigment due to which the blood is red in color. So what exactly this hemoglobin is? It is the quaternary protein. Right? So here, bache, in the adult hemoglobin, because adult and fetal hemoglobin is different, their affinity is different. Here you have 2 alpha chains and 2 beta chains are there. 2 alpha chains and 2 beta chains are there. That is what we have. When you talk about the hemoglobin, that is what we have. 2 alpha chains and 2 beta chains. What do we have, people, here? Yes, what do we have, people, here? We'll have 2 alpha chains and 2 beta chains. Am I right? Yes, please. Am I right? 2 alpha, 2 beta. So total, it's a quaternary protein having 4 polypeptide chain, made up of 4 polypeptide chain, 2 alpha, 2 beta. So now in the hemoglobin, globin is the protein part, heme is that pigment part. No, just say, let's suppose this is hemoglobin. Let's suppose this is the heme and it is having your iron in 2 positive state. That is what you have to remember. This is important. So heme is that pigment part, globin is the protein part, heme is that pigment part, and this heme is having what, bache? It is having 4 units. Each unit is having iron, and that too in +2 oxidation state, right? That too in +2 oxidation state, right? This is the oxidation state of iron here. It is +2 oxidation state. This is important. Now in this hemoglobin, right, how many iron do we have? 1, 2, 3, 4. Each can bind with the oxygen. So basically, what's the story here? This is the story here. This is what we have to see here. This is your oxyhemoglobin. So actually what's going to happen? Just now I told you that 97% of the oxygen will be carried in the form of oxyhemoglobin. So what is this hemoglobin? This hemoglobin is the respiratory pigment which it helps in transport of oxygen, right? Like myoglobin, it stores oxygen, but this one, it helps in transport of oxygen. So each hemoglobin having 4 polypeptide chain, having this 4 Fe2 positive, so one can bind with 1 oxygen. So here you have 4. So that is going to be the thing. That is how the oxyhemoglobin will form. That is how the oxyhemoglobin will form. That is how this reaction is. And when this oxyhemoglobin will form, of course, in alveoli, Of course, in alveoli. I'll tell you why. So basically, we have to make the oxyhemoglobin, and when will this hemoglobin bind with the oxygen? There are certain conditions for that. First of all, you have to remember this is oxygenation that is taking place here, right? Oxygenation means addition of oxygen. We have to add the oxygen to the hemoglobin. It is not oxidation. Oxidation is something different. It is what? It is the oxygenation. It is what? It is the oxygenation. Is that clear? Is that clear? Are you sure? It is the oxygenation. What is it, bache? It is the oxygenation, addition of oxygen. So this hemoglobin has to bind with oxygen, and it is not like that this process will take place randomly. No, there are certain conditions which are required for that, and the most important condition is related to the partial pressure, right? So for the oxygenation, that is for the addition of oxygen, there are certain conditions where this oxygenation will occur, of course, in alveoli. What is the reason here? Because bache, the partial pressure of here, the partial pressure plays a very important role. Partial pressure of oxygen needs to be very high. That is required for the oxygenation. So if partial pressure of oxygen is high, the partial pressure of CO2 will be low, of course. So now, along with that, there has to be low H positive ion concentration. If H positive is low, matlab pH is high. And what about the temperature? Temperature is also low here. Temperature is also low here. These are the conditions for the oxygenation, right? If we have to add the oxygen to hemoglobin, we need these conditions. The partial pressure of the oxygen needs to be high. So of course the partial pressure of the CO2 will be low H positive concentration, low temperature will be there, right? And bache, the role of hemoglobin is to transport oxygen. It cannot keep it. Now you tell me, if you have ordered something, right, the delivery person has to give it to you. If you guys have ordered something, right, if you people have ordered something, what your delivery person is going to do? Your delivery person is going to give that to you. Your delivery person has to pick it up, then that person is going to give it to you. That person cannot keep it, right? And of course, before delivery, right, it is the duty of that delivery person to take care of that thing, right? To take care of that thing and to give it to you. Same is the case here. Hemoglobin is a delivery person. Hemoglobin has to pick the oxygen, right? And that from where it will pick, where the high partial pressure of oxygen is there. Where is it? In the alveoli. Now it has to give that oxygen to the tissue, right? Because cells there, they need it. They need to break the glucose. So they need it there. Okay, they need it there, right? Right? They need it there. So what's going to happen? So hemoglobin is like, okay, fine, I'll pick up the oxygen, right? I will become oxyhemoglobin. But remember, this pickup and this oxygenation, it is temporary, right? It has to be temporary. It is reversible, right? It is temporary. It is reversible. Why? Why? Because of course it cannot keep the oxygen with it only. It has to give it to the tissue. So this binding has to be temporary. This binding has to be reversible. In the alveoli, we need to make the oxyhemoglobin, and in the tissue, we have to break the oxyhemoglobin. That is why it is like that, right? Right? So this part clear? This part clear? These are the conditions for the oxygenation, right? And where will you get these conditions? In the lungs, of course. Now, the next point that you have to remember, right? Do you know our 100 ml of blood, in our 100 ml of blood, how much HB is there? Hemoglobin is there. Do you know that? Matlab normally if I have to consider, it will be 15 gram of hemoglobin and 1 gram of hemoglobin, right? And do you know 1 gram of hemoglobin can pick 1.34 ml of oxygen? Are you getting my point? 100 ml of blood is having 15 gram of hemoglobin. So when we get our blood test done, they say 10 gram HB is there, 12 gram is there. So they are talking about the 100 ml of blood. It is not in our entire body, right? It is not in our all the blood that we have. We have 5 to 6 liters of blood, 4 to 5 liters in women. Done? So it is the 100 ml. It is the 100 ml. So it is normally 15 gram of hemoglobin, right? 1 gram of hemoglobin will carry 1.34 ml of oxygen. Are you getting my point? So when it is the 15 gram of hemoglobin, so of course 15 into 1.34 ml, 15 into 1.34 ml we have to do, right? Right, and it will be approximately, this math says approximately 20 ml of oxygen. Matlab your 100 ml of, so 15 gram of hemoglobin can carry 20 ml of oxygen. So can I say that your 100 ml of blood will have approximately 20 ml of oxygen? Can I say so? It will have approximately 20 ml of oxygen. Can I say so? Right? Can I say so? Yes or no? Can we say so? Yes or no? Yes? है ना? Right? So, bache, when we check it, right? Just let's suppose 100 ml of oxygenated blood is having 20 ml of oxygen. So, in its each cycle, right? In its each conducting cycle, this blood will give 25% of this oxygen. Matlab, when you will check the deoxygenated blood, instead of 20, you will see that 15 ml of oxygen it contains, right? Matlab, 100 ml of deoxygenated blood, right, will have this 15 ml of oxygen. So of course, the difference is of 5 ml. So you can say that the 100 ml of blood can transport 5 ml of oxygen. The 100 ml of blood can transport 5 ml of oxygen. And when we do some strenuous exercises, then this amount can reach up to, this 5 ml amount can reach up to 15 ml, because then our body, right, bache, then our body, it needs more oxygen. This part is clear? This part is clear, right? Yes. So here, this oxygen is not going to oxidize the hemoglobin. It is simply the oxygenation, the addition of oxygen, right? The addition of oxygen here, and that's what you have to revise. Okay, so oxygen is not going to oxidize the hemoglobin. The oxygen will simply be added here. Okay, okay, done, done. Okay, so of course, now this oxyhemoglobin, this blood will pass to the tissue. You know that in the tissue, the conditions are reversed. So oxyhemoglobin will break, it will give the oxygen to the, right, it will give the oxygen to the tissue. So now what we have to understand here, we need to understand something very important, that is known as oxygen hemoglobin dissociation curve. What we need to understand here, we need to understand something very important here, that is oxygen hemoglobin dissociation curve, या oxyhemoglobin dissociation curve, या oxygen dissociation curve it is. ठीक? Right, बच्चे? Right, बच्चे? So, it is a sigmoid curve that is obtained when the percentage saturation of hemoglobin with oxygen is plotted, right? When it is plotted, how is it plotted, beta? This is plotted against the partial pressure of oxygen, right? Right. So graph is plotted in between the oxygen concentration or partial pressure of oxygen and the percentage saturation of hemoglobin. ठीक? And the percentage saturation of hemoglobin. ठीक? And do you know each hemoglobin contains approximately 270 million hemoglobins. Each RBC contain approximately 270 million hemoglobin molecule. Imagine, just imagine. Now, what you have to remember here, let me tell you, each RBC is having approximately 270. Dekho, percentage saturation. Firstly, let me explain this, then I will show you the oxygen hemoglobin dissociation curve to you. Beta, it is simple. Let's suppose you have one hemoglobin molecule can bind with 4 oxygen molecule. Let's suppose you have 50 hemoglobin molecule, right? 50 into 4, 200 it will be, right? Now, out of that 50, if 25 hemoglobin, they are all attached with the oxygen. If out of these, yes, bache, if out of these 50, if 25 hemoglobin molecules, if they are attached to the oxygen, so what will I say? I'll say that 50% of hemoglobin is saturated. So it means 50% of the hemoglobin is attached to the oxygen. So this is the percentage saturation thing that we have to discuss here in this particular curve. Just let's suppose you have 100 hemoglobin molecule. Out of that 100, if I'm saying that 50 hemoglobin molecules are fully saturated, yeah, they are attached to the oxygen, right? They are attached to the oxygen, they are fully saturated. They are attached to the oxygen, they are fully saturated. 50. Right? What am I talking about? I'm talking about the 50 hemoglobin molecule. Matlab 50% of the hemoglobin molecule is saturated. So when I'm saying saturated, matlab oxyhemoglobin has formed. Right? So this is the curve that we have. So the very first thing that you need to know is, that the x-axis here will show the partial pressure of oxygen. ठीक है बेटा, the x-axis here will show the partial pressure of oxygen. And what about the y-axis, people? Y-axis will show the percentage saturation of hemoglobin. Y-axis will show the percentage saturation of hemoglobin, and the x-axis will show us what? It will show us the oxygen, partial pressure of oxygen. Now here in this oxygen hemoglobin dissociation curve, there are two things that we have to remember, right? Left shift and right shift. To remember that, we have a trick. The trick is LARD, right? To remember that, we have a trick. The trick is LARD. What is the trick that we have? LARD. So left side is the association, right side is the dissociation. LARD is the trick here. Left side means association, right side means dissociation. Bache, when I'm saying association, to associate, to join, to form, so left side means oxyhemoglobin will form. What is the meaning here? Here it simply means that oxyhemoglobin form. Now when it is the right side, what is going to happen here? Dissociation, matlab oxygen and hemoglobin, they'll separate. Oxyhemoglobin breaks. Oxyhemoglobin, it breaks. Their breakup is there. Right side matlab their breakup. Oxygen and hemoglobin, they will have the breakup when it is the right side. Is that clear? Is that clear? So right side dissociation, to dissociate, to get separated, right? Matlab breakup will occur. Breakup will occur. Right? And left side mein, right, the relationship has just started. The patch up is there. Oxygen and hemoglobin's patch up is there. Okay, so when we talk about this left shift, my dear people, so here, right, you know already the conditions. The partial pressure oxygen has to be high, the partial pressure of CO2 has to be low, the H positive concentration needs to be low, which means the pH has to be high, and other than that, the temperature also will be low. These are the conditions. So left side means oxygenation, oxygen will form. So these are the conditions that we have for the left shift. And here one extra thing, this bisphosphor or diphosphoglycerate is given, that is also low here. Okay, so this is the left shift. So left shift mein your oxygen partial pressure will be high, rest everything is low. Now right shift mein you have to do the opposite of that. So right shift is telling me about the breakup. So where will this breakup occur? So B for breakup, beta, B for Bohr's effect. So right side means you are talking about what? You are talking about the Bohr's effect. Right side means you are talking about what? You are talking about the Bohr's effect. Right, bache? Right, bache? So it is the partial pressure of oxygen will be low, CO2 will be high. So obviously high H positive concentration means pH has to be low, and the temperature, because cells are metabolically active, the temperature will be high. The temperature will be high. That's what you have to remember. So where will this left shift occur? Of course, in alveoli, because these conditions are in alveoli. So bache, this is also known as Haldane's effect. Haldane's effect. And this side, this Bohr's effect, B for breakup, so it will be there in the tissue, right? So don't you think the breakup is important? Right? Dekho, ultimately, what is the job of hemoglobin? To transport the oxygen. Hemoglobin cannot keep the oxygen, because if hemoglobin will start keeping the oxygen, then our cells will die because they will not get the oxygen. They need the oxygen to oxidize the glucose. That is much needed, people. That is much needed, people, right? You have to understand this thing. So this breakup is very important, because if this breakup will not occur, oxygen will not go to the cell. Matlab, if there will be the breakup, then only oxygen will see some other person, will say hi, right? One bye is important for new hi, na? That is the same case here. So this Bohr's effect is actually responsible for the transport of oxygen, right? The Haldane's effect is responsible for the transport of CO2, and this Bohr's effect is responsible for the transport of oxygen. Where will it occur? Where will this right shift occur? In the tissue, right? In the tissue, and it is very important for the oxygen transport. So you will see hemoglobin as somebody, partial pressure of CO2 is high, I'll bind with CO2. Partial pressure of oxygen is high, I'll bind with oxygen. CO2, hemoglobin is just like that. But this is important because this is the nature of hemoglobin. Hemoglobin has to transport the gases. So hemoglobin cannot be loyal to somebody. Hemoglobin is like, whosoever will have more partial pressure, I will just go for that person. Right? I will just go for that person. So B for breakup, B for Bohr's effect, and these are the conditions for Bohr effect: high partial pressure of CO2, low partial pressure of oxygen, high H positive ion concentration, which means the pH has to be low and the temperature has to be high. Okay? Okay, so important it is, right? And rest 3% will be transported in the dissolved state. So here देखो, in the NCERT this line is given na, every 100 ml of oxygenated blood can deliver around 5 ml under normal physiological conditions. So I have explained you that. ठीक, and beta, it is believed that when the partial pressure of oxygen is 25 mm of Hg, the 50% of hemoglobin is, right, saturated. It is believed that the partial pressure is 25 mm Hg, the 50%, right? Then the 50% of the what? The 50% of the hemoglobin is saturated. Understood? Now, why this high H positive concentration increase? Why the pH is low? Now, these things will be clear to you when you will understand the transport of carbon dioxide. So, carbon dioxide ka transport will clarify everything, and mostly the question, it came from this particular part, that is the transport of CO2. So, are you ready to understand that? Yes. Okay. So, ready? So Bohr's effect clear, dissociation of oxyhemoglobin, and why? Because H positive concentration is more. Okay. Chali. So when it comes to the carbon dioxide, I told you it's 20 to 25 times. Right? Its solubility is 20 to 25 times. Its solubility is higher than the oxygen. So it can diffuse easily. It can diffuse easily. After finishing this chapter, you will get a break. Okay? And that break will be a short break for 10 minutes only. Done? So, transport of CO2. There are three things. In dissolved state, and that is 5 to 7%. That is 5 to 7%, right? It will be dissolved in plasma. Then beta, 20 to 25%, 20 to 25% in carbaminohemoglobin, in carbaminohemoglobin form. So when we say carbaminohemoglobin, what is the meaning here? It's simple. Beta, CO2 and, right, CO2 and hemoglobin, when they attach, it will form carbaminohemoglobin. It will form carbaminohemoglobin, right, bache? And when it is the carbon monoxide and hemoglobin, then, right, then it is carboxyhemoglobin. So I have seen students making this mistake a lot. That's why I am telling you, this is what you need to take care. Right, this is what you need to take care. Take care. So carbaminohemoglobin and carboxyhemoglobin, these are the points that we have. Take care. These are the points that we have. Remember, so carbon monoxide, when it binds with the hemoglobin, carboxyhemoglobin, and when your carbon dioxide will bind with the hemoglobin, it is carbaminohemoglobin. Take. Okay. Okay, so of course, partial pressure is something very important for this binding. Take, right? Partial pressure is something which is very important for this binding. Now, what's going to happen? We have to discuss about an enzyme that is known as carbonic anhydrase. What is the name of that enzyme? Carbonic anhydrase. Dekho, in the tissues, when metabolism is going on, the metabolic waste will be released. That is the CO2. So in the tissues, the partial pressure of carbon dioxide is high, right? The partial pressure of carbon dioxide is high, right, bache? So basically what is going to happen? We have to talk about one enzyme. Jaise ye dekho, within the RBC, this is going to happen, bete. Carbon dioxide and water, they will join. Which enzyme will help? Carbonic anhydrase. Let me tell you, this is the fastest enzyme. Which enzyme it is? It is the fastest enzyme that we all know. It is the fastest enzyme that we all know. Carbonic anhydrase. Carbonic anhydrase. And you know that this is the RBC. In the surrounding, plasma is also there. In the plasma, carbonic anhydrase is present in negligible amount. It is present in negligible amount. Now, when this carbonic anhydrase, when this catalyze this reaction, you will get H2CO3. What's that? It is bache carbonic acid, and it is a weak acid. It will immediately dissociate into HCO3 negative and H positive. That's the reaction that we have, right? It will give us what? This. It will give us what? This. Take. So it depends upon partial pressure. So what is happening here? What is happening here? So carbon dioxide and water, they will join with the help of enzyme carbonic anhydrase, which is the fastest enzyme, right? Which is the fastest enzyme. Now it will give us carbonic acid, right? It will give us carbonic acid, and again this enzyme will dissociate it. So now you know it very well, right? Now you know it very well. So this H positive, now you know it very well. See, H positive ions are producing. H positive, its concentration is increasing. Matlab pH will go down. Matlab pH will go down. And what about this bicarbonate? This bicarbonate is very stubborn. Do you know that? This bicarbonate is very stubborn. This bicarbonate is like, I want to go out. I want to go out. I will go out. Samajh aaya? Samajh aaya? This bicarbonate is like, I just want to go out. I cannot stay within. I have to go out. And what about this H positive? Now, H positive concentration is increasing. This H positive, my dear students, right, this will, right, they will combine with hemoglobin. It will combine with hemoglobin. Now you know that the partial pressure of CO2 is high. Of course, this is going to, that is why this reaction is taking place. This is going to break the oxyhemoglobin as well. Your hemoglobin now, this hemoglobin will combine with, this H positive will combine with hemoglobin to form hemoglobinic acid, right? To form hemoglobinic acid. Remember, this hemoglobin is acting like a buffer. It has to maintain the pH as well, right? It has to maintain the pH as well. Okay, bache? Okay, so it will form hemoglobinic acid. What will it do? Hemoglobinic acid. Now these bicarbonates, so majority of them, they are like, we want to go out. We want to go out in the surrounding, that is plasma. So majority of bicarbonate, they'll diffuse out from RBC into plasma. They will diffuse out from RBC into plasma. They like, we will go to plasma. We'll diffuse out, we will go to plasma. Now we have to maintain the balance here. So beta chloride ions from the surrounding, right? Cl ion from the surrounding will enter here. Will enter here because they have to maintain that balance, that ionic balance. So this particular phenomena is known as chloride shift. It is also known as Hamburger phenomena. It is known as chloride shift, and it is also known as Hamburger phenomena. So in this phenomena, what is happening? Chloride ions are entering from plasma to RBC, and bicarbonate is leaving the RBC and going into the plasma. Okay? Okay? Yes? Right? Right? So now these bicarbonates here combine with sodium to form sodium bicarbonate, that is NaHCO3. So majority of your carbon dioxide will be transported in the form of bicarbonate. So that is how it is going to be. That is how it is going to be. So this is what you have to remember, bache. Now what's going to happen? Now what's going to happen? This blood, right? This blood, it will do. Now what's going to happen? This deoxygenated blood will reach to the lungs, right? In the lungs, you know the conditions are opposite. Partial pressure of oxygen is high, partial pressure of CO2 is low, you know all the things. So reversal will occur. Now in the lungs, what's going to happen? Now in the lungs, what's going to happen? Beta, basically you can say, right, whatever reactions we have studied, opposite of that will occur. Jaise ye dekho. This is the diagram that we have. Right, with the help of this, if you remember these reactions, they are easy. Now here in the alveoli, the conditions are opposite. So because of that opposite condition, because here the partial pressure of oxygen is high, the partial pressure of CO2 is low. Now this bicarbonate, sodium bicarbonate, again it will dissociate, bache. This bicarbonate again will be like, ki I will enter within the RBC. Right, I will enter within the RBC because in the lungs Haldane effect is going on. So these bicarbonate that will dissociate from sodium, they will enter in the RBC. Again, chloride ion will go out. So we call it as reverse chloride shift. We call it as reverse chloride shift, right, bache? So now, of course, because of that, these bicarbonate, now again hemoglobinic acid will break, H positive will again combine with this HCO3 negative. Who's catalyzing the reaction? Carbonic anhydrase. Again carbonic acid will form, CO2 and water will form, and this CO2 will go out. That is how the Haldane effect here will help in the transport of carbon dioxide. Okay, that is how Haldane effect here will help in the transport of carbon dioxide. Is that clear? Are you sure? Take done. So these are the reversal reactions. Okay, so under normal conditions, basically 4 ml, right? 4 ml of CO2 is delivered to the alveoli by every 100 ml of oxygenated blood. That's it. Baki regulation of respiration. This is, bache, very important when it comes to the regulation of respiration. The partial pressure of carbon dioxide in the arterial blood, in the oxygenated blood, it matters the most. Right? Trust me, it matters the most. Ye dekho. Neural system is going to help here. And in neural system, we talk about the respiratory rhythm center. So that special respiratory rhythm center, right? This special respiratory rhythm center, where is it present, bache? The special respiratory center is there in the medulla region of the brain. It is there in the medulla region of the brain, and we call it as respiratory rhythm center. We call it as respiratory rhythm center. So it is mainly responsible for the regulation, right? There is another center that is in the pons region. Both medulla and pons region, they are the part of hindbrain. They are the part of hindbrain, right? So they have pneumotaxic center. One more, apneustic center is also there. Okay, but let's stick to the NCERT there. So respiratory rhythm center is going to, it is the respiratory rhythm center. It is mainly going to control this regulation, and this pneumotaxic center in the pons region will moderate the function of respiratory rhythm center. That is a PYQ. That is something very important, right? That is something very important. So when there is a neural signal from this center, it can reduce the duration of inspiration and alter the respiratory rate. So basically, respiratory rhythm center in medulla is mainly responsible for the regulation, and in the pons region, pneumotaxic center is there that will moderate, that will even control the respiratory rhythm center. This is the point. Okay, so beta, there is a very highly sensitive to CO2 and H positive concentration. There is a highly sensitive area near to respiratory rhythm center. It is very sensitive to CO2 concentration. Means if there is a little bit of change in the CO2 concentration, it will get activated. And when that CO2 is changed in the arterial blood, when the partial pressure of carbon dioxide is more in the arterial blood, right, then it will send a signal for the inspiration, right? And it will make the necessary changes. And other than that, receptors that are associated with aortic arch and carotid artery, the carotid artery, the one that will carry the oxygen to the brain, right? They also recognize changes in CO2 and H positive concentration, and they send the signal to the respiratory rhythm center, right? So the role of oxygen in the regulation of respiratory rhythm center is quite insignificant. So that is why again and again I'm repeating the same point only, beta, that it is the CO2 concentration that will play a major role here. Okay, okay, right? And even body temperature and blood pressure also affect the activity of the respiratory rhythm center. Done? So it is always like this, that partial pressure of CO2, if it is more in arterial blood, that is in oxygenated blood, then there is a signal for inspiration, right? Then there is a signal for inspiration. Am I clear? Disorders, you know that, important. So first is asthma. First is asthma. So when it is asthma, it is the difficulty in breathing and the wheezing sound is there, right? Why there is? So inflammation of bronchi and bronchiole. Write down this line, important. Emphysema, right? Very important, bache. Those who are the chain smoker, those who smoke on everyday basis, their alveolar walls will get damaged, right? Respiratory substrate is decreased, so of course the exchange will get affected. You know, they need alpha-1 antitrypsin. Alpha-1 antitrypsin is given to treat this emphysema. Do check the reason why. Then occupational respiratory disorders, right? जैसे asbestosis, silicosis, जैसे let's say some people, they work in the grinding or stone breaking industry, dust is produced there, beta. So body has to cope up with that, body has to fight with that. Okay, so long exposure to that will result in the fibrosis and the lung damage will be there. Take, that's it. So next chapter that we are going to start is neural control and coordination, right? This is the demand of our students now, so how can we miss that? Chalo, neural control. आ जाओ जी, आ जाओ जी. Let's discuss neural control and coordination. So excited? हाँ जी, excited? No. No. Break? चलो ठीक है. Let's have a break for 15 minutes. So 15 minutes means 15 minutes only, okay? I'll sit here only. Take. So let's meet at 1:30 a.m. And people, you know that, right? We have a special class on Tuesday, right? We have a special class on Tuesday at 5:30 p.m. Okay? Hi everyone. What's up? Hello, what's up? Chalo. So let's start neural control and coordination. So now from this chapter, eye and ear topic, it is deleted. So we are left with the ionic theory of nerve impulse conduction. And the second topic that we need to focus here is the brain part. Neuron, of course, if you have to understand ionic theory of nerve impulse conduction, you really need to know about the neuron as well. So I thought that I'll give you the break for 15 minutes, but luckily you got it for Hanji, 25 minutes. So now for next 2 to 3 hours, do not expect break. Check, do not expect break. So what we have to do, neural control and coordination directly. Let's come to the point here. Okay. So beta, here the very first thing that you need to understand is that neural system. So neural system, you know that neurons will be there, and when we talk about the neurons, they are also known as the nerve cells, and they are the longest cells of human body. What are they? They are the longest cell of human body. Okay, so they detect, they receive, they detect, they receive, and they transmit different type of stimulus. They detect, they receive, and they transmit different type of stimulus. Okay, so neural organization, I told you already in animal kingdom as well, bache, that for the very first time you will see it in Cnidaria and Hydra, and here you will see the network of neurons. So that is a PYQ. Okay, that is a PYQ. So here we talk about the brain along with the number of ganglia and neural tissue, and vertebrates, they have more developed neural system, right? What do they have? They have more developed neural system. So of course in the neural system, if we have to make that flow chart, right, let's go as per NCERT only, right? Because see, brain is a memory based topic, so we have to revise it very quickly, right? But we have to revise it very quickly. Okay, so I need your support in that, and I want you people to please pay attention here, right? If you will keep on typing unnecessarily things now, if you keep on typing such kind of things now, whatever you are doing in the chat section right now, trust me, I'll end the class, right? I'll end the class. So Devashish and all, I'll block you all people, or I'll end the class. And I'll prefer to end the class because you are not interested in reading biology, right? I want you people, whosoever is chatting here and whosoever is thinking biology is easy, please after the NEET examination, right, do let me know your marks in the comment section here, right? Do mention that, and even you can send to me on the Telegram, or right, or even you can mail that to me, right? I really want to know about your marks. Literally, I really need to know about your marks. Trust me, you are not going to get good marks. I know majority of students, they have this mindset, biology is easy. I'm not saying it's not easy, but it is vast. There are so many things that you have to remember, and you have to keep on revising them. Okay, we are teaching from so long, right? But still, there are few things in which we also get confused. Okay, in which literally we also get confused. So that is why it's very important, right, to revise the things again and again. There are some things which you need to understand. There are some things that you need to mug up as well. But all you are doing is, "Arey madam, he is proposing me this and that." What nonsense is going on? Can't you just focus here? Why are you chatting even? Right, I'm not going to block the spammers. I will directly end the class because I don't have free time. I have other things to do as well. Okay. Right, stupid people, when we take the series, when we discuss everything, every time you are not completing this, you are not. When we are teaching, you are not even listening to us. That is why we don't, like, even at least I am talking about myself. I'm not talking about other teachers. I don't have that enthu left because I have seen that students, they are not taking the subject seriously. They are not at all interested in knowing something. So neural system, here we talk about two things. One is central neural system, and then there is peripheral neural system. We have central neural system, and then what do we have? We have peripheral neural system. Central neural system is there, and then what do we have? Yes, people, we have the peripheral neural system. So when you people talk about the central neural system, right, we have brain and spinal cord. What do you have? You have brain and spinal cord is there. Okay, this is the central nervous system, central neural system. So bache, brain and the spinal cord, right? Of course, the nerves, they have to emerge, they have to go out, they have to carry the information. There should be the nerves that it will carry the information to the central part, and there should be the nerves which will carry that information to the target organ as well, right? They need to carry the information to the target organ as well. So when we talk about this particular part, we have the peripheral neural system. What do we have there? We have the peripheral neural system, and in the peripheral neural system, you will have cranial nerves and you will have spinal nerves. Right, cranial nerves and spinal nerves, they are going to make it. And when it comes to this peripheral nervous system, bache, we will talk about what? Do you know that? What are we going to talk about here? What are we going to talk about here? We will talk about the autonomic neural system. We will even talk about the somatic neural system, right, bache? What we have to discuss here? We have to discuss the autonomic neural system here, and you will talk about the somatic neural system. So when the word is somatic, right, when the word is somatic, somatic neural system is the word. As for somatic, as for skeletal, right, this will innervate skeletal system. So of course it has to be what? It will be voluntary. It will be voluntary. Now this autonomic one, it is involuntary. So even your visceral, right, even your visceral nervous system will come here. So autonomic neural system mein beta, when it is the autonomic neural system, when it is what? When it is the autonomic neural system, you will talk about the sympathetic neural system, you will talk about the parasympathetic neural system. You will talk about the sympathetic neural system and you will talk about the parasympathetic neural system. That's what you have to keep in your mind. Is that clear? That is what you have to keep in your mind. So you can just have a look here. Right, you can just have a look here. So here you people can see the terms as well. You'll be able to understand this. So efferent fibers and efferent fibers are there. So for efferent and efferent fibers, the trick is ASME. What is the trick, bache, that we have here? The trick is ASME. What is the trick that we have here? ASME. Efferent means sensory, motor means efferent means motor. So efferent is sensory. So what is the function of sensory? It will carry the information from target organ to the central nervous system, and this motor one will carry the processed information, right? Basically sensory will carry the stimulus to the brain and nervous spinal cord, and this will bring back the information to the target organ. So that is why the word here is ASME. Efferent, sensory, motor, efferent. Efferent, sensory, motor, efferent. Efferent, sensory, motor, efferent. This is what you have to keep in your mind. So as I said, PNS is having two types, somatic neural system and autonomic one. So somatic will have S for somatic, S for skeletal. So when you talk about your skeletal system, it is what? It is, when you talk about your skeletal system, it is voluntary. Now, when you are talking about the autonomic neural system, so here it will relay the impulse from CNS to autonomous neural system. What will it do? It is further classified into sympathetic neural system and parasympathetic one. So sympathetic, bache, it will deal with the situations like fight or flight. Right, fight or flight, situations like that. And when it is the parasympathetic, it will deal with the situations like rest and digest, right, where relaxation is there, like this. Let's say, for an example, when you experience something, right, where you are just getting scared, right, what is going to happen? Your heartbeat will increase, right, you'll start sweating. So basically, who's going to be active there? Sympathetic nervous system. So it is responsible for increasing the heartbeat, it is responsible for that sweating, it is responsible for the dilation of the pupil as well. That's how you have to relate the things. And then it comes the visceral nervous system, and you know that it is also involuntary. It is also involuntary. So it is having all that nerves, fibers, ganglia, plexus, meaning network, right, by which impulse will transfer from CNS to the viscera, where we are talking about all the body organs. Take all the body organs. Now, as I said, we have to be quick, otherwise we will not be able to finish it. So let's have a look of this neuron. What type of neuron it is? It is a multipolar neuron. What type of neuron it is? It is a multipolar neuron. Here, bache, if anybody is interested in knowing about this chapter in a detailed manner, right, there is a detailed one shot available on this channel, right, you people can access that. Number one. Number two here is, right, number one. Number two here is, if you want, right, if you if you're like, no ma'am, we are just preparing for NEET 2025, we need the brief part of that, then I'll go as per the NCERT, right? I'm going to follow the same sequence only. So when you are talking about this multineural neuron, or ultimately when it is the neuron, what do you discuss? You talk about this part, right? What this part is, first of all, we have to discuss about the cell body here. What we have to discuss here, right, in simple words, when there is a neuron that we have, so we will talk about the cell body, which is also known as cyton, which is also known as perikaryon, which is also known as soma. And this cell body is having the processes. This cell body is having the processes, right? For that we use the word neurites, right? And here you talk about the dendrons, singular dendrites, or you are going to talk about the axon. You are going to talk about the axon. Take bache, right bache? So it is, right, when you are talking about this cell body, right, when you are talking about this cell body, of course, it is what? It is uninucleated, right? And remember, because you know that here the G0 phase will be there now, so you know that centriole is going to be absent here. We are not going to see the centriole. Other than that, the other thing that you have to remember is Nissls granules. What these Nissls granules are, right? They are the masses of, right, they are the masses of endoplasmic reticulum and ribosome. So in simple words, you can say they are meant for the protein synthesis. And other than that, neurofibrils, right, these proteins, cytoskeletal proteins, are also present here. Are you getting my point? They are also present here. Are you getting my point? So you are talking about the cell body, so it is having all the organelles except centriole, right bache? So Nissl granule and neurofibrils are also there in the nerve cell. Is that clear? Is that clear? Are you sure? Are you sure? So they are the aggregates of endoplasmic reticulum and the ribosome. So they will help in what? They will help in the protein synthesis. So these Nissl granules, they are also known as tigroid, right? Tigroid body. So if this will be the word that comes in the paper, so don't get disturbed. Okay. So this is something that you have to keep in your mind. Okay. Okay, okay, so protein synthesis will occur here. Done, bache? Done. So neurofibrils, they are just the cytoskeletal element that will help in the internal conduction here. And these are the branches, these are the dendrites. You know that dendrites will get the information from the presynaptic neuron. It will carry the impulse towards the cell body, right? So dendrons, in the case of dendrites, what is the word that we have to remember? That they carry the impulse towards the cell body, right? So towards the cell body, matlab from periphery towards the center. So it is centripetal is the word that you can use, and this is what you have to remember. Now this part is the axon. So when you look at this axon, the cylindrical part, this portion here, beta, this is known as axon hillock, right? This is known as axon hillock. It is the most sensitive part here. It is the most sensitive part of neuron here, right? And it is having many ion channels. What does it contain? Many ion channels. This is the axon here. So if you look at it, these are the Schwann cells. They are forming the myelin sheath in surrounding. I'll discuss about it as well. So at places, right? This is like this. Let's suppose this is first myelin sheath, second. So there is a gap always in between. There is a gap always in between, right? In between the Nissls granule. That is known as node of Renvier. So at this node of Renvier, right? At this node of Renvier, myelin sheath is absent. Myelin sheath is absent. Is that clear? Is that clear? So dendron will receive the stimuli. It will produce what? Centripetal towards the cell body. Centripetal conduction is there. And of course, this axon will carry the impulse away from the cell body. So can I say that when it is the axon, it is the centrifugal conduction? Can I say so? Can I say so? So you know that here, these are the terminal ending synaptic knobs or terminal arborizations. So this part regarding the structure clear? This part regarding the structure clear? Now, in axon, Nissls granules are absent. They are present in, they are absent here, right? This is something very, very, very important. In NCERT, it is not mentioned exactly like that, but that is how it is. Okay, so dendron mein to you will see, but in the case of axon, Nissl granules are absent. Okay, Nissl granules are absent. ठीक है बच्चा? ठीक है बच्चा? And overall in axon, only neurofibrils and Mitochondria will be there. Only neurofibrils and mitochondria is there. Only neurofibrils and what is there? Mitochondria is there. Is this information important? Yes, it is very important. Okay, so in dendrites, you will have Nissel's granules, but that granules are absent in the axon. Is that clear? Is that clear? Are you sure? Are you sure? So you know that it is the axon basically, which is the functional part of the nerve cell, right? It is actually the functional part of the nerve cell. Is that clear? Right? Functional part of the nerve cell. Is that clear? Are you sure? So now, जैसे some axons, in their case, जैसे we call it as Schwann cells, then you have the myelin sheath like this, right? That is what we have. That is what we have. So here when you look at it, right? Here when you look at it, the gap is there in between. We call it as node of Renware, right? Node of Renware. And this is what? This is the myelin sheath. I'll tell you what myelin sheath is. This is the myelin sheath. So this axon is the myelinated or medulated axon. And it is not mandatory. It is not mandatory that all the axons are going to be like this. No, students. Some are not having this myelin sheath, right? So they are known as what? They are known as non-myelinated. They are known as what? They are known as non-myelinated or non-medullary neuron, or you can say that non-medullary axon. So in this case, when this type of neuron we have, the ionic theory of nerve impulse conduction is there, right? The ionic theory of nerve impulse conduction is there that we have to study. Okay, when it is the case, then it will be saltatory conduction. Basically, jumping will be there. Jumping of impulse will be there from this side to this side to this side to this side. So, of course, it is going to be fast. It is going to be fast. Now, when we talk about the myelin, okay, ma'am, you told us about the. So, what is this myelin? What is this myelin? It is bache fatty material which is having high electrical resistance. It is the fatty material having high electrical resistance. It is the fatty material which is having what? Which is having the high electric resistance. It will not allow the ions to go here and there. So it is basically just like an insulator. It insulates here, right? It is just like an insulator. This part is clear? This part is clear? Yes. This part is clear. So, of course, this is the medullary sheath, right? This is the myelin sheath, right, bache? Yes or no? But do remember, whenever you talk about the myelin sheath, right, this myelin sheath is also covered by, right? This myelin sheath is also covered by a very thin cell membrane here, right? This myelin sheath is covered by a very thin cell membrane here, and that cell membrane is known as neurilemma. So neurilemma is outside this myelin sheath, bache, right? So of course neurilemma is at NOR, node of Renware region as well. So you know the node of Renware. So at places myelin sheath is not covering this axon. This is the node of Renware, and neurilemma is even covering this. Neurilemma is even covering this node of Renware. So neurilemma is the thin cell membrane which is basically doing what? Which is covering this medulla, या medullary sheath. So this is the neurilemma. So neurilemma is also known as the sheath of Schwann cells. It is also known as the sheath of Schwann cells. So you people can write down these points. Okay, it is also known as what? It is also known as the sheath of Schwann cells. Any doubt here? Any doubt here? Yes. Yes, so bache, remember, there are in the brain, in the nervous tissue, we just do not have the neurons, we have other cells also. So you can write this point directly that in PNS, that is your peripheral nervous system. In PNS, right, the myelin sheath is formed by Schwann cells. The myelin sheath is formed by Schwann cells, and the cells, right, the neurons which are not having the myelin sheath, in their case also, the Schwann cells are always present, right? Even if they are not synthesizing the myelin sheath, they'll still be present, right? So in PNS, myelin sheath is formed by this. Yes, bache, it is formed by this. Is that clear? Is that clear? And in CNS, that is your central nervous system. So when you people talk about the CNS, who's going to form the myelin? It is going to form, myelin sheath is formed by oligodendrocytes, right? So you have to, it is formed by oligodendrocytes, right? These are the type of neuroglial cells. Take, so remember it like this. Right, commanding officer. C for central nervous system, O for oligodendrocytes. Right, so CO you have to remember, or CO you have to remember. ठीक, CO you have to remember. ठीक, so even if there is an unmyelinated nerve fiber, बच्चे, even if there is an unmyelinated nerve fiber that will have the Schwann cells, they will not form the myelin sheath, but they will have this. ठीक, okay, so ये देखो, this is the point that I'm talking about right now, that ये देखो, myelinated nerve fibers, they are found in cranial and spinal nerves. Important statement it is, right? Important statement it is कि myelinated nerve fibers are found in spinal and the cranial nerves, and unmyelinated nerve fiber is also enclosed by Schwann cells that will not form the myelin sheath around the axon, and it is in autonomous and somatic neural system. This part is important. Do highlight it in your NCERT. Is that clear? This part is important. Please highlight it in your NCERT. Done? Sure? If it is done, do let me know, please. 1 minute. If it is done, do let me know, please. Yes, bache. Is it done? 1 minute. Done? Sure? This is literally important, right? This is literally important. Do not miss it. Okay? Do not miss it. Done? Okay, chaliye. So mediated, non-mediated nerve fiber, I hope this is clear. So myelin is basically lipid, proteins and water. What is this myelin? It is basically lipid, protein and water. So neuron, its examples and everything should be clear to you. Okay? So this part, as I said, is very important. Now, here there are words now: multipolar, bipolar, unipolar. Chalo, let's discuss that words also. Ye dekho. This is your multipolar neuron, okay, which is having many processes. Just look at it. This is your multipolar neuron, which is having how many processes? It is having many processes. Now, when you look at this part, this is unipolar. Uni means single. What is the meaning of uni here, bache? Uni means single, right? This cell body is having a single process only. Yes or no? Right, this is arising from the cytosol, a single process is there, and basically here you will see only one axon is there, only one axon is there, right? So nervous system, right, nervous system in the case of embryo will have such type, right? So unipolar will be there in the early embryos, it will be there in the early embryos of vertebrates and invertebrates, it will be there in the early embryos of vertebrates and invertebrates. ठीक, by means two processes are arising, one is dendrite, one is axon. So here the example is important. No doubt eye topic is deleted, but this is important, this is important. So you will see it in the retina of eye. So when we talk about the retina of eye, na, we talk about the layer, right bache? We discuss about the layer, jaise ki what do you discuss? Photoreceptor cell, bipolar cells and the ganglion cells. So ya fir inside to outside will be ganglion cells, bipolar cells, and then you will have photoreceptor cells that will include rod and cone. And other than that, olfactory epithelium, right? Olfactory epithelium is also having olfactory epithelium is also having bipolar neurons. Am I clear? Then pseudo means false. Pseudo means false. Uni means single, right? So here, beta, one process will arise, right? But it will again divide into two. Matlab it basically from the cell body one process is arising, but it will divide into two. So in the case of, right, where will you see it? In the dorsal root ganglia of spinal nerves. Where will you see this? In the dorsal root ganglia of spinal nerves. Are you getting my point? And then this is the multipolar that we have. So multipolar, it is in the cerebral cortex, right? It is in the cerebral cortex. So remember the examples that are important. And some are apolar neurons as well. Okay, some neurons are even apolar as well. ठीक है जी? So that's it, right? That's it. So this is about the types here. You should know about it, right? And I hope this part is also clear to you, right? जैसे ये देखो, this is the arrangement. This is your one neuron, and then followed by next is there. So at the end, when you talk about this part, my dear students, these are the neuron endings like this. Like this, and here let's suppose this is the dendrite of next neuron. So in between these two, this gap is actually known as synaptic cleft. This gap is actually known as synaptic cleft. So this one is your presynaptic membrane, this one is your presynaptic membrane, and this one is your postsynaptic membrane, your postsynaptic neurons membrane, whatever you want to write right now you can. And in between these two you have the synaptic cleft. So these 1, 2 and 3, these three, they form actually the synapse. They form actually the synapse. ठीक? They will form the synapse. And you know that the neuron ending of one neuron here, there will be certain vesicles. We call them as neurotransmitters, my dear students. What do we call them? We call them as neurotransmitters, and they'll release here once the impulse will reach. Okay, so this part is clear. This part is clear. Neurolemma clear. Neurolemma clear. So here देखो, this is one neuron, and then there is the second. Chali. So now let's understand, after this part, let's understand actually that what exactly is this ionic theory or what of nerve impulse conduction. So I will try to simplify it for you as much as I can. So ionic theory of nerve impulse conduction. So of course you will discuss it in non-myelinated nerve fiber. Okay, where will we discuss it? We will discuss it in non-myelinated nerve fiber. Okay, but yeah, before that, let's discuss the different type of cells as well. ठीक है, let's discuss different types of cells as well. ठीक है, जैसे in the case of neuron, according to you, मतलब in the case of nervous system, according to you, how many types of cells are there? How many different types of cells are there? When you talk about the nervous tissue, right? When you talk about the nervous tissue, so according to you, how many types of cells are there? Dekho, we also talk about the neuroglial cells, which are making more than 50% of the nervous tissue. Neuroglial cells are there. Which cells do we have here, beta? Neural glial cells are there. What is their role? They are the supporting cells. They are for supporting, packaging and protection, right? They are the supporting cells, right? They are for the supporting of cells. They are helping in the packaging. So here, what do you discuss? You talk about the astrocytes. Aster is the word, they are star-like. You talk about the oligodendrocytes. You talk about the microglial cells as well. So my dear students, when we talk about this microglial cells, they are the macrophages of nervous tissue. Or you can say that phagocytotic cells of nervous tissue. Okay? Phagocytotic cells of nervous tissue. Microglial cells. And astrocytes, bache, astrocytes, they are again, as I said, they all are for the support and the packaging, right? So these astrocytes, they form blood-brain barrier. I'll show you the picture as well. They'll do what? They'll form the blood-brain barrier, right, bache? So astrocytes will form it, right? Astrocytes, they are having, bache, cell junctions. Astrocytes, they are coupled with the cell junctions, and they will not, right? They do not allow the toxins to enter. They will not allow the toxins to enter. Is that clear? Right? Neurotoxins to enter. Is that clear? Is that clear? Now these oligodendrocyte, just this one is having many processes. You know that they'll form myelin sheath. They'll form myelin sheath in central nervous system. right? So there are different types of cells, neuroglial cells they are. Okay, ji? So they are found in the brain and the spinal cord. They help in the supporting the nerve fibers. Is that clear? Is that clear? So they comprises about 50% of nervous tissue. Right? About 50% of the nervous tissue. Is that clear? Sure? Are you sure? Right? So other than that, bache, we have ependymal cells and neurosecretory cells are also there. Other than that, na, ependymal cells. These cells, bache, they line, just today in structural organization also, just in brain, right? The cavities, the ventricles are there, they are lined with ependymal cells. So these ependymal cells, they form that there is a fluid, na, cerebrospinal fluid, CSF, sorry, cerebrospinal fluid. Then neurosecretory cells are also there that we discuss in the case of hypothalamus. Okay, so they are also something that we see in the nervous tissue. They are also something that we see in the nervous tissue. So ependymal cell, they are present in that ventricles, that cavities of the brain, right? And even in the central canal of the spinal cord. So they will form the cerebrospinal fluid, bache. Right. So yes, right. So they will have many, brush bordered, they can be, ciliated, they can be. So in the absorption and flow of cerebrospinal fluid, they will help. These ependymal cells will help in the absorption and the flow of that. Neurosecretory cells, you can relate it with the hypothalamus because you know that they will form the neurohormones. They will form what? Neurohormones. So these are the cells that we will see in the nervous tissue. Is that clear? These are the cells that we are going to see in the nervous tissue. Now, we have to discuss this ionic theory of nerve impulse conduction. First of all, when you talk about the neurons, please try to understand the topic because everything is in the NCERT. They are excitable cells. Matlab, these cells are having the property that they can react to a stimulus, isn't it? Isn't it? Right, they are excitable state, right? Basically, they are excitable cells, means they can react. They can react to the stimulus. They can just do what? They can just, yes, bache, they can basically react to that stimulus that can conduct it. Now, why? Why? See, as I said, we have the ionic theory of nerve impulse conduction. So, who has given that? Hawking and Huxley. So, if you want me to explain it now, you have to understand each and everything very carefully. So once you will understand each and everything very carefully, then trust me, the topic is going to be easy for you, right, bache? The topic is going to be easy for you. So in the case of neurons, they are excitable cell. They have the differential permeability as well. It is their special property, right? In the case of neurons, you are going to see that they have so many ion channels. We will discuss about that ion channels also, right? Other than that, you have to remember that when it comes to their membrane, right? When it comes to their membrane, right? Their membranes are in polarized state. Why is it so? Because they have differential concentration gradient of ions across it. They are selectively permeable in nature. Ye dekho. Here it is written. I'll explain each and every line here, right? Each and every line I'll explain. What are they saying? I will not focus on the writing here because ultimately it is the NCERT that we have to remember, right? But I will explain each and everything. So firstly, tell me, up to this part all clear? Up to this part all clear? Sure? Done? Sure. So this is the neuron, right? Just here it is clearly written that they are the excitable cells because their membranes are in polarized state, right? They are the excitable cells because their membranes are in polarized state. Can you see that? Right? One minute. ठीक, चलिए, excitable cell because their membranes are in a polarized state, right? Right? Do you know why the membranes are in a polarized state? Because they have differential concentration gradient of ions across it. Why is it so? I'm writing the answer. Because due to the differential ionic concentration across it. That is why they are like this, due to the differential ionic concentration. This is a neuron, excitable cell. It's a special cell. It can respond to that. Actually, when you talk about the neurons, na, right? When you talk about the membranes, na, but we talk about certain ion channels. We discuss about certain ion channels. You know, overall in our body, there are different types of ion channels, right? But different types of channels are there. Just say, let's suppose if somebody is going to press our hand, right? They are going to do this to our hand. We are able to feel it, right? Right? We have certain mechanical, like we can feel such kind of things. You know that. So basically, when you talk about these ion channels, na, there are different types of ion channels. Just like we will discuss about the leaky channels, right? We discuss about the voltage gated ion channels. Leaky ion channels, voltage gated ion channels, these are also that we discuss, right? Other than that, we also talk about ligand gated ion channels. They will open and close due to the change in the voltage, right? They will open and close when a suitable chemical will bind to them. So leaky channels, voltage gated ion channels, ligand gated ion channels, and etc. And you know that we also talk about certain pumps, right? We also talk about certain pumps, right, bache? Right, bache? So there are different types of ion channels which are there in the neuron membrane. ठीक है जी? Right, there are different types of ion channels which are there in the neuron membrane. So जैसे voltage gated है, ligand gated है, your leaky channels are there. Some are even. Right, some are even, right? They are even sensitive to what? They are sensitive to touch, pressure. Just even I told you that if somebody is going to hold my arm like this, I'm able to feel it, right? I'm able to feel it, isn't it? Isn't it? So basically, there are what? There are there are some channels which can respond to that. They are basically mechanical channels, yeah, mechanosensitive channels it is. Okay, so there are different types of channel: ligand gated, voltage gated, mechanical gated, ligand gated, voltage gated, leaky channels, mechanical gated channels that are sensitive to such kind of things. Okay, there are different ion channels. Other than that, there are pumps that we have to discuss. Now, for this particular topic, we need to know about these two, right? We will be discussing about these two. There are different types of ion channels, but to know this topic, these are the type of ion channels which we need to focus. Okay, right? Which we need to focus. Dekho, overall, if I talk about the neuron, right? This is your that axon is a functional unit. Let's consider that exoplasm. This is the surrounding extracellular fluid. Beta, overall, when this neuron is not conducting anything, and overall, if I have to tell you, overall, the number of ions are same here. It's not like that it varies, right? Overall, the it can vary. Just let's suppose inside this neuron, I'm giving you one example, then I'll come to the ion channels. Your potassium is high, outside potassium is also there, it is low. Outside sodium is high, inside it is low. Okay? Inside you will have more, organic proteins which cannot control the ion channels here. And outside also you have some chloride ions which, they are not at all, right? They are not at all helping in this particular process. Okay? They are not at all helping in this particular process. So these are the various things that we have here, right? These are the various things that we have here when we talk about the neuron. So now the point here is, when a neuron is not conducting, right? When a neuron, right? So differential permeability, right? Already I told you that why they can conduct, right? Because they have, right? Why can they conduct? Because they have differential permeability. This is something that I told you already. Okay, this is something that I told you already. So I am saying that when you look at this neuron, right? I'm now telling you about certain ions. I am saying when you look at this neuron, this is the membrane that we have. And again, I'm using this word because it is going to be very helpful here, that this membrane is having differential permeability. This membrane is having differential permeability, right? What this membrane is having, people, please listen to me carefully. What this membrane is having? This membrane is having the differential permeability, right, to sodium and potassium ions. That is going to help in the nerve impulse conduction. This is what we have to understand, right, bache? Right, bache? So this differential permeability and electrical excitability, these are the two things, right? These are the two things, bache. This differential permeability and this electrical excitability, electric field, right? They will help in, right, exciting the fiber. They will help in conducting the nerve impulse. This part is clear? This part is clear? Yes or no? Yes or no? So I am telling you about this membrane. I am saying that outside also there are certain ions. Inside also there are certain ions. Overall, the concentration of this ion is same. They are of different types, right? Overall, but overall in totality, if I have to tell you, these ions are almost same, but it's just that there are two different types of ion channels. So can I say that this membrane is separating, because outside also you have the ions, inside also you have these type of ions. So can I say that this membrane is separating two different types of solution? Can I say that this membrane is separating two different types of solution? Yes or no? That this plasma membrane here is separating two different types of solution. They have different chemical composition. They have different chemical composition, but overall, the total number of ions are same. Right? Total number of ions are same. Just say outside, remember this. Outside, remember this. Sodium is more, potassium is less. Chloride is also there, but it is playing no role, so we are not discussing. Inside, sodium is less and potassium is more. Moreover, inside there are organic proteins. There are basically proteins. These proteins, they have high molecular weight. They cannot cross this plasma membrane. They have high molecular weight and they cannot cross this particular plasma membrane here. ठीक है जी, they cannot cross this particular plasma membrane here. So of course, inside we will have more, and these proteins are negatively charged. So inside we have more negatively charged ion. ठीक है? Okay. Right. So outside your sodium ion is predominating, right? Chloride ion is also there, but that is of no role here. That is having no role here. So I'm not discussing. Inside, it is your potassium that predominate. So in normal condition, when it is not conducting anything, this is the situation. Is that clear? Is that clear? Is that clear? Sure. Sure. So outside and inside, as I said, these proteins are there which cannot leave. They have negative charge. So inside normally you will see is negatively charged, outside is positively charged, right? This is what we see when the neuron is not conducting. I'll come to the channels also. When your neuron is not conducting, means when it is not conducting means it is at rest. So this is the resting potential, right? It is at rest. Just when you are doing rest, when you are not doing anything productive, you feel negativity inside na, you feel low inside na, yes or no? You feel negativity inside na, you feel low inside na, yes or no? Yes or no? You feel negative, you feel low inside, yes or no? है ना? Right? 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 है ना? So अब you have to understand, this is the one aspect, right? I'm breaking this topic into two parts, right? Just that so that you can understand. Now you know the structure of neuron. Just imagine this is the neuron. I told you that inside, intracellular like composition of ion is different, extracellular it is different. But I told you which one will predominate where. Overall on the neuron membrane, there are different types of ion channels. Just say you will talk about the leaky channels also here. You will talk about the voltage gated ion channels also, right? And we have to discuss about certain pumps as well. That is your sodium, potassium, ATPase pump. That's what we have to discuss here. Now, when I'm saying leaky channels, when I'm saying what? When I'm saying leaky channels, they are potassium leaky channels and they are sodium leaky channels. They are potassium leaky channels and they are sodium leaky channels. Potassium and sodium leaky channels they are. The word is leaky channels. The word is leaky channels. What is the word here? Leaky channels. So here, down the concentration gradient, or I would say passively ions flow. passively ions are going to move. This is the role of leaky channels here. And when you talk about the voltage gated, the word is voltage gated. So you have sodium voltage gated channel, you have potassium voltage gated channel, right? So there will be the change in the voltage and these channels will open. It's like that, their gate will open. So they are always open, right? Along the concentration gradient things will move and they will open and close at the specific, right, voltage. Now the number of, right, remember, potassium leaky channels are more in comparison to sodium leaky channels. These are the facts that you need to remember. Now, this is the one line. And I told you that this membrane is having differential permeability for sodium and potassium ions. Now, understand that. Dekho, outside sodium is more, potassium is less. Inside potassium is more, sodium is less. Now, try to understand few things, right? Just I told you when the neuron is resting, this has to be the condition. Inside negative, outside positive. This is the polarized state. Your membrane is polarized. This is due to the different ion concentration and it is due to the differential permeability of membrane. Now I am saying that how is it possible? I am saying like, let's suppose this is the neuron. But here you have leaky channels. Let's suppose these are the potassium leaky channels and let's suppose these are the sodium leaky channels. So basically potassium leaky channels, their number is more. So of course more and potassium leaky channels, their number is more. So more and more potassium ion will go outside because inside it is more, outside it is less. So more and more along the concentration gradient it has to move. So more and more potassium ions will go outside. Now sodium ion, they are more outside, they are less inside. So they are, right, through leaky channels, what will be their flow? They will move from outside to inside. But in comparison to potassium, their concentration that will enter inside is low. This is the number one reason. Number two is we have sodium potassium ATPase pump. What do we have? We have sodium potassium ATPase pump. What do we have? We have sodium potassium ATPase pump. It's a pump, right? Here there are few tricks that you need to remember. The tricks are no kiya kintu. No kiya kintu. N means sodium, O means out, K means potassium, I means inside, A means ATP, A means against. And here kintu means K for potassium, I in means inside, and 2 potassium will move inside. So what is the meaning of this? Just say here, I told you we have the pump, sodium potassium ATPase pump. Beta, this pump, it is having the ATP. So it is against the concentration gradient, it will move. Right? It is a pump, so it will use the ATP to move the things against its concentration gradient. So already outside, you know that sodium is more, potassium is less. And inside, potassium is more and sodium is less. But still, what's going to happen here? What is going to happen here? Dekho, Na out. Matlab no kiya, Na out. Sodium will go out. Matlab sodium is already out more, but still from inside it is going out. So 3 sodium ions will go outside and 2 potassium ion will move inside. That is the function of this pump. That is the function of this pump. So basically what is happening? Basically in this membrane, neuron, we are saying differential permeability for sodium and potassium ion is there. What are we saying? That there is the differential permeability for the sodium as well as for the potassium ion. What is there, people? There is the differential permeability for the sodium as well as for the potassium ion. Number one, we are saying because there are more leaky channels for the potassium, that is why more and more potassium is going outside, because these leaky channels are working as per the concentration gradient. Inside there is more potassium, outside there is more sodium. Now sodium leaky channels are comparatively less in comparison to potassium leaky channels, so less sodium will enter within the cell and more potassium will leave. Number one. Number two is sodium potassium ATPase pump, right? So when you talk about the sodium potassium ATPase pump, so here you say that 3 sodiums are moving outside, 2 potassiums are coming in. 3 sodium are moving outside, 2 potassium are coming in. right? That is also helping in maintaining this polarized state of the neuron. Inside is negative, outside is positive, because more and more positive charges going outside. And inside, as I said, high negatively charged organic proteins are there, which are having high molecular weight. Negatively charged proteins are there, which are having high molecular weight, right, which are having high molecular weight, right? So they will stay inside. So that is why outside more positive, in comparison to that inside there will be less positive. So that is the difference. So in this polarized state, this is the, right, it is somewhere in between -50 to -70, but that is how you can mark, right? That is how you can mark. So this part is clear, this part is clear, this part is clear. So one more line, just I told you about the differential permeability. So again writing it here, when a neuron is in its resting state, it is more permeable, right? This is the property of the neuron, to potassium than to sodium. That is something you have to remember, okay? That is something you have to remember. Important it is, right? Direct PYQ it is. Take important it is. Direct PYQ it is. Is that clear? Is that clear? So what is it? It is important and it is a direct previous year question. ठीक है? ठीक है? Read it from there. All these, the membrane is impermeable to negatively charged. Consequently, the exoplasm inside will have high potassium, negatively charged protein, low sodium. So fluid outside the axon will have low potassium, more sodium, right? So concentration gradient is there. So now active transport is also there. I told you 3 sodium will go outside, 2 will come in. So as the result, outer surface of the axonal membrane is having the positive charge, while its inner surface will become negative. It is polarized. So the electrical potential difference across the resting plasma membrane is basically its resting potential. I told you it is -70 MV. So whatever is written in NCERT, I have discussed that. Now, when a substimulus is applied at a site, let's suppose this is the point A. Now this is important. Okay, now, so basically I told you who's going to maintain the negative state. Now we will have to discuss something known as depolarization. Okay, now we have to discuss something here which is known as what? Which is known as polarization. Okay, which is known as polarization. Okay, so directly I'll cater all the questions. Don't worry about that. Don't worry about that. Just ye dekho. This is the site, right, at which stimulus is coming. Of course, here you have to remember the terms like threshold stimulus, right? Matlab it's not like that ki any stimulus can excite the neuron. We need that minimum stimulus. It should be above that minimum value. So how that nerve impulse, how the action potential will generate? This is a direct PYQ. It should be the influx of sodium ion, right? When there will be the influx of sodium ion, when sodium ion will enter. Just say, let's suppose this is the point at which stimulus is coming. At which stimulus is coming. So what is going to happen here, bache? What is going to happen here? Yes, there is a disturbance here now. So there is a stimulus. So because of that stimulus, there is the leakage of sodium channel inside. Matlab sodium will start moving in, right? Sodium will start moving in. Are you getting my point? Sodium will start moving in. Are you getting my point? So when sodium will start moving in, don't you think it will lower down the potential difference here? It will lower down the potential difference here. Yes or no? Yes or no? Right? Sodium leakage is there, right, down the electron gradient into the nerve fiber. So don't you think that it will lower down the potential difference here? Because at the point of the stimulus, what is happening? More and more sodium is coming inside. So inside is becoming positive. Here at this point only, outside is becoming negative. So basically there is the lowering down of the potential difference. There is the lowering down of the potential difference. Yes or no? Yes or no? Yes or no? So because this transmembrane potential difference, it is decreasing. So now, right, lowering down of potential difference, what will it cause? It will increase the membrane. Matlab this is the way this neuron membrane behave. It will increase the membrane's permeability. It will increase the membrane's permeability for sodium ion. Matlab, now membrane is more permeable to sodium ion. More and more sodium ion will come inside. More and more Quenisha sodium ion will come inside. More and more sodium ion will come inside. Are you getting my point? Are you getting my point? More and more sodium ion is coming inside. So because here potential difference is getting lower, more and more sodium ion is coming inside. So the membrane's permeability for sodium ion will increase, right? And this is the special property of the neuron membrane which is differentiating it from any other ordinary membrane. This is the special property, right, of this neuron membrane. Is that clear? Now when this kind of change will happen, then voltage gated sodium channel will open, of course, right? They will open, and because of this opening, more and more sodium will come inside. Inside will become positive, outside will become negative. Now the way current moves, plus to minus, ye dekho, at this point, right, this point the action potential has generated somewhere around plus 20 MV. Now this will move, right? This will move from this point to this point. So this point is still negative, this is positive, you know that the current will move, right? So this stimulus will move from A to B. So here the point is, here the catch is that no doubt I told you that when there is a stimulus, there is the influx, right? When there is a stimulus, there is the influx of sodium within. There is the influx of sodium within. Without any doubt, this is the point, right? Without any doubt, it is the point. Right, it is the point here. Now what is the point here? What is the point here? Now look at this. This side is having inside positive, outside negative. You know na, when we start doing something, when we put all our efforts, so internally we start feeling positive, we start feeling this na, we start getting this emotion na, we start feeling positive now, that wow, the things are good now. So this state is basically the action potential. But when you are in action, when you are doing something, to change your situation now, you start feeling positive inside. So this is the depolarization of membrane. This is the depolarization of membrane, right? This is the action potential. Are you getting my point? Are you getting my point? Right? So because there is the decrease in the potential difference, membrane's permeability for sodium ion will change. So what is going to happen? What is going to happen? The voltage gated ion channels, they will open. And you know, these channels, they can open and close by itself. It depends upon the potential difference, right? It depends upon what? It depends upon what? It depends upon the potential difference. So when there is the depolarization, voltage gated sodium channels are open, but voltage gated potassium channels, they are closed. I'll make a table as well. It will be closed only, right? That's why more positive inside. Now the charge will move, right? Plus will go here, this plus will go here. So basically the funda here is, right, no doubt this membrane, neuron's membrane will change its permeability. It will be more permeable for sodium in comparison to potassium when there is the potential difference decrease. But this situation is just for few seconds. Are you getting my point? Are you getting my point? This situation is just for few seconds. This reversal of polarity is just for few seconds. Matlab it is less than for 1 by 1000 seconds. Imagine, right? Because after this situation, the membrane will again change its permeability. Membrane will say, no, I am loyal to the potassium only. How can I allow the sodium to come inside? I have to change the thing. The membrane is like, again. Membrane will be like, no, I'll change the permeability. Potassium should come in. Potassium, right? The potassium movement should be there. And because of that, the repolarization will occur. Okay, right? So within a fraction of second, potassium will start diffusing outside the membrane and it has to restore the resting potential. So that is how the repolarization will occur. So the ion channels, right, that are going to play the role here, I will write it. So firstly, polarization is the situation, then depolarization, when the neuron is having, you know, that neuron is, when neuron will get excited, when the action potential is generated, and then there is repolarization, right? Again, it has to come back to the polarized state. Okay, ji? And of course, the hyperpolarization means when excess of positive charge is outside the neuron, right? So we have to mention the ion channels also. So let's suppose leaky channels, potassium leaky, sodium leaky, voltage gated, nahi, sodium potassium ATP pup, Now my screen is like, I cannot work more. Then there is the sodium voltage gated channels, potassium voltage gated channels, right? Right? So this is what we have. So of course, when you are talking about the leaky channels, right? When you are talking about what? Leaky channels. Matlab, it is basically what? It is basically the kind of passive diffusion. It will be there in all the possible conditions. Leaky channels means what? Leaky channels means I am talking about the passive movement. I am talking about the passive diffusion of the ions. This is what I'm talking. This is what I'm talking. I am talking about the passive diffusion here. Then sodium potassium ATPase pump. So when you people talk about the pump, it will only be closed. It is always open because it has to maintain inside negative, outside positive. It will only be closed when there is the depolarization. At the time of depolarization, because you know that voltage gated sodium channels are open. Here it is closed. Here it is closed. Here it is closed. Okay. Okay. Right, so this is the situation. This is the situation, right? Yes or no? Right? जैसे ये देखो, this passive diffusion, it will just simply continue. It will be like this only. It will just simply continue. It will like this only. Sodium potassium ATPase pump. It will always be open. It will only be closed when there is the depolarization. At the time of depolarization only, beta, it is closed. Otherwise, it is always open. Now, sodium voltage gated ion channels. They will only open when there is an influx, when the depolarization is required. And when will the sodium voltage gated ion channel will open? When there is the repolarization and even in the state of hyperpolarization as well. So if you will remember this part, it will be more than sufficient for you. So repolarization, polarization, right? So this depolarization clear somewhere in between plus 30 MB to plus 45. So you can even mention plus 20 also, sometimes plus 20 also. ठीक है? So here depolarization में inside is positive, outside is negative. Here also inside is negative, outside positive. Repolarization में भी inside negative, outside positive. Okay, here also inside very negative. Okay, that's the situation. This is all about this topic. You read it from the NCERT, trust me, it will not at all be difficult. So is it clear to you? If it is clear to you, do let me know in the chat section. If it is clear to you, do let me know in the chat section. Quick. Students, you can even be the part of my Telegram group, Ambika Sharma Biology. You can join our official Telegram group as well. You will get the PDF. But before that, do let me know, are you able to understand the things or not? Are you able to understand the things or not? It's not a difficult topic, isn't it? It's literally not a difficult topic. And what are you doing? Why the number of likes they are less? Chalo, hit the like button as well. Quickly, hit the like button as well. Quick. Quick. And yes, these are the amazing sessions for all of you. And ये देखो, जैसे short sessions where we will revise everything. ठीक है जी? These are the short sessions where we have revised what? This is the strategy. This is the most important chapters. Okay, now my screen is like, nahi, I cannot work. है ना? My screen is like, I cannot work now. I'm done. ये देखो. So in the video section, you have these short videos, जैसे plant growth and development is there. This is the wonderful block revision strategy for all of you. This is a cell cycle and cell division. The next chapter that I'm going to post is photosynthesis, right? The next chapter that I am going to post is photosynthesis. So these are the short sessions for all of you, which you people can use for the quick revision. ठीक है? बाकी here बच्चे synapse, I told you synaptic cleft also, right? So you know that we have two things. Here two type of synapses there, electrical and chemical. So electrical में membranes of pre and post, they are continuous. मतलब it's just like a gap junction. It is in the proximity. And electrical synapses here, the current can flow directly from one neuron to the another. So transmission of impulse across the electrical synapse is very similar to impulse conduction along a single axon. Okay? So it is very fast, it is rare in our system. And when it is the chemical synapse, what is going to happen? The neurotransmitters, right? Ultimately, these are the neurotransmitters that will be released here. So I'll explain you the phenomena directly here. So of course, when the action potential will reach here, right, at the end. But what is going to happen? Dekho, action potential when it reaches the presynaptic terminal, right, the depolarization will, right, depolarization is there. Because of that, ion channels will open. Which ion channels? Calcium ion channels. Because of that, more and more calcium ion will come inside. Right now, when this calcium ion will come inside, right here at the ends, these neurotransmitters, they'll fuse with this presynaptic membrane, right? These synaptic vesicles, rather, they will fuse with this presynaptic membrane. The neurotransmitters will be released here in this cleft. Right, when these neurotransmitters, they'll be released, they will bind to the receptors to the postsynaptic neuron. They have the ion channels here. These neurotransmitters will bind here. So when these neurotransmitters will bind here, the opening and closing of channels will occur, and the action potential will pass to the next neuron, bache, right? It will pass to the next neuron, and neurotransmitter which are present there, they'll be inactivated. Actually, if I have to give you the example, imagine if here the neurotransmitter is acetylcholine. Take, right? So it is an excitatory neurotransmitter. So this here we have the enzyme also, just acetylcholine esterase. So this enzyme is going to break this acetylcholine after some time, and then it will be, right, it will be taken back by this neuron. Matlab basically it will be transported back into the presynaptic neuron. So that is how it works. So those people which are which are clinically, which are getting treated for the clinical depression, in their case also, doctors, what they give the chemicals, they give the chemicals, the happy hormones to them. This is one example is Prozac. So they'll make sure that these, right, these happy hormones, they should not get destroyed here very quickly, right? So that, they can just lift up the mood. So that is how the things are. Okay, that is how the things are. Okay. So that's it. That's all. Again, these are the kind of changes that will occur in the next neuron, and I have added many diagrams for that. Ye dekho, neurotransmitter will bind there and will start the expression further. Okay, and these are the different types of leaky channels, leakage channels, voltage gated ion channels, ligand gated ion channels, signal gated ion channels are there, right? So you know that their closing and opening, it depends upon the change in the voltage, right? In change in the voltage. Chaliye baki CNS dekho. Again, it is memory based. I'll tell you about the trick. Forebrain. In the brain, we discuss three parts. We have forebrain, we have midbrain, and we have hindbrain. Forebrain is there, midbrain is there, hindbrain is there. See, in the forebrain, the trick is, in the forebrain, there will be cash on delivery. Forebrain mein hui cash on delivery. In the forebrain, what is there? In the forebrain, what is there? In the forebrain, there is the cash on delivery. There is the cash on delivery. So in cash on delivery, what will you get? You will get cerebral hemisphere, or you can call it as cerebrum. Cerebrum. O stands for olfactory lobes. D stands for diencephalon. So in the forebrain, there will be the cash on delivery. What will we get in cash on delivery? The ion. T-H-E. So T stands for thalamus. H stands for hypothalamus. E stands for epithalamus. These are the three parts here. So in the forebrain, there is the cash on delivery. What we got in the cash on delivery? The ion. Matlab, diencephalon is having three parts. Now, in the hindbrain. Now our hindbrain is like, I want to read physics, chemistry and maths. So P stands for this pons viruli. The C stands for cerebellum, also known as the little cerebrum. And this M stands for medulla oblongata. This M stands for medulla oblongata. Are you getting my point? This M stands for medulla oblongata. Are you getting my point? Right. See, those who are saying that we are feeling sleepy, even I am feeling sleepy, but the point is, we have wasted our entire year. Now, we have to wake up for these last 20 to 35 minutes. Why? 20 to 25 days. Why? Because we have to utilize this time. And even if when you have studied for the complete year, na, this last 1 month is very important. Last 1 to 2 months, they are very, very, very important. Always they are very, very, very important. Why they are very important, bache? Because in this time, if you will not study sincerely, if you don't revise sincerely, if you don't prioritize the topic, prioritize your studies, na, anything can happen. It doesn't matter how well you were doing before these March and April month. But even in March and April month, if you will get sincere, if you start revising sincerely, trust me, you even before that, if you were scoring 400, you can directly jump to 600 plus marks. That is the importance of this time. Trust me, that is the importance of this time. Okay. So understand that thing. Theek? Understand that thing. Okay. Chaliye, let's go. So midbrain, C2, CQ. What is the trick for midbrain? For midbrain, it's simple. It's C2, CQ. C2. Crura cerebri. And CQ is corpora quadrigemina. Corpora quadrigemina. Right? So C2, CQ is the point here. Crura cerebri, corpora quadrigemina. So it is crura, cruel uncle. Cruel uncle. So we also call it as cerebral peduncle. Cerebral peduncle. Cerebral peduncle. So these are the parts of the brain. Are you getting my point? These are the parts of the brain, and this is a very simple trick to remember that. So in the forebrain, what will be there? Cash on delivery. What you got in cash on delivery? Third eye. Okay. For the midbrain, what do we have? C2, CQ, and C2 is our cruel uncle. So basically, it is cerebral peduncle. Cruel uncle, because it is crura cerebri. Hindbrain is having what? Hindbrain is having physics, chemistry, and maths. Right? It is having physics, chemistry, and maths. Okay. Okay. Right. So this is what we have. Right. So now you have the central nervous system. You have the brain part. You have the right. It will be there in the who's going to protect it? Cranium. What is cranium? It is the brain box. It is the brain box. And let me tell you, it is made by the fusion of bones. Do you know how many bones are there? Do you know how many bones are there? Cranium. Remember? Frontal, parietal, temporal, occipital, ethmoid, sphenoid. Frontal, parietal, occipital, ethmoid, sphenoid. Total 8. Seriously, are you sure? Are you sure? How come 8? Acha dekho, frontal, parietal, temporal, occipital, ethmoid, sphenoid. Frontal 1, parietal 2, temporal 2, occipital 1, ethmoid 1, sphenoid 1. 1, 2, 3, 4, 5, 6, 7, 8. Are you sure? Are you sure? I'm talking about the cranium only. I'm talking about the cranium only. Are you sure? Are you sure? Dekha, we are also revising the locomotion and movement. We are also revising the locomotion and movement. Right? Right. So frontal, parietal, temporal, occipital, ethmoid, sphenoid. Frontal 1, parietal 2, occipital 1, temporal 2, ethmoid 1, sphenoid 1. Take. Okay, chaliye, let's go. Now, the next question can come from the meninges. These are the protective coverings that are covering our brain and the spinal cord. So for the meninges, what is the trick? It is DAP. For the meninges, what is the trick? It is DAP. What is the trick? It is DAP. Dura mater outermost, then it is arachnoid, then it is pia mater. Okay? So outermost is dura mater, a very thin, middle layer is arachnoid, inner layer, right, which is in contact with the brain tissue is the pia mater. Is that clear? So the name of the meninges, right, like the sequence of meninges, is it important? Yes, ma'am. It is very important. It is very important. So look at these meninges here, right? Look at this meninges here. Right, so ye dekho, dura mater, right? Can you see that dura mater? Then this is the space in between. This is the space in between. This is the arachnoid here, and this is the pyometer here. Take. So this is subarachnoid space also. Firstly, have a look, then I'll explain everything here. Right? Matlab, this is the section that you can clearly see. Or ye dekho, this is the spinal cord part. Right? Here also they are showing the same. They are showing the intervertebral disc, and they are also showing this layer. So this is the durameter, bache. Then arachnoid is there, then pyometer. So in between this arachnoid and pyometer, you have subarachnoid space. That space is having CNS, a CSF, cerebrospinal fluid. This is important. So be it spinal cord or it is brain, the sequence is DAP only. The sequence is DAP only. The sequence is DAP only. Durameter, in between durameter and arachnoid also there is a gap. Right, subdural space is there, but that is not having the cerebrospinal fluid. But this arachnoid space and pyometer, here in between these two, you will have subarachnoid space with central nervous, with the cerebrospinal fluid. Understood? Understood? Pakka? Pakka? So skull, durameter, and all this point. Clear? Clear. Dekho, there are so many diagrams. Right? There are so many diagrams. Okay? Cheek. So subarachnoid space is having the cerebrospinal fluid. It is having what? It is having the cerebrospinal fluid. Not given in NCERT, but it is important. So forebrain and its functions. Right now, here for the brain, because see, it's a memory based topic. So I'll say go for all the PYQs. Go for all the PYQs that you have ever seen, in your, like which were there in the NEET examination, right? Just go for what? Just go for the, right people, just go for what? Just go for the PYQs of this part. Everything will be crystal clear. Just dekho, two hemispheres are there, outermost is cerebral cortex. So how are they connected? They are connected by the track of nerve fiber. How are they connected, beta? They are connected by the track of nerve fiber. How are they connected? Yes, please. How are they connected? They are connected by the track of nerve fiber. So this type of language, this type of terminology, right? What is track? What is this? What is that? It should be clear to you all. Okay? Just for an example, if it is the collection of, right? If it is the collection of cyton, if it is the collection of a cell body, let me use this language here. If it is the collection of cyton or cell body, means so many cyton or cell body together. So we have two terms here. One is nuclea, another is what? Another is ganglia. Another is what? Another is ganglia. So ganglia is the term which we use for PNS. So of course, nuclei is the term that we use for CNS. Is that clear? So when there is the collection of nerve fibers, now this is for the cyton, and when there is a collection of nerve fiber, you say you have neuron, when it is having some sheaths, right? You will call it as nerve, right? The group of nerves will form nerve fiber. It is like that. Okay? So here you have two words. One is track. One is track. Matlab that the word, the corpus callosum, right? Just now we have discussed it. Track is the word. So track is the word that we use for CNS, and then the other word is nerve that we use for PNS. So nerve fibers, so many nerve fibers together will form nerve. So many neurons, they are covered with a sheath, they'll form nerve fibers. So many nerve fibers together will form, right? They will together form nerve. So this language should be clear. So here we are saying we have the corpus callosum. Here, what do we say, bache? Here, what do we say? What is present? Corpus callosum is there. Corpus callosum, track of nerve fibers. Okay, bache? Okay. Next thing, the cerebral cortex is having motor area, sensory area, large region, which are not even sensory, not even motor. Association areas are there. They are having the role in, memory and communication. So what is the important thing here that you have to remember? Right, ye dekho. Fibers of track which are covered with myelin sheath. When myelin sheath is present, right, that is in the inner cerebral hemisphere, it will give white appearance. That will form white matter. So in the brain, outside cerebral cortex, gray matter, inside it will be white matter because it is having the medullary sheath. Now, hypothalamus, very famous question, beta, it is the thermoregulator. It has multiple centers, and what we have to call it? We have to call it as thermoregulator, right? Thermoregulator. The diagram you need to master. right, whatever is there in the diagram, if they are having just cerebral aqueduct, so if they are having any other name, focus there. So baki hypothalamus, it is the thermostat associated with temperature. Urge for eating is controlled by that, and drinking also, right, so it is having neurosecretory cells that will form neurosecretory hormones that we will discuss in chemical control and coordination. Okay beta. And now cerebral hemisphere and amygdala, hippocampus, they form. Hippocampus is associated with your memory. No hippocampus, no memory will be there, right? It is forming the limbic lobe. Along with the hypothalamus, they are regulating sexual behavior, expression of emotional reactions. This part from this side is important, right? This is directly important. So it is totally memory based, and here you please solve MCQs, right? As much as you can. You should solve, you have to solve the MCQs here as much as you can. Is that clear? Okay, then midbrain hai bache. In the midbrain, it will basically connect what? Hypothalamus of forebrain. So this line can be asked as a direct question, and pons of hindbrain, right? So it is having a canal. The canal is cerebral aqueduct that will pass through the midbrain, and it is having the corpora quadrigemina. Quadrigemina is the word, quad word is associated with 4, right? So what is the word here? Cerebral aqueduct. It is the canal that is passing from the midbrain. Check. Okay. Sure. So this canal is also known as aqueduct of Sylvius. This is the another name of this, aqueduct of Sylvius. Okay, aqueduct of Sylvius. Is that clear? Is that clear? And cerebrospinal fluid, please do not forget that cerebrospinal fluid provide the buoyancy. Brain is, you know, in cages, but we don't feel it like this. Why? Because it is in cerebrospinal fluid. So that is not I'm discussing. I'll stick to the NCERT because ultimately paper will come from this part only, and we have too much to remember. We have to stick to this. So then here pons, cerebellum and medulla is there, right? So pons will have fiber tracts that interconnect different regions of brain, and cerebellum is also having infolding. It is also known as the little brain. Do you know when somebody is drunk, right, then they are not able to maintain the balance because alcohol, it hit this part and the balance will be disturbed. Okay, so medulla is connected to the spinal cord. So medulla will have many center, respiration, cardiovascular reflexes and all, and 3 major regions will make the brainstem. So this can come in your NEET 2025 as well. This question can come in your NEET 2025 as well. So brainstem mein you have midbrain, you have pons region and medulla. Take. So brainstem is the connection in between brain and spinal cord. It is the connection in between brain and spinal cord. Understood? Baki dekho, different types of nerves are there. Indeed. Okay, so that is also the part of your syllabus, cranial nerves, spinal nerves. Okay, they are literally the part of your syllabus as well, right? I do understand. Okay, I do understand this thing ki, if you are studying this for the first time, you will not be able to do that, and I'm not forcing you to do that. So cranial nerves, they are 12 pairs. At least remember these things. So there is a trick here. Upata fagvasha. Upata, or it can be utata. Utata ya upta fagvasha, right? Fagvasha. This is the trick here. Utata or upta fagvasha. Utata or upta fagvasha is the trick here. That is basically for what? For the cranial nerves, right? So all up ak. Utata fagvasha, then it is all up ak. Olfactory, optic, oculomotor. So this is pathetic or trochlear, right? Why I'm saying utata ya upta? Because this is having 2 names. Okay, then trigeminal, abducens, facial, right? Then vestibular, then uta fagva, facial, it is auditory. So this is also having another name. This is having the name auditory. Then this is glossopharyngeal. Then vagus is there. Spinal accessory is there. Hypoglossal is there, right? This is the spinal accessory. Hypoglossal is there. Check a hypoglossal is there. These are the cranial nerves, right? They have, you know, different functions. That is how they are originating from the brain. And now in the brain also, these are the cavity which are known as ventricle. Just say first, second, they'll join. This is the third one. Then from the midbrain, cerebral aqueduct is there, which will connect the third and the fourth ventricle, which is there in that hindbrain part. So that's it. Okay, That's it. So this is just the trick. The function is also given. I'll share the PDF in the Telegram group. Right? We'll share it. You will get to know. So one trick is utata या उपटा मतलब P or T, anything you can write. Utata phagwasha, utata phagwasha, right? This is for the cranial nerves. So all up, olfactory, optic, oculomotor, pathetic or trochanterian, right? Then it is the trigeminal, right? Then it is the abducens, facial, auditory, glossopharyngeal, vagus, spinal accessory, and hypoglossal. So for that, there is one more trick, that is, some say marry money, but my brother says big brains matters most. Some say marry money, but my brother says big brain matters the most. So even if you can remember it like Upta Phagwasha, your choice. Otherwise, only one of the two athlete felt very good. Nahi bhai, go for this. Uttata Phagwasha. Take. So some say marry money, but my brother says big brains matter the most. So some S, sensory. Again, sensory. M, motor. M, motor. Wherever you have the B. B means both. Both. They are sensory and motor both. Then again M, motor. Then again brother, matlab both. Facial is both. S, sensory. Both. Motor and motor. Right? So this type of question can come ki how many pairs are sensory and this and that. Baki spinal nerves. You know this thing, right? So they are what? Bas. Baki reflex section is something very simple. Now, bache, you can revise it, stick to the NCERT, that's it. Now, the next chapter is this. Chemical control and coordination that we are going to touch. Is that okay? And here, what is the most important thing? This hormonal part, this mechanism of hormone action. Right? This mechanism of hormone action is very important. And in that top 50 concept series, I have discussed that. Bache, dekho, at when we started at 1:30, right? Not at 1:30, 1:40. Right now, after 1 hour, if you will expect the break, no chance. I'm sorry, I cannot. Literally, I cannot. Right? If you're tired, you can end the class. I'm not tired. I'm so used to it. This is my favorite job, to take the classes, right? To not to sleep, to get the dark circles. Hai na? I am compromising my beauty sleep because I don't have any other work to do. I just have one work. I need to take the classes only, right? And trust me, I will not get more than 2 hours to sleep. Okay, I have another work. I have classes also, right? I have the classes in my batch also. It's not there in NTA syllabus. Sanjay, nerves are given. Peripheral part is given. They are having this nerves. ठीक है ना madam, you are tired. नहीं madam, we are tired. You are not tired. You are used to it. You compromise your beauty. You compromise your sleep. You know now, if we don't sleep on time, we get the dark circles and all, right? And I'll get the migraine issues. I'll get the like severe headache. And if I don't sleep properly, especially on time, right? Then I'm the most irritating person that you can ever talk to. Trust me on that. Trust me. Chalo, we can have one washroom break, hai na? We can have one washroom break. So that will be just for 2 minutes, right? I'll go to the washroom, I'll come. You can also go to the washroom, then you can come. So then let's target this part. So we have to finish this locomotion and movement, the skeletal part. Right, so let's target it. And if you are thinking, madam, what about the remaining 2 chapters? If you will support, I can even take the class till 7. I don't have any problem. It's all up to you. It's all up to you. Right? So if you will support, I can even take the class for 7 up to 7 a.m. or 8 a.m. It's totally up to you. And if you will support me till 8 a.m., trust me, we can finish everything so smoothly. Right? And whatever is left, you know that in botany, we don't have much part. Living word, it's a matter of 1 hour. Biological classification, 2 hours. Plant kingdom will take 3 to 3.5 hours. That is important. Then anatomy, very quick. Just need to go through the sections only. Very quick. Morphology, directly diagrams we have, examples we have to revise. Families, again, in the recorded format, you people will get. And I will not suggest you to do the families if you don't know about the syllabus. Baki, plant physiology, plant growth regulator, matter of 30 minutes. Okay? Growth regulators, 30 minutes for remaining part. 1 hour for that chapter, 2 hours for photosynthesis. So botany part is very less. So if required, I'll discuss the biomolecules there. I'll discuss the cell cycle there. Okay? So if you allow me, I can take the class till 7 a.m. I'm okay with that. I don't have any problem with that. Okay? So chalo, I'll be back in a minute. Hanji, let's start. Drink water. Drink as much as you can. Very important it is. Done. So how's the energy, people? How's the energy? So how's the energy? Hi, nahi, it's not easy, है ना? Need a lecture on morphology. Panduji, we'll take it. Hi. So I just asked how's the energy. Many people they are like, nahi. We are just running away. Chalo. So hormone action. Dekho. Hormones, here what are we discussing? We are talking about the endocrine gland. So when we are saying endocrine gland, when I was giving you the examples of structural organization part, I have discussed that with you all. So endocrine glands, bache, they are the ductless glands. What are they going to do? They will directly pour their secretion into the blood. Now from that blood, how these hormones, because these hormones are what? They are the chemical messengers. These hormones are what? They are the chemical messengers. They will be synthesized in what? They will be synthesized in very less quantity. Right, bache? right bache? Chalo, let's start with that basic introduction only. So human endocrine system, and this is the, so you know that ultimately your neural system, your this chemical coordination, it is required to carry out the body function in a coordinated manner. So neural coordination is fast, but it is short lived, right. The nerves, they do not innervate all the cells of the body and cellular functions, and it is true, keep it in your mind. Nerve fibers, right, they do not innervate, nerve fibers, they do not innervate all the cells of the body, and the cellular functions need to be continuously regulated, right. So that's why this coordination is required. So function is carried out by hormones, and neural system and the endocrine system, they jointly coordinate and regulate the physiological functions in the body. Okay? So these hormones, they are non-nutrient. They are not something which are nutritious for us. They are non-nutrients. What are they? They are non-nutrient chemicals, bache, which act as intercellular messenger in between two cells. It is important. So if in this particular statement, instead of inter, if they will add the word intra, then it will become incorrect. Okay? So keep it in your mind. So it is the intercellular messenger, and the products, and they are produced in trace amounts, very less amounts. Okay. So hormones, they are secreted by organized endocrine glands. In vertebrates, they are also having simple endocrine system, and there are, whereas large number of chemicals act as hormone, they'll provide coordination in vertebrates. So this is what we discussed. Let's suppose here in the brain part, pituitary, master gland, hypothalamus, supermaster gland, pineal gland is also there, that will maintain the circadian rhythm, our biological clock. Thyroid and parathyroid gland, here you have the thymus, the one which will decrease, right, whose size will decrease when we, increase in age. Okay, ji? So this is the pancreas, which is known as heterocrine, also known as composite gland, right. It is the mixed gland because it is having the exocrine and endocrine part both. You have the adrenal gland, the cap of the kidney here. Then gonads are there, testes and ovary. This is what we discussed. So that intercellular messenger thing is very important. Now you know that they are the ductless gland. They will directly pour their secretion. Where will they pour their secretion? They will directly pour their secretion into the blood. So from the blood, this secretion, from the blood, where the secretion will pass, it has to act on its target organ, right. Then how will it get to know? Because bache, these hormones, they are also very specific. Why? Because they have the receptors, right, to which they have to bind, right. They have the receptors to which they have to bind, right, and accordingly, you know that the responses will be initiated, right. In the top 50 concept series also, I have discussed this topic with you all. So hormone and receptors, they are present on the cell membrane of the target cell. So if they are present on the membrane, they are membrane bound receptors, but it's not mandatory that all the receptors are present on membrane only. Some are inside the target cell. They are the intracellular receptors, nuclear receptors there. So these hormones are also referred to as the first messenger, my dear people. They need to bind with the receptor. They have to form the complex here. So of course they are specific, and then the response will be generated. So when you talk about the nature of hormones, there are different types. Do not forget the NCERT. जैसे ये देखो, steroids are what? They are the type of lipids only. जैसे sex hormones, all the sex hormones are steroidal in nature. The adrenal cortex के hormone जैसे cortisol है, right? So they are what? They are also steroidal in nature. Your iodothyronins, they are having iodine. You're talking about the hormones of thyroid gland. Then amino acid derivative, epinephrine, norepinephrine, right? These examples are important about their nature. They can ask you. So you remember adrenaline, noradrenaline, adrenal medulla releases that, right? Adrenal medulla is going to release that. Are you getting my point? Adrenal medulla is going to release that. Are you getting my point? Are you getting my point? So now peptide, polypeptide, yeah, protein hormones are your insulin, glucagon, pituitary hormone, hypothalamic hormones are there, right? So now here we have the receptors present. Now what's going to happen, bache? Let's see. Let's look at this diagram. There are certain there are certain hormones, right, whose receptors are present on the cell surface only. Why is it so? What is the requirement of that? Because these hormones, right, they cannot cross the plasma membrane. It's not mandatory that all the hormones can cross the plasma membrane. It is a protein hormone. It cannot cross the plasma membrane. So its receptor is present on the cell surface. So of course they are specific. Just say here you are taking the example of FSH. What is this FSH? It is the follicle stimulating hormone. What is this FSH? It is the follicle stimulating hormone. What is it, bache? It is the follicle stimulating hormone. So because it is the follicle stimulating hormone, my dear people, so what's going to happen? It has to act on the, right, you know that in the male body also, in the female body, it is having the role, right? So in the case of male body, it will be FSH, S, Sertoli, it will act on Sertoli cell. In the case of female body, where will it act? On the ovaries. It has to act on the follicles. It will help in the proliferation of the follicles. So how will it act? So basically its receptor is present on the cell surface, on the ovarian cell membrane. Now because this hormone cannot enter in the cell, its receptor is there on the cell surface, so it has to initiate the response only. So when these type of hormones whose receptors are all there on the plasma membrane, in their case within the cell, the second messenger will form, the second messenger, because they cannot cross this plasma membrane, so their response has to pass. So this can be asked as a question, the example of, right, second messenger. So you have the calcium ion, IP3. This is inositol triphosphate. Inositol triphosphate is there. Take IP3. Cyclic AMP is there. Cyclic GMP is there. It is adenosine monophosphate, guanosine monophosphate, and one is DAG. What is this DAG? This is diacyl glycerol. What is this DAG, my dear people? This DAG is diacyl glycerol. Diacyl glycerol. Are you getting my point? These are the second messengers. So of course they will generate the biochemical response, the physiological response, the ovarian growth will be there. So these type of second messenger, they will form only when the hormone is attached to the cell surface receptor. Now when the hormones are steroidal in nature, examples are important. They are steroidal in nature. What are they? They are steroidal in nature. They can directly cross the plasma membrane. So their receptors, so here there is no second messenger. There is no second messenger when the receptors are present within the cell. So receptors are within the nucleus. You know that the genome mRNA, the physiological responses will be generated. That's how you have to remember this. So one question from mechanism of hormone action can come, and that is how we have to revise it. Okay. Baki bacha, here the gonadotropins and all, you know that we discuss it with what? We discuss it in the human reproduction chapter, so don't worry about that. Now come to the next part. Let's revise the human skeleton. What say? Let's revise the human skeleton, and then let's revise the sliding filament theory. Firstly, let's revise the human skeleton because it will hardly take 15 minutes, right? It will hardly take 15 minutes. So let's revise it. So are you ready for the human skeleton part? Are you ready for the human skeleton part? Quick bache, are you ready for that? Because in the top 50 session, I have all discussed the sliding filament theory, and I took the decent amount of time to explain it. So let's revise, right? And on this channel separately for the revision of pituitary gland, adrenal gland videos are there, and I will post more, right? Matlab, we have just started. It's not like that now my marathon, next marathon will be on Friday. So till Friday, madam will be, you know, will do nothing. Nahi, I'll post the videos. Don't worry about that, right? Beta class, dekho, if you will allow me the time till what? If you will allow me the time till 7, then we can finish all the chapters. If you are like, nahi madam, 5 is sufficient, right? 4 is sufficient, then of course few chapters we have to complete in the next marathon, right? I'll make it very clear. If you will allow me time till 7 or 8 a.m., then to each and every chapter, and that too in detail, we will cover. So it's totally up to you, right? Then later on nobody will write, array, what about the remaining chapters? Then revise it, right? The session, the videos are available on the Unacademy NEET English channel, then revise it. So it's all up to you, right? It's all up to you. Now let's talk about what? Let's talk about the skeleton, right? Let's talk about the skeleton. 1 to 2 questions can come from a skeleton part as well this time. Now in the case of adult, when you are talking about the human skeletal system, of course we know it's bony. How many bones are there? 206 bones are there, right? Matlab bones are there, cartilages, right? You know, right? They are the part of the skeleton. Mainly we are talking about the bones here. So here we have what? We have the axial skeleton, right? Imagine a doll, right? Imagine a doll, the Barbie doll. You're removing the arms and the legs. That remaining part is the axial skeleton. And other than that, what do you have? You have the appendicular skeleton. So when you talk about this appendicular skeleton, it is having 126 bones, my dear people. And when you are talking about this axial skeleton, it is having how many bones? It is having 80 bones, right? It is having 80 bones. Are you getting my point? How many bones do we have here? How many bones do we have here? How many bones do we have here? We have the 80 bones. And trust me, it is easy. We can revise it very quickly. 1 second. ठीक? Okay. ठीक है? So in the case of axial skeleton, what are you going to discuss here? Simple. बच्चे, here when you talk about this axis, मतलब this skull part, this cranium, facial bones, ear ossicles, the vertebral column, sternum, ribs, that's what we have to discuss here, right? That is what we have to discuss here. Now when you talk about the skull, in the skull we will discuss about the cranium. You know that I mentioned 29 bones. I'll tell you the reason as well. Cranium, 8 bones are there. Your face bones, that is your facial bones, they are total 14 in number. Now here, ear ossicles, the bones which are present in the middle ear, they are total 6 in number. They are what? They are total 6 in number. And there is one more bone, that is hyoid bone. Now when you talk about this hyoid bone, it is a U-shaped bone. It is 1 in number. It is the U-shaped bone, bache. It is the U-shaped bone. It is a tongue bone, and it is let me write it here. So it is what? It is the U-shaped bone, also known as horseshoe, also known as horseshoe-shaped bone. It is. What is it? Horseshoe-shaped bone it is. Right, bache? So it is situated in the midline of neck, anteriorly at the base of mandible. Mandible, do you understand? Mandible, that is your lower jaw. So interiorly at the base of, where is it? At the base of mandible, and posteriorly at 4th cervical vertebrae. ठीक है जी? Okay. Posteriorly at the 4th cervical vertebrae. Right? Posteriorly at 4th, at the position of 4th cervical vertebrae. So that's what we discuss. Now, sternum, that is the breastbone, that is 1 in number. Vertebral column, having vertebrae, you know that. After fusion, 26, before fusion, 33. Right, in embryonic stages. So that is what we have to start first. Then we will go for the appendicular skeleton. ठीक है जी? So this colored part here is your axial skeleton. ठीक है? So let's start with the cranium part first. Then you can calculate the bones. जैसे as per NCERT total 22 they have mentioned, but later on they say hyoid bone, ear ossicles are also the part there, right? Hyoid bone and ear ossicles are also the part there. That is how they divide. So cranium mein I told you about 8 bones are there, bache, right? All the bones, they are fused. So here you have to remember one joint. You know na, these bones are fused. We call it as sutures, right? It is the fibrous joint. It is the immovable joint, right? It is immovable. Of course, we need in the case of our skull immovable joint only. So when it is the cranium, beta, so it is frontal, parietal, occipital, temporal, sphenoid, ethmoid. So frontal 1, parietal 2, occipital 1, temporal 2, sphenoid 1, ethmoid 1. So this is what we have when you talk about the cranium. When you talk about the cranium, this is what we have, right? This is what we have, right? So you can just have a look of this diagram. You people can see this. Frontal is there, parietal is there, temporal is there, occipital is there, right? You have sphenoid and ethmoid. So they belong to the cranium. So they all are fused with immovable joints. We call them as sutures, right? Sutures. Okay, right? Now here, what else do you need to remember? I'll directly pick up the most important point. So when you talk about this occipital bone, right? In the case of this occipital bone, right, bache, Like let's say this is the bone. So this part of the skull, you know that it is attached to the vertebral column. That's why this movement is there. So it is the occipital bone which will be attached to the first vertebrae, that is atlas. So when you look at this, this bone will have these condyles, right? So here in this chapter, we will use the word articulation. What is the term that we will use? Articulation. Matlab this one part will attach with another part. Right, bache? Right, bache? So here two occipital condyles are there. Right? Two occipital condyles are there. How many? Two occipital condyles are there. Okay, bache? So that is why our skull is dicondylic. That is why the skull is dicondylic. And in this occipital bone, here, what else do you have here? Here you have this aperture, yeah, opening. This is foramen magnum. Right? So the spinal cord, basically from this part, this hindbrain, the part of hindbrain will continue with the spinal cord. So this is foramen magnum. This is foramen magnum. This is the one thing that you have to remember, right? This is the occipital bone where you can, right, which you can revise again. So other than that, there is the sphenoid bone that we have discussed now. In the case of sphenoid, S for sphenoid, S for sella turcica. Beta, in that particular bone, right, there is a depression which we call as sella turcica. It is the home of your pituitary gland. That is also important. That is the home of your pituitary gland. That is also important. Is that clear? What is that? That is the home of your pituitary gland. Is that clear? Which bone are we talking about? We are talking about the sphenoid bone. We are talking about the sphenoid bone. So S for sphenoid, S for sella turcica, and yes, it is the butterfly shaped bone. So directly PYQs I'm picking, okay? So sella turcica is something that you have to remember. Now, let's talk about the facial bones. So, dekho, nose, nasal, right? Total 14 facial bones are there. At least remember the number that they are total 14. So, we have two nasal, two lacrimal. Lacrimal is here. Just we have the lacrimal gland, tear gland. Here it is. Lacrimal. Palatine is also two. Zygomatic, our cheekbone here. It is the zygomatic. Then inferior nasal concave. Maxilla is your upper jaw. Mandible is your lower jaw. So here in the facial bone, rest everything is paired. You just have mandible. So it is the lower jaw. Your lower jaw is made up of one bone. And let me tell you, this mandible is the only movable bone of skull. Only movable bone of skull. What's that? It is the only movable bone of skull. Is that clear? Is that clear? And it is the vomer that is single. That's it. So the only movable bone of skull is your mandible. It is your mandible. It is your mandible. So now I told you about the position of the okay, this is the of the hyoid bone, right? You people can see this. This is your hyoid bone. This is your hyoid bone. Take, it's U-shaped. As I said, it is what? It is U-shaped. What's that? It is a U-shaped bone. Hyoid bone, it is U-shaped bone. It is, or we can also call it as horseshoe-shaped bone. Horseshoe-shaped bone, right? So this is the, this bone, what is special about it? It is not articulating or attached. It is not articulating with any other bone, right? With any other bone. So it is attached with the help of ligaments. It is not articulating with any other bone. It is just attached with the help of ligaments. So ye dekho, frontal, parietal, right? So this arrangement, and this is the hyoid bone that we have, right? So this is the hyoid bone, larynx, and this is the hyoid bone. So it is not articulating with any other bone, right? With the help of ligaments it is attached. Is that clear? Hyoid bone clear? I don't know why, but I always forgot to add the ear ossicle. So ear topic is deleted from our syllabus, bache. Ear topic is deleted from our syllabus. But yeah, this is again important. Ear ossicles. So where do you see them? Of course, they are in the middle ear. They are in the middle ear. What we have to remember? We have to remember here, Miss has. What we have to remember here? We have to remember here, Miss has. So M stands for malus, I stands for incus. M stands for malus, I stands for incus, S stands for stapes. So malus, incus and stapes is there. Malus, incus and stapes is there. So this stapes is what? It is the smallest ear ossicle, or even it is the smallest bone of our body. It is the smallest ear ossicle and smallest bone. So malus, incus, stapes. So Miss has, H is hammer shaped, anvil or stirrup. So it is regarding their shape. So this thing you have to remember, that it is the smallest bone. Which bone it is? It is the smallest bone. Which bone it is? It is the smallest bone. Is that clear? Right? Smallest ear ossicle or the smallest bone it is. Check. Smallest ear ossicle or it is the smallest bone. Got it? Yes, everybody. Is that clear? Is that clear? Take. So ear ossicle. So this is all about the skull part. Right now, you have the sternum. When you are talking about the sternum, it is dagger shaped. It is flat. Right. So how can we revise it? Here, the most important thing is, firstly, let's discuss sternum and ribs. I think na. No, I have added the diagram. I forgot to add the diagram. That is very good diagram. But still, this is the sternum. So I cannot even teach you extra because of the time constraint. So you people can see this. This is the sternum part. Right? Side view. This is the sternum. Okay, this is the sternum. Firstly, let's have a look here, then we'll discuss the another part. So as I said, it is dagger shaped, flat bone it is, right bache, having the manubrium, body and xiphoid process, but that is not important. Look at this head wala part here, manubrium of this sternum. So here you will see first rib and look at the clavicle here. First pair of rib is attached to this manubrium, rest is to the body. Okay, we'll discuss it. So this is the sternum. Now, from this part, yes, my dear students, one question can come from the ribs. One question can come from the ribs. Okay? Right from the ribs. Okay. So, when it comes to the ribs, you know that there are 12 pairs. So, 12 pairs matlab total 24 they are. 12 pairs matlab they are total 24. 12 pairs matlab they are total 24. They are flat bones and they are bicephalic. This is the word. All the ribs, they are bicephalic. So whenever you talk about the ribs, now one question can for sure come from this part. One question from joint, one question from this part. Two questions this year they are expected. Minimum 2 questions are expected from human skeletal system only. Right? From human skeletal system only. So I just told you, you know that these are the ribs. Literally we have the ribs. So these ribs, ventrally, they are articulating with sternum, right? And dorsally, they are articulating with what? With vertebral column. Are you getting my point? With vertebral column. Are you getting my point? So these ribs, I told you, they are what? They are bicephalic. They are bicephalic. Bi means 2. Cephal means head. Right? Bi means 2. Cephal means head. So this bone, this flat bone, dorsally, it is having, let's suppose dorsally. Dorsally, it has to get attached to the vertebral column. Dorsally, it has to get attached to the vertebral column. So here it will have 2 sides, two points here, 2 sides to which it can get articulated with the vertebral column. With the vertebral column. Is that clear? Is that clear? So that's how you have to remember. Okay, that is how you have to remember. Is that clear? Is that clear? Bicephalic. So here we say, so 1 to 7 pairs, 8th, 9th or 10th pair, or 11th or 12th pair. Beta, this is true ribs because they are vertebro-sternal. Matlab they are attached to the vertebral column, particularly to that thoracic vertebrae and to the sternum. They are known as false pair of ribs. Why? Because dorsally it is attached to vertebral column, right? Dorsally it is attached to vertebral column, but ventrally, right? Ventrally it is not. Right, so it is basically what? Ventrally, it is not attached to the sternum directly. With the help of hyaline cartilage, also the costal cartilage, indirectly they are attached to the sternum. So this pair is known as what? It is vertebrochondral. It is vertebrochondral. What's that? It is vertebrochondral. ठीक है बच्चे? So they do not articulate directly with the sternum, but join the, right? Ventrally do not articulate. So one question can come from this part. Directly with the sternum, but to seventh cartilage, sorry, seventh ribs with the help of hyaline cartilage, also known as costal cartilage. So 11th or 12th, they are vertebrochondral. They are just the vertebral ribs. They are the floating ribs. They are the floating ribs, also known as vertebral ribs. They are. Take so it is bus dorsally articulated with the vertebral column. That's it. This is important. This is important. बाकी ये देखो. These ribs are also here also. It's not like that the hyaline cartilage is not helping. It is helping. And remember, the first pair of ribs, it is articulated with this manubrim, right? Baki costal cartilage or the hyaline cartilage is helping these ribs to get attached, but the 8th, 9th, 10th, they are indirectly attached because they are attached to the 7th only, right? So it is like that. So 11th or 12th is the floating pair which is protecting the kidneys, right? 11th or 12th is the floating pair which is protecting the kidneys as well, because kidneys, you know na? Kidneys, do you understand? Do you understand what kidneys are? No, madam, you don't. Madam, we don't. So this part is important. Okay, this part is very, very, very important, I would say. Now, देखो. Firstly, let's discuss the vertebral column. Vertebral column. So it is CTLSC. What's the trick here? CTLSC. What's the trick here? CTLSC. What's the trick here? CTLSC. Cerebral, thoracic, lumbar, sacral, coccyx. CTLSC. Come to lunch, stay calm. Right? If you cannot remember CTLSC directly, come to lunch, stay calm. So 7, 12, 5, 5 and 4th. It is 7, 12, 5, 5 and 4th. What is it? It is 7, 12, 5, 5 and 4. What is it, bache? It is 7th, 12th, 5, 5 and 4. That is what you have to keep in your mind. Again, I'm repeating. 7th, 12th, 5, 5 and 4. This is what you have to keep in your mind. Take. So here this 5, this bracket, it is showing fusion. So after fusion, it will form one bone. After fusion, it will form one bone. So that is why I mentioned in the starting itself that before fusion, it will be 33. But after fusion, it will be 26. After fusion, it will be 26. Is that clear? So after fusion, it will be what? 26. So it is CTLSC. It is CTLSC. It is CTLSC. So these are the vertebrae. And now you know that how we switched from quadrupedal to bipedal locomotion because these curves, right? Just a lumbar hai. These curves, because of that we are having the bipedal locomotion. But here that's the point that you need to focus. Ye dekho. In between the vertebrae. Firstly, focus here. There will be intervertebral disc. That is also a PYQ. Right? What will be there? There will be intervertebral disc. And let me tell you, that is also only in mammals. Matlab, it's a feature of mammals only. It is a feature of mammals only. Is that clear? Is that clear? So it is fibrocartilaginous, present between the centrum of the vertebrae, which act as a shock absorber. It is fibrocartilaginous, so it is present in between the centrum of vertebrae and acts like a shock absorber. Take, acts like a shock absorber. ठीक. So when you talk about the vertebrae, so the first vertebrae, what is the name there? That is also atlas. Second one is axis. So occipital condyle articulates with this atlas one, ठीक है. So first vertebrae, second vertebrae, first vertebrae, second vertebrae. Is that clear? Pakka? Sure? Vertebrae. ठीक. Done. Cervical, thoracic. So they are 7, right? They are even the right. Chalo, filhal, you write it. Then we'll discuss further. So vertebrae, first is atlas, second is axis. So now when you look at this, right, when you look at the vertebrae here, you can see that. So in this vertebrae beta, right, this central hollow portion where it is the vertebral foramen, it is also known as neural canal, right? So through which spinal cord will pass, neural canal. And when you look at the body, vertebral body, or we can also use the word centrum here, right? We can also use the word centrum here. So number of cervical vertebrae 7 in almost all mammals, including the human being. First is atlas, second is your axis. Remember that. So this hollow canal, vertebral foramen or neural canal, it is the hollow portion, right? The spinal cord will pass through it. Okay, the spinal cord will pass through it. ठीक है जी. So cervical and lumbar, these are the curves ना, which are directed forward. जैसे ये देखो, cervical, lumbar, they are the one. These are the curves. These curves in the vertebral column will help in the bipedal locomotion. So ये देखो, these are the curves. ठीक है, they are directed forward, right? Sacral and thoracic, they are directed backward. So this lumbar curve is helping in the erect posture. Is that clear? Is that clear? Right? Erect posture. ठीक? चलिए. Let's move to the appendicular skeleton, which I personally think is very, very, very easy. Okay. How many bones do we have in appendicular skeleton? How many bones do we have in appendicular skeleton? People, please be quick. How many bones do we have in appendicular skeleton? 126 bones. Take. Okay, my bad. Huh. Limb or girdles. So girdles में pectoral, also known as shoulder girdle, pelvic, also known as hip girdle. ठीक? So pectoral girdle is having clavicle, also known as collarbone. It is having scapula, shoulder blade. Pelvic, hip bone. 2 coxal bones are making it, which is also known as innominates. Take. So 2 here, 2 here, 2 coxal bones, which are made by the fusion of 3 bones. We'll talk about it. So girdles may you have total 6 bones. Okay. Shoulder. This shoulder girdle will help this forelimb to attach to the axial skeleton. Okay. This hip girdle will help this hind limb to attach to the axial skeleton. So total in girdle 6 bones are there, right? A total in girdle 6 bones are there, right? Pectoral girdle, clavicle, scapula, collarbone, the 1, 2. This is also known as the beauty bone. They are here behind scapula, shoulder blade. Take then 2 coxal bones. So this coxal, each coxal bone is made by the fusion ilium, ischium, pubis. These 3 bones, when they fuse, they form each coxal bones. So look at the spelling of the ilium as well. Take. Now limb. Forelimb. 2 forelimbs are there, 60 bones. 2 hind limbs are there, total 60 bones. So forelimb mein you have humerus, radius ulna, ya radio ulna. So this ulna U in Hindi is ungli. This ulna is towards this side and this radius is towards the thumb side. Then bache wrist, right? Wrist bone. We'll call them as carpals. Palm bones, metacarpals. Here, these are the phalanges. So, humerus 1. Radius ulna, matlab one radius, one ulna. One radius, one ulna. Carpal, again, here, 8. Metacarpal palm bone, 5 in number. Now for phalanges, there is a formula. 2, 3, 3, 3, 3. It is 2, 3, 3, 3 and 3. 2 here, 3, 3, 3 here. So total 3 plus 3, no, that's right. So it 14 now. So ultimately 1 into 2, 2 into 2, 8 into 2, 5 into 2, 14 into 2. Matlab 2 here, 4, 16, 10, 28. Matlab total number of bones. 38, 40 or 20. That's it. That's it. That is what you have to remember. Now hind limb, you have femur. Again here, tibia, fibula. Femur is the strongest bone of our body. So here it is 1 into 2. Here it is 2 into 2. Then of course, here you talk, here bache, we also discuss the patella. It is also known as kneecap. It is 1 in number. Matlab 1 into 2, matlab 2. Right? And this is a sesamoid bone. That is also PYQ. Then after that, tarsals. Now see, rest everything is same, but here you will discuss one extra patella. Patella, knee, right? Knee. So tarsals, they are 7. 7 into 2 will be 14. Then metatarsals. Uh, 5 into 2, 10. Phalanges again. 14 into 2, that is 28. Right? So firstly you take a look of that, then we will discuss it further. Chalo, write down. Done? Everybody clear? Sure? Which count is not clear? Ye dekho na. How many forelimbs do we have? 2. One humerus, total 2. One radius, one ulna, one ulna. 2 plus 2, 4 it is. Right? Then carpals here, 8. Wrist bones, 8. 8 here, 8 here. Total 16. 5 here, 5 here. 5 twos are 10 here. So 2 phalanges here, 3, 3, 3, 3. Right? Total 28 it is. Now, femur, this femur, then tibia, fibula, then patella. Here you have kneecap, right? You have kneecap. This is known as kneecap. This patella is known as kneecap. What type of bone it is? It's a sesamoid bone, right? It's a sesamoid bone. So when you are saying sesamoid bone, it's a very small bone. It is a very small bone and it is embedded there in the tendons or muscles there. It will be embedded there in the tendons or muscles there. ठीक है जी? Okay. Right? Right? So they are very important for the joint stability. So this patella, kneecap is the example. In the wrist also, in the foot also, we have the sesamoid bone, but this is what you have to remember. Right? Right. It helps in elevating the bone as well. So I am saying that ungli is this is the little finger. In Hindi, we call it as ungli. Fingers, we call it as ungli. So ulna, ungli, you can relate. This little finger, ungli, relate. Right? That is what I'm saying. Nothing else. Right? So this count is also easy. This count is not at all difficult. Right? So So firstly, let's have a look. ये देखो. So you have humerus, you have radius ulna, carpal and all. Now this humerus has to attach with what? With the shoulder girdle, right? It has to join with that, right? We will discuss the girdle part also, but firstly you mark it. ठीक है जी. So girdle में I told you clavicle, which is also known as collarbone, is there, right? Also known as beauty bone. Here we have na this beauty bone. I'll show you the picture. And scapula is a large triangular flat bone situated in the dorsal part of the thorax here, right? And it is in between bache second and the seventh rib. Matlab it is here, this scapula in between seventh and second rib. Clavicle is towards this side. Now clavicle and scapula, they need to articulate as well, right? They are making the shoulder. So even the clavicle and the scapula, scapula, they need to join, right? So they are also basically with the scapula, even the humerus will join and even your clavicle will join. Okay, right. So we need to talk about what? Dekho. So this dorsal flat triangular body of scapula is an elevated ridge, right? And that is basically the acromion, right? So basically scapula is having the elevated ridge, the acromion process. So with this acromion process, your clavicle will articulate, right? Your clavicle will articulate. And in the scapula, bache, we have the cavity, we have the glenoid cavity to which your humerus will articulate. Okay, right? So ye dekho, the clavicle articulates with this acromion process. Below the acromion there is a depression, there is a cavity, glenoid cavity, to which the head of humerus will get fitted. Is that clear? We'll show you the better pictures, but that is how you people can revise. But now I don't have the time for the break. Okay, you do it please. You do it please. We'll take the break at 5. We'll definitely take the break. Right, at 5. So this part clear. So humerus, radius ulna is there, carpal, metacarpals and phalanges. So it is the scapula that is having acromion process to which clavicle articulate. It is having the glenoid cavity to which humerus articulate. Again, it's important, right? Again, it's important. Okay. Okay. Now, pelvic में 2 coxal bones are there, and I told you ilium, ischium, and pubis. When they fuse, you will get it, right? So when they fuse, they will form the cavity called acetabulum, and your femur will articulate there. Your femur will articulate there. ठीक है जी? So now, one thing that you have to understand when you are talking about the pelvic girdle or the hip girdle. In the hip girdle, the vertebral column is also, matlab this is also, the shoulder girdle is not articulating with the vertebral column, but this hip girdle is articulating with the what do we call it? It is articulating with the vertebral column, and here these 2 bones are joined, and here you have pubic symphysis, right? So 2 half of this pelvic girdle, they'll meet ventrally to form the pubic symphysis, which is the fibrous cartilage, right? Which is the fibrous cartilage. So let me show you the pictures. Look at the clavicle and scapula part first, right? So clavicle and scapula, what will be there? There will be the acromion process. What will be there? There will be the acromion process. Understood? Understood? Take. Done? और ये देखो, this is the deltoid ridge here in the scapula. Deltoid ridge here in the scapula. Okay, so this is the pelvic girdle. Baki, Rendo, I'll show you this, otherwise you'll cry. These are the carpals, metacarpals. These are the phalanges. 1, 2, 3. 3, 3, 3. Here also there is a very small sesamoid bone. Take. So ye dekho pelvic girdle. See this, it is articulating with the vertebral column as well. And this is that fibrous cartilage, pubic symphysis. In the female, you know, na, pelvic girdle is broader because they have to carry the baby, they have to give the birth to the baby. Okay, so that's it. That's it. Baki, now come to the joints. Okay, beta. So we have to speed up. The questions will definitely come from this part. So you know that when we are talking about the joints, they are very important for the movement, right? Right. So locomotory organelles may definitely the joints has to be there, right? They are very, very, very important, isn't it? They are very, very, very important. So study of joints is known as, anybody here in the class who's aware of it? The study of joint is known as arthrology. What is the study of joints? What is the study of joints, people? It is known as arthrology, right? Study of joints is what? It is the arthrology. Okay? So basically when you are talking about the joint, there are three types. Then I will read it NCERT to NCERT. First is your immovable joint. Fibrous joint. Right, first is what? Immovable joint, fibrous joint. It is the fixed joint and it is also known as synarthrosis. It is also known as synarthrosis. So examples are important here. So it is the fixed joint. So jaise I told you about the sutures, ठीक? About the sutures, right, bache? So cranium mein bones, they are just attached to each other, right, end to end with the help of dense fibrous connective tissue, right? So here you will see cranial bones with dense fibrous connective tissue, they are attached now. So we call it as suture. They are the sutures that we have there, right? What do we have there, beta? We have the sutures here. What do we have here? We have the sutures here. So skull bones, they are attached end to end with the help of dense fibrous connective tissue. Then is imperfect cartilage. Next is imperfect joint, cartilaginous joint, slightly movable joint. Right, so like here it is synarthrosis. So this one, because it is cartilaginous, so you will call it as synchondrosis. Take. So which joint it is? It is having the slight movement. Okay, ji? Slight movements are there. Okay, so here when we discuss about the examples, so you have that intervertebral discs, you have that pubic symphysis, right? What do you have here? Intervertebral disc, pubic symphysis is there. Pubic symphysis is there. Right? What is there? Pubic symphysis is there. Okay, done. Right? So joint between adjacent vertebrae and vertebral column, your intervertebral disc we are talking about, pubic symphysis is there, and the joint in between the sternum and the ribs. Take. So look at this part. So देखो, joints are the point of contact between bones or between bones and cartilage. The force is generated by the muscle, right? The force which is generated by what? The force which is generated by the muscles, bache, it is used to carry out the movement through the joints, right? And the joints, they act like fulcrum. How do they act, people? How do they act? They act like fulcrum. How do they act, people? They act like what? They act like fulcrum. So mobility at the joints, it varies depending on the various joints. So fibrous joint, examples, done. Cartilaginous joint, examples, done. So joints between adjacent vertebrae and vertebral column, we are talking about the intervertebral disc, right? And other examples are also there. So synovial joints, right? So they are the synovial joints. Which joints are they? Which joints are they? They are the synovial joints. So they are the perfect joints, freely movable joint. It is the perfect joint. Perfect joint it is. It is the freely movable joint, beta, and it is also known as diarthrosis. What is it, beta? What is it? Hello, good morning. It is also known as diarthrosis. Which joint it is, bache? It is diarthrosis, right? So here you will see, just say, let's suppose this is the what we have. So here you will see synovial membrane, synovial fluid is something that we are going to see here, that will provide it the movement here, right? Synovial fluid is also there. So basically you know that these are the two articulating surface of bones, right, bache? So they are having this fluid. It is acting like a grease, which will obviously decrease with the old ages, right? That's why the movement in the old age, that's why the issues are there, right? And here you will see what here in the surrounding, which cartilage you are going to see, right? This one, this is the articular cartilage. This is the articular cartilage. What's that? It is the articular cartilage. Is that clear? In between synovial cavity and then having the synovial fluid. Take? Sure. So let's revise the examples from the NCERT only. So, bache, ball and articulating such an arrangement allow considerable movement. These joints help in movement in many. So ball and socket type of joint, right? Right. So first one is the ball and socket type of joint, right? So it is having the free movement in all the planes. Okay. This joint, this is the ball and the socket type of joint. Which joint it is? Yes, people. Which joint it is? It is ball and socket type of joint, right? So it is having that free movement in all the planes. So in between humerus and pectoral girdle. So is it just in between humerus and pectoral girdle? No, of course not. It is in between the femur and acetabulum as well, right? It is in between femur and acetabulum. Do mention it. Right, matlab it is in between femur and the your hip joint. That is what I'm saying. Then bache, hinge. Jaise laptop is having the hinge na, tick tick. Jaise this part. So your elbow joint is there. Your knee joint. Arey haan, one other point that I forgot. Right, ruk jo. I'll discuss. Dekho. You have femur. You have this tibia fibula, I told you, and I told you about the acetabulum as well. Here you have the patella. I'm sorry, I forgot to tell you this initially. This is the kneecap. It is also known as the sesamoid bone. So kneecap is this patella, but when you talk about the knee joint, how that knee joint will form? Firstly, in the knee joint, femur will be there, patella will be there. Now see, one female is already present, femur. So here, from this part, the tibia will be there. So these three will form knee joint. So it is not, femur is not, right? These three will form knee joint. The femur is not articulating with the fibula, right? So this is what I'm trying to say. So tibia is thicker, fibula is thinner. Tibia thicker, I just forgot to tell you this, I'm sorry. And fibula is thinner. So one is thicker, so tibia is thicker, fibula is what? Fibula is thinner, and this thicker bone, because it is bearing the body weight, so this tibia is actually known as shin bone. It is actually known as shin bone. Understood? It is actually known as shin bone. So it is the patella, which is the sesamoid bone as well. It is formed by the ossification of tendon. Right, bache? Right. Okay. So femur will articulate with what? With the coxal bone, with the patella and tibia, but not with the fibula. Okay? Femur is not at all articulating with the fibula. Right? Femur is not at all articulating with the fibula. It is saying only one female will there. Right? Two females will not be there. Shin bone se shin chan. Very good. Right? So this knee joint. Right? So this knee joint is the hinge joint. This knee joint is the hinge joint. right? And even elbow joint is also the hinge joint. Baki, this is a pivot joint. It is in between atlas and axis, right? This movement, this is a pivot joint. Okay, atlas or axis maybe, and radius and ulna will also have this. So baki gliding joint, it is in between the carpals. Okay, it is in between the carpals, and of course in between the tarsals as well. Not just the carpals, of course tarsals are also having the same type of thing, no? Tarsal. So saddle joint is between the carpal and metacarpal of your thumb. These are the examples. Thumb, right? These are the examples, right? So metacarpals or phalanges may, right? Now in the case of metacarpals and phalanges, there is ellipsoidal type of joint, not given here beta, but it is the ellipsoidal. Vrinda Krishna, it is the ellipsoidal type of joint. Okay, so our thumb is sad. So here you will see what type of joint? Saddle joint. Any doubt? Any doubt? Okay. Hanji, disorders 110% they are important. So Myasthenia gravis, this is autoimmune. Muscular dystrophy is basically your genetic disorder. Now it is because of that dystrophin gene, the progressive degeneration of skeletal system is there. The disorders of this particular chapter is very important. Myasthenia gravis, it is autoimmune disorder. In autoimmune disorder, our own body cell, immune cell will start destroying our body cells only, right? Right, so this is something that you have to keep in your mind. Okay, so this is affecting the neuromuscular junction, leading to fatigue, weakness and paralysis. We have to remember tetany bache, there is a difference in tetany and tetanus. This is tetany, which is rapid spasm, wild, right, contractions are there when there is low calcium. So any reason can be there. Say we have one hormone, parathormone. It is also known as Collips hormone. It is also known as Collips hormone. So bacha, it maintains the calcium level. If calcium level is low in our body na, it will increase it, right? It will call the bone reabsorption and all, so that calcium level it increases, right bache? So the calcium level it increases, right bache? So of course, so there are various reasons for this tetany. Vitamin D is not there, then again the calcium ions, less vitamin D will also affect the calcium concentration na? So that is how things are related. Baki beta, arthritis, again very important. Arthritis is having arthritis, this part is very important. The questions will come from this part. So basically arthritis, what is this arthritis? It is the inflammation of joints. What is this arthritis bache? It is the inflammation of joints, right? It is the inflammation of joints, right? Inflammation of joints it is, right? So here you will have different types. You will have RA, matlab it is rheumatoid arthritis. Arthritis, beta, it is again autoimmune disorder, right? It is again the autoimmune disorder. So to check it na, immunoglobulin M is checked. So it is also, the rheumatoid factor is basically a type of immunoglobulin. So here there is the inflammation of synovial membrane, right? What is going to happen here? There will be the inflammation of what? Inflammation of synovial membrane is there, right? Inflammation of synovial membrane is there. Take inflammation of synovial membrane is there. Okay, ji? Okay. Then you have gouty arthritis also. Gout. So uric acid here. There will be the, right, this is what? This is the gouty arthritis, right? This is the gouty arthritis, right? So it is due to the excess formation of uric acid, right? Or its inability to excrete, or inability of body to excrete it, right? Or body is not able to excrete it out, right bache? So it will get deposited in the joint, this uric acid. Then osteoarthritis. Osteo mane bony, right? The word is osteoarthritis. It is degenerative, and here what is going to happen? There is the degeneration. Now you know what articulate cartilage is. It is the degeneration of articular cartilage. It is the degeneration of articular cartilage. Is that clear? Is that clear? Articular cartilage. Okay, so it is age related, right? Of course, so it will affect spine, knees, and hand. And there is also the proliferation of new bones. ठीक, sure. Chaliye. So this is about this chapter. Skeletal part done and dusted. Do revise it. You can take the help from the top 50 concepts topics, because there also I have picked up the most important topic. The remaining topics from this portion I have discussed there, right? So you people can take the help, right? I'm picking up the topics which we were unable to discuss in the recent times, okay? Baki ye dekho beta, muscles. Jaise I told you na, structural organization wala part, it will be discussed here. So overall in the case of muscles, let me give you the quick idea, but yeah, we have discussed that part also. Okay, we have discussed that part also in the top 50 concept. So when you talk about the skeletal muscle, right, let's have a quick revision of that. You remember this is the muscle, right? Do you remember this is the muscle? So this muscle will have what? This muscle will have muscle bundles. Do you remember that? This muscle will have so many muscle bundles. What will it contain? It will contain so many muscle bundles. So that muscle bundles are also known as fascicles. That muscle bundles, they are also known as. So what is the word that we have? We have the word muscle bundles. They are known as fascicles. We have the word muscle bundles. They are known as fascicles. So these muscle bundles, they are also connected with what? They are connected with the connective tissue sheath. They are connected with what? They are connected with the connective tissue sheath. And this connective tissue sheath is known as fascia. What is it, bache? It is known as fascia. Take now in these muscle bundles, what will you see? These muscles are having so many muscle fibers. Within these muscle bundles, so many muscle fiber will together form these muscle bundle. So these muscle fibers are anatomical unit of muscle. They are what? They are the anatomical unit of muscle. Now when you talk about these muscle fibers, just say, let's suppose if this is the muscle fiber, right? With the help of worst diagrams I'm going to teach you, but trust me, they are worth reading. So here the outer covering will be sarcolemma, right? And here this is the sarcoplasm, and you will see these parallel myofibrils here. These myofibrils, the way proteins are arranged on it, will give it the striated appearance. So this is the sarcolemma, right? And at places you will see this sarcolemma is invaginated, it is folded also, right? This is sarcoplasm here, and the endoplasmic reticulum here will be known as sarcoplasmic reticulum, right? So this sarcoplasmic reticulum, my dear student, is the storehouse of calcium. It is the storehouse of calcium. What's that? It is the storehouse of calcium. Okay? These are the muscle fibers, and of course they are the multinucleated one. Syncytial it is. So it is multinucleated one. It is syncytial, right? Multinucleated one. Okay? So they are having the myofibrils. So let me show you the diagram here. Ye dekho. This is what we have. This is what we have. We have the muscles here, right? So you know that bones to bones, that's again an important part. I told you, structural organization will cover like this. Bones to bones, who's going to attach it? Ligament, ligament. Yes, bone to bone, who's going to attach it? Ligament, ligament. And what about bone to muscles? What do you have then? You will have the tendon then. You will have the tendon then. So just look at it. Muscle bundles. What is the word that we use there? Fascicles, right? This is the ultrastructure of skeletal muscle. So here these muscle bundles are having so many muscle fibers, right? When you look at these muscle fibers, so what will you see? Myofibrils. So there you will see the alternate dark and light band because of the arrangement of your myosin and actin. Myosin and actin. Take care, myosin and actin. So just look at it. Just look at it. Bone, then tendon, muscle is there, right? So that's how the arrangement is. So look at the myofibril. So when you look at the myofibril, what do we have in the myofibril there? Alternate dark and light band. So what do we see there in that alternate dark and light band? ये देखो, I, actin. So basically on the myofibril, what do you have? I band, alternate I and isotropic. I, actin, thin filament it is. You have A band, anisotropic, right? Or it is dark band, arc. So I, actin, and it is light band, I. Actin it is having, and this is the dark band, and here you will see myosin, right? Thin and thick filament. So bache, basically what is going to happen, just say let's suppose I band, A band, I band, A band, I and A band. That is how it is. That is how it is. So you will see in the case of I band, it is bisected by a Z line. So when I'm using the word bisected, means I band will be divided by into two halves by the Z line, right? I band will be divided into two halves with the Z line. So this Z line is what? It is the elastic fiber. This Z line is what? It is the elastic fiber, right? It is the elastic fiber. Now, right? Now here you will see the dark band. When you talk about the dark band, just in our dark phase, our friends are with us. Our friends are with us. No doubt, normally we say that in the I band actin is there, and then the dark band myosin is there. But along with myosin, you also see the actin. In NCERT, it is not mentioned, but also see the actin, right? You also see the actin here. Okay. So now when you look at this part here, from the I band, from the A band, from the center, there is one M line. Right, just say this is the elastic fiber, right? So M line is there. Now when you look at this dark band, na, so in this dark band, ye dekho, this part is the H zone. This part is the H zone, right? What is this H zone? This is the Henson zone. This is the Henson zone, right? It is the part where myosin is not overlapped by actin. It is not overlapped by actin. Take, not overlapped by actin. And in between, M line is there, which is thin fibrous membrane, which is holding all of it together, right? It is the thin fibrous membrane, right? And here this, you know, this myosin, thick filament, it is also attached to this Z line. And how? Because there will be, this is the titin protein. So now, in between these two successive matlab one after another. So in between these two successive Z lines, what do you have? This is the basic unit of contraction, right? That is the sarcomere. So when you talk about the sarcomere, it is the basic unit of muscle contraction. So in the case of sarcomere, what do we discuss? In the case of sarcomere, my dear people, we say that there will be two half I band and one A band is there. Take two half I band and one A band is there. Two half I band and one A band is there. Am I clear? Sure? Sure? Take, so this is what sarcomere is. This is what sarcomere is. So it is the basic unit of contraction. So here in this part, again I'm repeating in top 50 concepts. Okay? Again, I'm repeating people that in top 50 concepts, right? That in top 50 concepts, we have revised it, right? We have. Hello guys. Somebody please focus here. 2 half I band and 1 A band is there. One A band is there. So please focus here. Baki ye dekho, this is how the arrangement is. Ye dekho, at places, your sarcolemma, it is invaginated. So when it is invaginated, right, it is forming the T tubule. So on the either side of this T tubule, na, you have sarcoplasmic reticulum. And you know that the nerve fibers are going to innervate this muscle fiber. So basically the junction at this nerve fiber and muscle fiber, the junction here is known as neuromuscular junction. What's that? It is the neuromuscular junction. What's that? It is the neuromuscular junction. It is also known as motor end plate, right, bache? It is also known as, right, motor end plate. So the signal will pass here, right, the action potential will pass here, sarcoplasmic reticulum will release the calcium ion, that will bind with the troponin, that will slide the troponin, tropomyosin will unmask the sites on the actin to which myosin head can bind, and then there is the cross bridge formation and everything will be there. So in the top 50 concepts also in detail we have discussed that, right? You do not forget about the enzyme, right? And here what are the most important things? Jaise agar maan lo, if I have to show you the structure here also, in the case of thin filament you have actin, then actin mein you have G actin. So that G actins, they will polymerize, they will form F actin, filamentous actin. So two F actins, they will helically coiled like this, and then tropomyosin will run throughout the length of this actin, will cover it. Actually in the G actin, like suppose this is the G actin, you have the site to which the myosin head can bind. Then at places you have troponin as well. So troponin is a trimeric polypeptide, bache. So it is having 3-part troponin. It is troponin tick. So troponin T is there, troponin I is there, troponin C is there. So this T will attach to the tropomyosin and to the other two components because it has to make sure that tropomyosin will not allow the tropomyosin covering the actin properly, so that you know myosin head cannot bind with that. Number one. Then comes the troponin I. It will inhibit their joining na, and C will bind with the calcium when there is a signal for muscle contraction, right? So basically that's how they polymerize and form. And here you'll see meromyosin. So many meromyosin will polymerize to form myosin filament. So meromyosin mein you have HMM, heavy meromyosin, you have LMM, light meromyosin, and heavy meromyosin mein beta you will have this globular head plus short arm in heavy meromyosin, and light meromyosin mein you will have tail. So this globular head will be it is an active ATPase, okay, active ATPase. So in I think multiple times, even in the NEET end series, even in top 50 concepts, right bache, I have discussed it multiple times with you all, right? Hope you guys remember, hope you people remember, right? I have discussed it multiple times with you all. है ना? नहीं? नहीं? और क्या? So from there you can revise, but yes, one point that I would like to mention here, देखो, first of all, these are the changes that are going to take in the muscle contraction, and that's directly a PYQ. Indeed, that is a PYQ. Okay, indeed, that is a PYQ. Okay, bache? So length of I band will get reduced. It is not the length of actin or myosin that will get reduced. No, it is the length of I band that will get reduced. Length of A band will remain same. Length of sarcomere will decrease. Length of myofibril will decrease. Henson zone will disappear. So from these statements, PYQs are there. So of course, when there is a sliding, right, the actin, the number one question is actin will slide over the myosin. So when they will slide, Henson zone, where only myosin is there, not overlapped by actin, it will not stay. Henson zone will disappear. So length of I band gets reduced, but the length of A band will remain same. Length of sarcomere will decrease. Length of myofibril will decrease. Henson zone disappear, and actin and myosin filament will remain same here. right? Their length will remain same here. So PYQs, revise it. Baki here, one more thing that you have to remember here is bache, that when they go globular head is an active ATPase, when it joined with the actin, we say the cross bridge form. So when, right, ADP and P, right, matlab here the ATP, when the ATP gets attached to the head, the cross bridge breaks. Again a PYQ, the cross bridge break, right? Why? Because this globular head, it has to break the ATP, then that energy, ADP and phosphate, that energy will help this myosin head, right? It will help this myosin head to bind here and to form the cross bridge, right? And this is how the sliding will occur. So breaking of cross bridge will occur when the ATP will bind here. When the ATP will bind here. I is isotropic, A is anisotropic. So what is going to happen? Nerve impulse will arrive at the axon terminal of motor neuron. It will trigger the release of acetylcholine. Now beta, this acetylcholine will leave this gap. Now you have studied the neural control and coordination, so you must know this. So this acetylcholine will diffuse across here, right, and it will trigger the action potential in the muscle. After that, acetylcholine esterase, which is present here in this gap, it will break the acetylcholine and this neuron will take it back. Okay, then bache, when action potential will reach here through the T tubules, what will happen? The surrounding sarcoplasmic reticulum, they are the storehouse of the calcium, they'll start releasing the calcium ion. So sarcoplasmic reticulum, which is basically the storehouse of the calcium, it is basically the smooth endoplasmic reticulum, bache, right, that is forming the sarcoplasmic reticulum. So this calcium ion, right, these calcium ion and the sarcoplasmic reticulum, they allow the calcium ion to be there in the sarcoplasm. So you know that what will happen to these calcium ion? They will join with the troponin C. When they will join with the troponin C, they'll expose the binding site for the myosin, because when they will join with troponin C, it will slide, tropomyosin will go away. So the myosin binding site on actin will get exposed. Okay? The site will get exposed. Are you getting my point? That site will get exposed. After that, what's going to happen? You know that after that, the contraction is occurring. Power stroke will be there, ATP will be used, so myosin head will bind to actin, right? Then the it will swivel and release thin filament, it will pull towards the center of the sarcomere. Simple, right? Then again back to basic. Then calcium release channels, matlab sarcoplasmic reticulum will again close, calcium will again enter, and you know that again the relaxation will be there. So this diagram, this diagram will explain it wonderfully, right? So ATP, when it binds to the head, cross bridge breaks. ATP when binds to the head, cross bridge breaks. That's what you have to keep in your mind, okay? Take okay. So ये देखो, actin filament, tropomyosin, calcium ion will bind with it, it will slide. So troponin calcium complex will pull tropomyosin away, exposing the myosin binding site. So that's how the cross bridge will break. Right form, when ADP and P are bound to myosin, as myosin head attaches to the actin. Okay? Okay, then this point, so ADP and P, they release, cause head to change position and actin filament to form. So ADP and P, when they are bound to the myosin beta, when they are bound to the myosin as myosin head attached to the actin, right? They are bound to the myosin as myosin head attached to the actin. So ADP and P, they will release, they'll cause the head to change position and actin filament to move. Okay, so binding of this ATP will cause the myosin head to return to the resting position. So that's how it is. Okay, yes, done. Sure. Good morning. So all the important topics we have covered here, don't worry. Okay, in top 50 concepts, all that urine formation, globular filtration, all the topics are done and dusted. Okay, so do not forget to revise what GFR is, right? This type of question, that 125 ml per minute, the amount of blood filtered by the kidneys in per minute and per day, right? These are the kind of questions that can come here. Take, so do not miss that. And in the counter current mechanism, if you people remember the story, from one side it will enter and leave. Just remember that. Baki, Beta here, right, no doubt, if you are thinking that ma'am will just discuss this part and will finish the chapter out, no, it's not like that. It's not like that. But yeah, I'm picking up a very important topic that I believe from where the question can come. You know that enzyme, almost all enzymes are protein, right? I'll touch this part in the next class also, right? In the botany part also, I will touch this part. It is not your botany and zoology, it is the complete biology now, right? It is the complete biology now. So I am just touching a very important topic, right? Right, so I'll touch it, I'll discuss it with you all, and all the important point from this part we'll discuss, and then, right, rest we will discuss in the next class, don't worry, right? Whatever is left, it's not like that I will not discuss it now, so we will discuss. Dekho, these 15 marathons will complete the syllabus, then we will have the quick marathons, complete syllabus marathons as well. In that complete marathon syllabus, what are we supposed to do in that complete marathon syllabus, bache? We will just do that thing na, quick revision. So we will pick up the most important things, we will solve the questions with the help of PYQ. So whatever is left in one part na, that will be covered in the next. So we are here for you only, okay? We are here for you only. It's not like that ki we'll leave something, we will not. We have to, we don't have any choice. You can watch my lectures on Unacademy NEET English channel. You'll see all the chapters are covered in detail. And when I started the Game of NEET 2.0, I picked up the chapter that we missed in Game of NEET. Okay? So we are here for you. And even if we change the name of the series, by content we have to deliver, na? Content we have to deliver. So are you aligned na? Are you aligned? Ki Hanji ma'am, it is true that you are teaching everything. You're not leaving anything. है कि नहीं? So good boy. So you know that almost all enzymes are proteins. Do we know that? Yes, ma'am. Do we know that? Yes, ma'am. Almost all enzymes are protein. Almost all enzymes are protein. We know that sometimes nucleic acid, they also act like what? They also act like enzymes. We call them as ribozyme. Now when we talk about these enzymes, they are simple. Simple means they are just made up of protein only. Just made up of protein only. Like trypsin is there. Like what? Like trypsin is there. What is there? Trypsin is there, etc., etc. There are certain enzymes which are known as compound enzymes, or they are also known as conjugate enzymes. Now in these enzymes, we have the protein part, bache, and we also have non-protein part. The protein part is there, and along with that, non-protein part is also there. So in the case of protein part, it is known as apoenzyme. Non-protein part, it is known as cofactor. So if we want this enzyme to be functional, okay, this compound enzyme to be functional, so holoenzyme it will be. It will become holoenzyme. Matlab, it has both. It should have apoenzyme plus cofactor. It should have both. Apoenzyme plus cofactor. It should have both apoenzyme and cofactor. Is that clear? Is that clear? Is that clear? Are you sure? Are you sure? So this cofactor is having three things, right? This is the direct topic from the enzymes. Three things. First is prosthetic group. Second is coenzyme. Third is metal ion. Third is metal ion. Are you getting my point? Third is what? Third is metal ion. So first part is the prosthetic group, then it is the coenzyme, then it is the metal ion. Prosthetic group is also organic, but the point here is it is tightly bound. That's it. That's it. That will make the difference here, tightly bound. This is also organic, but this is organic, but it is loosely bound. It is loosely bound. ठीक? right? That's what you have to remember. Tightly bound, loosely bound. Just say niacin, vitamin, that is what we discuss here. Prosthetic group, right? Heme and all. Do remember that. That is important. Baki metal ion, you know that this is zinc for carboxypeptidase. Magnesium is required for all the enzymes of glycolysis. So these are the examples. So one question from the enzyme part will come from this side. Okay, one question directly. Directly, I'm giving you this. Baki classifications of enzyme में again we have the tricks. It is O, T, H, Lil. It is O, T, H, Lil. When we classify them, what do we have, people? It is O, T, Halil. When we classify them, what do we have, bache? We have O, T, Halil. O, you know that oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. Repeat it with me. Repeat it with me. Oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. Oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. Oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. Okay. 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 OT Halil. OT Halil. Right. Oxidoreductase, also known as dehydrogenase. Transferase, hydrolase, it will do hydrolysis. In the lyase also, lysis is there, but other than hydrolysis, double bond will be there at the end. Isomerase will convert one compound into the another isomers. Ligase, molecular glue it is, right? Molecular glue it is. Direct question from the enzyme. I told you, now we'll pick up all the important things here. Don't worry about that. Now, nervous tissue done. Okay. बाकी बच्चे ये देखो, epithelial tissue diagrams are there, right? They are mentioned. Muscles parallelly we are discussing. Just you have to revise the examples there. Now, when it comes to the connective tissue, okay, right, most abundant and widely distributed it is. Yes, we have extra things that we can discuss here also, but let it be. Let's forget about it. Let's forget about it. Let's focus there. So ये देखो, most abundant and widely distributed tissue in the body, right? So it is having the special function of linking and supporting. Linking and supporting. So we have soft, we have liquid, fluid connective tissue, जैसे कि blood and lymph is there, right? We have the mineralized one, right? The skeletal system, जैसे bones and cartilage, also the example of this connective tissue only, right, बच्चे? So here the question can come from the diagram. The question can come that which one is the loose connective tissue. So areolar and adipose. Areolar will have the air spaces. So diagram, is it important? See, first of all, this is the adipose. The large fat globule is there. Nucleus is at the periphery. So it is monolocular, single locule, adipocyte it is. Take. And here when you look at the diagram, macrophages, phagocytotic, they are fibroblast. What type of cell they are? They are the chief matrix producing cell. Fibroblast, right? What is this fibroblast here? What is this fibroblast here? It is the chief matrix producing cell. Yes or no? Yes or no? So it is having another function also. It is having another function also, right? Most abundant or what? I don't remember. I'm asking you this, right? I'm asking you this. What is the what is this point here? Yes, I just forgot about it. You tell me now. I don't know. I don't know. When we discuss about the fibroblast, what do we discuss here? When we discuss about the fibroblast, it is a chief matrix producing cell. That is a direct point that we need to remember, right? That is a direct point that we need to remember. So other than that, is it the largest cell? Yes or no? It is a chief matrix producing cell. Of course, it is the largest and the most abundant cell of the connective tissue as well. Largest and the most abundant cell of the connective tissue as well. Is that clear? Right? So it will produce fiber as well. Fibroblast produce fiber. So collagen, that will form the most abundant protein of animal world as well, right? So collagen fibers, white fibers there. So yes, diagram is important. So mast cell, the one which will release the histamine. So diagrams do not leave this. Loose connective tissue and dense connective tissue, their examples are important, okay? Okay, so fibers are there. So collagen fibers, white fibers, elastin fibers, yellow fibers. So these fibers, they'll provide strength, elasticity and flexibility. ठीक ठीक, so loose and dense and specialized one is there. Examples are important only, just pick up the example. So loose में fibers, cells and fibers are loosely arranged बच्चे. जैसे you have areolar tissue, even you have the adipose tissue as well. So their location matters. अब dense में dense, regular or irregular both are there. So of course here the orientation of fibers will show a regular or irregular pattern. Right, so dense, regular and irregular it is. So dense regular में collagen fibers are present in rows between many parallel bundles of fibers. So tendon, you know, tendon and your ligaments are the example here. So dense regular में collagen fibers are present, tendon which will attach skeletal muscle to bone, just now I told you. Or ligament में बच्चे one bone is attached to the another, that's it. So examples here are very important, that's it, the diagram and the example. Right, the diagram and the example, dense, regular, irregular, right, bus. So just focus on the diagram as well as on the example and the cartilage part also, that they are made up of what. So chondrocyte ठीक है, but chondroclast you remember, cartilage destroying. They are kind of macrophages here, same way, osteoclasts. So do revise it. So if you will solve the PYQs from this part, that will be more than sufficient. So that's it, that's all. Rest we will discuss in the next class for sure. Right, we will discuss in the next class. Baki bache, these are going to be wonderful sessions for your quick revision. Every hormone, their function, everything is mentioned. Okay? And you know that in the description box here, because there is one day left only. Click on this link, right? If anybody is like, I'm giving the NEET examination, ma'am, just to see what kind of paper is there, but I want to join a coaching, right? I want to go for the preparation. So then do not wait for your NEET result. Start your preparation early, so right, so that you can complete your syllabus on time. So see this in the description box, there is a link and you will have the access of all our plus subscription. Matlab you can have the access of all the batches that are running across the Unacademy platform. So if on YouTube we can teach like this, so of course in my batch, when it comes to my batch, I put my everything there, right? It's that is the problem, that students, they don't stay consistent. Otherwise, so I just want, like, when it comes to my batch, right, I don't compromise it at all, right? If we have decided something that yes, we have to finish the syllabus in this time, we are going to do that. If you remember our crash course, right, we promised the student to finish the syllabus till 31st of March. It's done. The another crash course, that syllabus will be finished till 20th of April. So that's how the things are going on. So if you really want to start your preparation, you can start now. So in the description box, there is a link, bache. Baki I'll share the PDF of the content in our Telegram group. Okay, so we'll share there. Don't worry. And biomolecule cell will be discussed in the next class, and in short video sessions, short revision sessions, okay, in short revision sessions, rest like body fluid and circulation will be quickly covered there. Okay, will be quickly covered there. Take. So I have another class, right? I have my classes in my batch also. So we'll see you all, and as I said, I'll put the short videos for the quick revision. But yeah, frog and cockroach before 15th of April. Yes, before 15th of April, that's a promise. I'll pick up. Out of that, I'll pick up one topic. Either I'll, I'll complete that in one video only, or I'll put 2 different videos for the frog and cockroach. Okay? Done? Chali. So hope you enjoyed the session. If it is so, do let me know in the comment section. I will wait for your wonderful comments there, right? And see you all in our next session also. And you know that now, though this it is processing. So today your HSP sir is also going to take the marathon. So do support him as well. Okay. So if you like the session, do let me know, right? Do let me know in the next class. So next class mein cell cycle we will start, plant kingdom we will start, anatomy, right, then plant physiology, morphology, and this biomolecule remaining portion we will discuss there. Okay, chalo, bye bye, take care, thank you guys, and yes, we'll wait for your comments. Take care. And Tuesday 5:30 p.m. special class will be there. I'll ask my team to pin the link in the description box. We'll make a short as well so that you can remember that, and in the Telegram group also I'll share the link. Yes, Namita, they are important, right? They'll be sufficient enough.